We have a dedicated team of staff members that does imports from all around the world and exports into Sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP.
So good morning and welcome to day 2 of the East Africa, Central Africa, African BioGenome Open Institute workshop. It's a great day and I'm looking forward to another great day as we had yesterday. We have an exciting program and today we will begin with a presentation. Minute I open my program. So today we are beginning with a presentation from Professor Marie-Louise Avana, who is with the Africa Forest Forum Secretariat. She joined the AFF as the African Forest Forum Secretariat in Nairobi in 2018 as a senior program manager of the African Forests, People, Culture, and Climate Change. Her role is to facilitate knowledge generation, capacity building, and advocacy for better understanding on how forests and trees and people who depend on them in the various African landscapes respond to climate change and variability and strategy to better harness associated opportunities at national, regional, and international level. She comes from the University of Zhejiang, where she's an associate professor of agroforestry and forest sciences, holding a PhD in agroforestry and tree biotechnology. And as you can imagine, her interests are all to do with forest and forest ecology. So Professor is joining us online, and I welcome her to make her presentation. Prof, you can share your presentation. And I welcome you, and we look forward to your talk. Thank you very much.
Thank you very much, Prof, for giving me the opportunity. I believe you can see my presentation.
Yes, we can. Kindly make it in presentation mode.
No, I don't seem to see how to do it that way. Maybe let me stop share.
So if you go to the top, you see slide on the top, up, yeah, go left, up, yeah, go left, that on that level, left, keep going left, which is your right. No, yeah, keep going again. No. Again, no, keep going on the opposite direction.
Go to slideshow.
Slideshow. Yeah, slideshow. Look at the slide. Yeah, no, keep going. No, not that one. Up. No, up again. Yeah, keep going on the left. Keep going. No.
I've seen.
Okay, seen it. Yeah, okay.
Thank you very much for facilitating and for giving me this great opportunity. Can you hear me?
Yes, I can, please.
Yes, thank you. As presented, I'm attached to the African Forest Forum. So my presentation this morning will focus mainly on the forest sector, though I will be talking on biodiversity, but focusing mainly on the forestry sector. So I will start by giving you an overview of the status of Africa's forests. We know Africa's forests cover an area of nearly 700,000 acres, 700 million acres, and this represents 21% of the Africa's land mass. And what is important to note is that this forest is naturally regenerated, therefore hosts a high level of biodiversity with 30% of rainforests, and these rainforests are distributed mainly between West and Central Africa and Eastern and Southern Africa. Then the distribution of forest itself is mainly in West and Central Africa and Eastern and Southern Africa, knowing that in Southern Africa, most of the forests are planted. We also have mangroves and other woodland, and including agroforestry areas that also host a high level of biodiversity. But this Africa's forest is actually being threatened by different human activities, and Africa is actually leading in terms of the higher rate of deforestation and forest cover loss. As for the last decade, we have been losing nearly 4 million acres of forest per year, and Eastern Africa also being leading this in this process with more than 50% of this area loss. Another important aspect is to note which percentage of the Africa's forest is dedicated for biodiversity conservation, and we see that it's nearly 24%, and this 24% are distributed through 7,800 protected areas, and among these representing 17% of the total area of Africa. This land cover, if you want, you have a general look on the land cover, you see that Africa is also covered by different type of land use, and this land use goes from desert to the rainforest, as mentioned, and this type of land use, land cover also give way to different type of habitat for biodiversity. And the reclassified land cover show that the most area covered by forest is within Eastern and Central Africa. The status of biodiversity in the Africa's forest, as I mentioned, the diversity of habitat also ensure an extraordinary diversity of life. And we have for plant species nearly 73,000 plant species, but a significant proportion of these plants are being threatened by extension and due mainly to land use conversion and therefore leading to their habitat destruction. And also climate change and unsustainable extraction are playing also a great role in this biodiversity loss. The Congo Basin in Central Africa hosts actually 11,000 species of plants, including 600 tree species, and 1,100 are endemic species, meaning that they can only be found in this area in the Congo Basin, and among them 69 are already threatened for extinction. And the biodiversity of this forest, we are losing almost 800, is threatened also because we lose almost 800,000 acres of forest per year in the Congo Basin. In Eastern and in East Africa, we also have a diversity of ecosystems, and we provide habitat to nearly 2,500 plant species. So the higher traits also of biodiversity for each of the regions are varied. So you can have, for example, in West Africa, the highest trait to biodiversity is agriculture and logging. In Central Africa, it's mainly hunting and wildlife trade. And in East Africa, it's agriculture. And in Southern Africa, we are more concerned by the invasive non-native species that are used in forest plantation. So the red areas on the figures also shows area of hotspots for biodiversity. So an assessment done by the university, by the Bern University of Agriculture and Food Science also indicate that you have some, if you assess the biodiversity intactness in index in this area, it also showed that many areas, for example, in Central Africa, show approximately, we can say, fairly good rate of intactness, showing that there is some scope for conservation, but also very different spots of degradation. In Eastern Africa, you can see that it's more whitish, showing that we have more area with less than 30% intactness of biodiversity, and therefore a great trait for biodiversity loss, according to this analysis. So based on their analysis, they suggested some areas that could be considered for as hotspot, and the criteria they used were mainly forest loss, land cover, and road network that are also great drivers of biodiversity degradations. So the 10 areas proposed, we see 4 of them are within Eastern and Central African region, as you can see on the map there. So in this context, what is important is how we can ensure that the sustainable management of high-value species are mainly through domestication of indigenous species, because many of these species are used as food, medicine, and some of them biochemicals are extracted and used to synthesize drugs. And for example, there is the International Center for Cancer indicated that the they use more than 1,400 tropical forest plants for their drugs to fight against cancer. And also, you can see from the map there the number of trees that provide food of importance to smallholder in different areas of the world. There you can also see how Africa is actually leading the process. You also have here a diversity of non-timber and timber products that are extracted by people in Africa. So some of you here within the region can recognize some of the species you use, basically the African plum and other other important species that are used by the population. So this also, and we also have, for example, many of these species are providing different type of product. For example, the baobab itself only can provide more than 78 different type of products. It's only baobab. Then, and you also see from the map of Africa, different area where nuts are being produced in high quantity and used and traded by the population. You also have different plants that are of importance in terms of medicinal use. And it's also important to note that 80% of people in Africa still depending on this plant, on this medicinal or traditional plant for their health, for their primary health care requirements. And 1 billion of people also use herbal remedies to treat children diarrhea. And the market for herbal medicine can go up to 60 billion per year. So, the exposure also to the positive environment also has an impact on the physical health, physical and mental health. So the forest environment therefore provide a good opportunity for the human health care. The NTFP, this is non-timber forest products, which are products extracted from forests and that are other than timber. Also, they also provide good basis for cosmetic use. So you have growing consumer interest now for natural and other organic cosmetic products, depending on forest species. Currently, the global supply is obtained from only about 15 plant species, but more than 1 million of plants are known to be to have that interest, that can have potential for cosmetic use. We also have prospect for good market for this species, but it's also important to note that when the market for a species is well developed, it represents also a threat if there is no precaution taken to ensure alternative supply and not only depending on forests. Another case study of interest is the gums and resin species that are produced all along Africa in more than 24 countries in drier areas. We did an assessment and realized that many species from many countries provide these gums and resin. And we also characterize different types of gums and resin. And from the figure here, you can see that for some countries, these gums and resin represent a good opportunity for export market. The process of domestication, I'm trying to suggest here as a way to ensure that there is alternative supply for this forest product, is mainly to building through a chain of going from wild species to domesticated species. And we are taking here the example of maize. You can see the difference. I know I have maybe we may have on the here people who are well informed in terms of who are well knowledgeable in terms of this. But what I'm trying to indicate here is that this same process has to be replicated on forest species to ensure that our indigenous species can be improved in terms of quality, in terms of quantity, to meet the need for food and also for other products and services. And this builds mainly on ethno-ecological studies and also using multidisciplinary approaches. So the approach, for example, is building from the history of the crop development in Africa, indigenous knowledge, horticultural and genetic science, to develop, therefore, improved material from a specific species based on the interest of the population. And we have different areas in Africa where we see that different plants have evolved from wild to cultivated crop. And the domestication syndrome, going from this wild population to the cultivated one, is mainly a modification or an improvement in terms of morphological, physiological, and biochemical property of the plant to meet the desired requirement of the desired trait, the desired characteristic of the users. And along the way, there is therefore a loss of genetic diversity, because we move from the very diverse resource base to select just those who meet our interests. So the process, therefore, we have been using in the domestication process, the domestication of indigenous species is based mainly on priority setting, and this priority setting is based on indigenous knowledge, and we also have some morphological and genetic characterization that help to identify among each selected species the variety that meet that satisfy the need of the consumer, and also the requirement, the demand on at market level. Therefore, based on this selection, we can we move through production of planting material using horticultural techniques. And then the planting material can be therefore integrate and manage on farm for multiple benefit for the farmers and also for the environment. And it's also good to note that the one aspect to make sure to develop this this product is to work also on value added technologies to make sure that we gain all the benefit from this product, as shown earlier, for the case of baobab, that we can produce more than 78 products from different plant parts of baobab. So it's also important to ensure that after developing, encouraging people to invest in terms of domestication, there is a market that can absorb this product and therefore and ensure the benefit for these people. So in different area of mainly on the dry land also of Africa, we have try to have some priority species, priority product for domestication, and you can also see there, and this also guide the selection of this species for the domestication purpose. Thank you for your kind attention.
Thank you very much, Professor Marie-Louise. And we are indeed taking home the message that we are losing about a large proportion of our forests on a daily basis. And this is something that Africans, and as Africans, we need to be concerned about. And the need to domesticate plants that will be able to have a dual purpose of giving us products for commercialization, and also at the same time giving us that necessary forest cover. Professor Marie, if you can remain online, we would be happy to engage with you with a few questions. But right now, I'd like to introduce to you Professor Felix Muchie from Cameroon. He's currently an associate professor in animal breeding and genetics, and the head of department of the animal science department in the University of Dishang in Cameroon. He's also the head of biotechnologies and bioinformatics research unit, and the principal investigator of an IAEA grant. He's mainly concerned, his main research interests are animal biodiversity, conservation genetics, especially those of native species, and characterization. He's supervised many students. He has authored and co-authored about 140 papers, including 15 book chapters, 5 books, 20 manuals. Professor Felix is somebody I have worked with during my course of career, and I know his passion for conservation genetics. Prof, if you're online, we are ready to hear you, and you can share your presentation on your side. Prof. Felix, are you there?
I don't think he's on the call.
He was connected some few minutes ago.
Okay, so do you know his name? The name
Felix.
he used? The
Felix,
name he used?
F-E-L-I-X.
Yeah, I can see just two, Felix Mange and Y.
Muchie.
Muchie.
Yes, he's not, I don't think he's.
Maybe he had the. Oh.
Okay, maybe because of power issues,
yes,
dropped out. But
because he was online, he even say hi to people there. I saw his message.
No problem. But we can perhaps then listen to Professor Adi Felike from Addis Ababa University.
Okay, I think Felix is, I can see he's on the call.
Excellent. Okay. Professor Felix, are you able to go now?
You need to unmute, Professor Felix. You need to unmute.
Thank God you can ask him to unmute.
All right. Okay.
We can see you.
Yeah. Yeah.
Prof, are you able to share your presentation?
Yeah, sure, please. I was a bit, let me just check it. Where is it? I need just to.
So at the bottom of your screen, there's a green button called share screen.
Share screen. Yeah, I've seen it. I'm now trying to know exactly where the documents are. I'm sorry. Sorry to be, because it's my first time to share this thing using the machine. So Microsoft Share. But I've sent you my presentation. Is it possible to save time if someone can do it from your side? Is it possible?
Yeah, so I don't know if Paul received it. So are you at the bottom? Did you see a share button? Please?
Yeah.
Okay, can you click the share button at the bottom of your screen?
Yeah, I did it.
Okay,
And
it's not sharing.
Yeah, I'm trying to check where the document is because, I didn't put it in my Dropbox. So it's not giving me a way to have access to other folders. So,
okay, what would happen is if Professor Ann carry on, then let's try to get your slide up and send it to the JVC, if that's okay.
So, Prof. Felix, we can have your presentation after the next one. By that time, we will be able to share the presentation from our side.
All right.
And now we can invite Professor Adi Feleke from Addis Ababa University. Are you online? Professor Adi Feleke? Yes, thank you very much. Prof. Adi, you can share your screen, or if you'd like us to share your screen, your presentation, let us know.
Yeah, I think Prof. Adi, you need to unmute. You are now a co-host.
Okay. Yes, I can unmute myself. Sorry, I was not able to unmute myself, so let me share my screen. I already have shared you. Is it okay?
Yes.
Thank you. So good morning to you all, and welcome to this very interesting workshop and conference on biodiversity. So my talk will focus on the microbe world and how the diversity looks like in Africa in general and in Eastern and Central Africa in particular. When we talk about microbial diversity, we should keep in mind that we are encompassing the bacterial world, the fungal world, the viral world, and the protistal world. So my contents will focus on first the role of microbes in the biosphere, just a gist of it, and then how much is known about microorganisms and the challenges of studying microbial diversity as compared to the other plants and animals diversity. And then we'll slightly talk about the role or the contribution of genomic-based technologies for studying, for understanding microbial diversity and microbial ecology in different settings. And then we will also discuss about what is next. How do we embrace African diversity, microbial diversity, and explore more and understand more for sustainable life and for sustainable health. So as we know, Microbes play a very vital role in the biosphere. They are very important both in the natural and engineered ecosystems. They are very important contributors of preventing climate change from exacerbating if we are able to make use of them. They are very important in human life, in human health. They are usually part of every animal, plant, and other visible organism in the world. And now there is a strong advocacy to include, for example, soil microbiology into One Health, because human health is always connected with animal health, with soil health, with plant health, and with other ecosystems as well. So we also, on the other hand, want microorganisms to take care of our wastes. We generate a lot of wastes, and the more industrialized a nation becomes, the more waste it generates, and there should be some part of the ecosystem that we should take care of this. And by applying microorganisms, it's possible to take care of our wastes and also generate energy, sustainable energy. For example, biogas, which is usually the product of the roles of different microorganisms in converting waste into energy. As we know, the largest proportion of the microbial world are not cultivated. So we have, for example, as data show, more than 99.9% of the microorganisms in the soil, especially the bacteria in soil, are not yet known. They are not yet being understood very clearly. So we don't have any cultivation strategies to explore these huge proportion of bacteria in the soil. If we come to water, for example, in wastewater, more than 90% of the bacterial world is not known. In drinking water, the water that we drink, even though it looks clean, we only know a little, very less than 1% of what is there in the drinking water. So nearly from 93 to 99.9% of the bacteria is not known, is not understood. So when we come to Africa, before we come to Africa, let's talk about what is there in the world. How many microorganisms of all types have been understood, and all their physiological properties known and stored very well for reference. So as based on a database, BacDive, which focuses on bacterial diversity, worldwide, we have nearly 93,000 strains identified. And of all these 93,254 strains that are in the databases, it's only 51,557 strains that have a complete geometadata. That means complete information is available on where they come from, where they are isolated from, what type of, is it soil, is it plant root, or is it animal, or is it human, clinical, or any other source is known, and where exactly it was isolated is known. So it's really important to have a complete information so that we can understand where is located and how we can enhance the applicability of that information. When we come to Africa, it's really very small. So we only have, like, out of the 51,000 strains with complete geometadata, it's only 3,767 isolates that are available and that are reported from Africa. When in percentage, it will be less than 10%. So these strains are spread within the northern, the southern, the eastern, and a little bit into the Central Africa. So we need to do more. We need to explore more with the available technologies, with the genomic technologies, in order to understand the microbial diversity that we have in Africa. And then when we see where they are sourced from, all these 3,767 isolates or strains, 42% of these isolates come from the soil, the environmental setup. So that is somehow good news, because as we know, mostly these microbiological studies and application of genomics to microbiology was very advanced in the health area, in the human health area. But when we look at the strains and all the information, it's the environmental, and mostly it's the soil microorganisms, and a little bit less in the plant, about 32% from plant and animal hosts. Out of this 32%, 23% comes from the plants. And the rest are infectious agents and human body and engineered sites. When we talk about type strains, that means these are like reference living strains that are well defined with complete phenotypic, physiological properties, well studied reference organisms. Africa has only 108 type strains as per the data from this month. So this is really a very low information, and we need to do more. We need to understand the diversity that we have, despite having a very big diversity in Africa. Only small proportion of the information about this microbial diversity is captured and stored in databases. Mostly these strains are bacteria and archaea. We don't have fungal strains. We don't have strains from the small organisms, from the unicellular eukaryotic organisms. And most of them are, as you see, concentrated in the southern, northern, then a little bit of Western Africa. And we need more in the Central and Eastern Africa as well. So we now the technology is expanding to have more and more of the metagenomic data generation. That means you have collective genomes from a specific environment, a specific host, or a specific site, and then you can understand who is there in that particular setup, in that particular sample site, and what is their roles, who is doing what in terms of its interaction with others, interaction with the environment, and also with other physiological properties. So metagenomic data, even worldwide, it's not that much. This is from a paper which was published in 2023. And then we only have worldwide 333 hits. As you see, the distribution is a little bit clustered to some continents. And Africa, as you see, has a very low hit. And then with complete geometadata, we only have 235,000 hits. That means, different areas. For example, it can be environment, it can be the water, or it can be the soil, or it can be the plant root. And then host associated can be fecal, or it can be gut microbiome. It can also be respiratory tract microbiome and others. So from such sample points, we have very, very low studies and very low data collected from Africa. And then, as you can see, most of them are they are clustered towards the eastern and southeastern part of Africa, and a little bit in the Western Africa. very, very small proportion in the Central Africa. So what do we have here? It looks like, okay, we are rich in diversity, we are rich in microbial diversity, but we have not captured this diversity very well. And unless we capture it, as has been indicated by my previous speaker, we are losing it due to different reasons. We are losing soil microbial diversity, maybe due to overuse of fertilizers. So we don't know what we have, and then we are losing what we even don't know. So it's really important for us to understand the richness, the diversity of the African ecosystem in terms of the microbial world should be captured very well. This is just this graph that you see in the middle is just to show you from a study to show you how the gut microbiome of the African population, selected African population, which was studied, how different it is from the US population. So the higher bar that you see is how different it is, the Tanzanian gut microbiome versus the US gut microbiome. So we have these high differences, and a lot of studies has been done in the northern countries, in the well-developed countries, and all the information is there, but we cannot use what is there, what is already produced by them is not applicable for Africa. So we need to explore more on what we have as Africans in terms of the microbial world. So there are different approaches. Now we have the genomic platform, and we have a very, very important infrastructures here and there in Africa. So we have to make use of them in a sustainable manner so that we explore and we understand our microbiome world. This is a picture that I took from the group, the work by Tulani from the University of Pretoria. They were advocating that African microbiomes matter. And then they suggested 3 pillars so that we can capture, we can understand the microbiomes of Africa from different aspects, not only from the human, but also from the environmental aspect. So we need coordinated research. We need a very good network, just like the African BioGenome Project. That is helpful not only to communicate between researchers and between countries within the continents, but also to communicate effectively between disciplines. We have the human health discipline, we have the animal health discipline, we have the plant health, and all focus on microorganisms. So we need to connect and collaborate and explore each other's discipline through coordinated research networks. And then the second one is infrastructure development in a sustainable manner. Now, the word sustainable is really important because now as African countries, I think we know that some of the countries we have, a lot of sequencing machines, but only few of them are functioning. And we are just, expending a lot, and we are wasting our money for those which are not functionally active. So we have to sustainably develop our infrastructure in terms of hubs, in terms of within countries or between countries. And then the third pillar that they suggested is the interventions of policies by strengthening local leadership through effective science communication. We have to be very vigilant as scientists to share our findings, to communicate with the policymakers, with the local people as well, so that they get the awareness and they own the information. And that also leads to conservation as well. So I would like to finish by this bird, which has the name Sankofa. Maybe some of you know. It's a word of the Akan tribe in Ghana, which means it's not bad to fetch what is at risk of being left behind. So we have to look backward. We have to look, what we have as, what we have as diverse microbial populations, as diverse and rich microbial communities in all the ecosystems that we have. Once we have all this information, we can move forward on how to make use of them, how to utilize them in a very sustainable manner, and how to solve or alleviate the problems of climate change or famine or any other challenges that we have as Africans. So let's embrace Sankofa and let's move forward. Thank you very much.
And thank you very much, Professor Adi, for that. Very, very insightful presentation. And key highlights from your presentation is that African microbiomes matter, and we better need to understand our microbiome. And therefore, we in all spheres of human, plant, and other worlds need to connect and collaborate. I think what was also very key was the idea that we need to develop our infrastructure sustainably, that we have sequencing machines on the continent that are not working, or others are working, but they're never used. And therefore, the idea is not to place instruments anywhere, but is to place them in a sustainable manner for development. We have to be vigilant as scientists to be able to effectively communicate. These are key messages. I didn't introduce Professor Adi, and she's a professor of she's an associate professor of environmental microbiology in Addis Ababa University, and has a PhD in applied microbiology. She has extensive networks. She has done extensive work on the African microbiome, and she's heavily involved in the One Health research with a focus on water, wastewater management in cities of low-income studies, with substantive authorship and co-authorship of publications. Thank you very much, Professor Adi. I'd like to request you to remain online as we listen to Professor Felix. We have your presentation, Prof. Felix. If you're online, you can now present your presentation, and we will also move the slides from this particular side. And welcome, Professor Felix.
Yeah, thank you. Thank you very much. Are you getting me? Is it okay?
Yes, let's carry on.
Yeah, thank you so much. It's a pleasure for me, and it's a privilege to be part of this talk, and thank you so much. So I think the idea of this presentation is to give a glance of what animal diversity is in Central and East Africa. So we're going to talk about the situation, evolution, and the perspective that we see. I just want to pick from what Prof. Ade just said. I think there is a need to have more concerning the awareness for a proper ownership. Next slide, please. So the presentation will follow this outline after a glance of what Central and East Africa animal diversity is about. We're going to talk more about animal diversity, situation, and the evolution in that geographic context. And after all, we're going to talk about the initiative to tap on this biodiversity and see what major perspective and partnerships will help. So concerning what Central Africa is about, as you can see on this map, it's a very vast country, it's a very vast region made up of small countries. Some of them are very large, like Chad or DRC, and some of them are very minute and somehow even forgotten. We have insular countries like Sao Tome and Principe, Equatorial Guinea as well, some islands. And the region is expanding from the northern southern region, even southern to dry areas. And we can have an idea that these 9 countries is made up of diversity of landscape, people, and languages. There are more than 5 different languages, so you can understand that across border somehow, though we have the same population, but languages, most of them from colonial history, can make understanding each other very difficult, and political instabilities as well. In some of these countries, civil wars have been for long, and this has consequence on the biodiversity and the conservation. Next slide, please. And the wild animal diversity in Cameroon is known to be very rich. For those who don't know, Cameroon is derived from Camaroes, that is Portuguese, that means shrimps. And Cameroon is made out of a rich biodiversity, ranking 4th in floral diversity and 5th in fauna diversity in Africa. And this made 92% of Africa's ecosystem. So, and Cameroon is most of the time called Africa in miniature. So the Central Africa could be called also African miniature because of countries having a lot of diversity, mammals, reptiles, birds, amphibian, fish, plants as well. I think other colleagues and even microbes, other colleagues have already said a lot about that. So there is a need to understand exactly what is happening, and the Central Africa is not isolated because Central Africa is like a continuity with Eastern Africa. Next slide, please. Next slide. Yes, and the East Africa is, as you can see on this map, Central and East Africa has been decided because of African Union geographical boundaries, but you can see there is if we can try to understand exactly what the map is about, we have few insular countries like Madagascar and some other neighboring countries like Seychelles and Comoros, but we have a country expanding from the northern to the southern part of Africa with a kind of landscape, climates, people, and also historical background, but united by something, not only the riches of biodiversity, but also the risk and the decrease and the loss of biodiversity, though it's been exploited by people for so long. Just want to remind that East and Central Africa are known to have been the center of humanity. We know the very first humanoids have been discovered in Chad, and we have also in Kenya, which means this Central and East Africa is at the heart of humanity, is something we have to think about. And new knowledge from new outputs from science is showing there are many things happening, like of recent in Mozambique, in Mozambique and Zimbabwe boundaries. I think there is a discovery showing that there have been a very old civilization in that area, with which can exactly push some limits or some boundaries concerning our understanding of animal and plant domestication. We'll come back on that later on. Next slide, please. So for livestock in this particular region, particularly Central Africa as well as Eastern Africa, there is a lack of policy concerning animal genetic resources. The lack of monitoring is been also indicated. Also, the in-control breeding system. I think there is a wonderful paper by Koske about crossbreeding, what is happening, the rampant disease outbreak, what is happening concerning animal genetic resources. Because in Central and Eastern Africa, what has happened concerning livestock particularly is very obvious that there is a lack of understanding exactly the riches and the wealth of native livestock species, and the crossbreeding is bringing a great loss to what has been considered already adapted animal breeds on the continent. So there are few studies that have been made. On the left side of the slide, you can see one report that we co-authored in 2016 about what is happening in animal genetic resources in Central Africa. There are some other documents that can be seen through FAO or EU-IBA or some other organization showing exactly that the riches is not really unveiled, because there's still wide diversity of animal species, particularly in livestock, and the knowledge from local people has not been really tapped and understood. So there is a need to embed and involve local people who have been dealing with these animals for centuries, and to understand exactly their ways and why they have chosen these species and breeds. Because people using animals are not only the owner, they're also those doing the breeding fabrics. So there is a need to understand, and using technologies that has been presented yesterday, wonderful presentation we have yesterday. So those ideas and those fabrics, those genetic fabrics, could be used and deciphered using genomic technologies to have traits. And some of the traits that could be very interesting are the traits that could help to withstand disease outbreak. Like in the upper slide, you have what we call black belly sheep. It's one of the only sheep living in the forest, in the humid forest in Cameroon, found also in Congo and Gabon, in very deep forests. They are brown, and some of them are black. How do they survive in these very harsh conditions? Those things should be investigated. We have also prolificacy, because now with climate change and the intensification of agriculture, there is a need to think about diversity and prolificacy for biology efficiency. Next slide. And then if we go to biodiversity per se, biodiversity of this animal is rich in food. Some people are using it for medicinal uses, like camels, like camel or dromedaries. Let's say dromedaries in Chad, for instance, because in Central Africa it's found only mostly in Chad, in Cameroon, but in Somalia, Ethiopia, Kenya, and some other countries where they are very useful, but not known because neglected in the research mainstream. So there is a lack of conservation program of fauna in the region. We have some, as earlier presenter said, there are some protected areas, but most of the animals and some traits and the description are mostly found not in Africa, but in European museums. So there is a vast deforestation despite all conferences on climate change. Next slide, please. Next slide, please. Yeah, and you can see, as I've mentioned, about the diversity and also the mainstream, and these calling people in academia to open eyes on things that are almost forgotten. On the left side of the this slide, you may see these caterpillars. There are more than 500 species. Some are known and some are unknown, that have been used in Africa to feed people and to feed millions of people, but they are not in the mainstream of our research. And we know how they play a great role because these are caterpillars. And so butterflies or moths play a great role in pollination. We have some other forest ruminants like these duiker that has been tamed in some areas, in some communities. People know how to tame and even to keep in captivity those kind of animals. So they are weak conservation programs. So the perspective, because we have been talking about forest protected areas, but they are in Central and East Africa, is very, very difficult to hear about freshwater or seawater protected areas, because they are not only terrestrial animal and avian, but they are also aquatic. And because of the richness of the fauna, Central and East Africa is suffering of poaching, but also of sometimes outbreak of diseases like Ebola and some other Lassa disease, Lassa virus disease, and some other diseases. Next slide, please. Next slide, please. Yeah, it's just at a glance, the deforestation is and the forest degradation is very fast. Someone has already said it, so I don't want to put it again. Agricultural and pastoral expansion is a trend, and there is a need to reduce the human-wildlife conflict and zoonotic diseases. And we know what is happening with the contamination, poisoning of water resources, and the disappearance of pollinators. Mining is a problem, particularly in DRC. I mean, in the country of Great Lakes, we know what is happening because of minings. It could be political or correct or not, but mining is becoming a huge problem because of not only forest and soil degradation, but also of aquatic contamination. The pollution is coming very fast, and there I think the contamination will result in great losses. So there is a need to link mining and the conservation of animal biodiversity in Africa. So there is a weak or inappropriate policies and legislation in most of the country. We know we spoke yesterday about the Nagoya Protocol, but for instance, in some Central and Eastern Africa countries, the law from countries are very weak or maybe very stringent. That could be that could hamper the desire to understand and maybe to exchange samples to do exactly the job we are intending. I mean, this consortium is intending to do. Next slide, please. Yes, the wildlife, not domesticated, still very great, and this gives enormous opportunity for those who are working on domestication as poaching, because poaching is because the nature is giving a wide diversity of animal, and poachers are coming, but it really from very far, and international traffic is becoming a crime, even for communities. We still remember what happened in Cameroon and Chad, where hundreds of elephants were killed in less than 1 week, and people that came and slaughtered more than 200 elephants came very far from neighboring countries. Next slide, please. And we can see on this slide what the wild animal is providing to poachers. You can see the network, and this only for wild animal. What could be done to have this type of trade being in benefit of the community of Africa? And this showing for me the wealth that Africa is, Central and East Africa, as we can see on the map, can have from the diversity of the livestock or wild animal we have. But also we know that we have to take into account what will happen in case of unhygienic conditions. The last slide before we wrap up. There's few current initiatives to tap in this biodiversity system, and the Central Africa Gene Bank has been launched, facility hosted at the University of Chang, training and formalities under AUI bar. Administrative procedures are not yet completed for functionality, as well as in East Africa Gene Bank, already working. They're working on microbes, they are developing SOPs. And just to say that in Central East Africa, political boundaries could not become finally a threat to facilitating biodiversity and biotechnology work for the benefit of the community. So we have to think about that to instead of having small spots of things that could not finally make the difference how can we put together the facilities to have things happen? Next slide, please. Next slide. Yeah, there are a few things that have been happening, biobanking and sequencing, but in the nutshell, there is a need to work on the conservation of breeds under threat, particularly genomics of diseases, resistance, the program of in situ conservation, and bioarchiving system. Because for now, most of our bioarchiving systems are more or less hosted in Western countries. So there is a need to put hands together, as there is a Bantu say, meaning that only hands put together can be able to tie a bundle. So there is a need to put hands together. And the next slide is showing, exactly what could be done as perspective core projects in poultry, goats, and some other birds. AU-IBA endorsement for this, because AU-IBA is in charge of animal diversity. And also Africa Genome Bio Project Quick Off in Central Africa and East Africa, backed up by IRI, as we know. Also, Africa Animal Genetics Core book launching, because there is a need to have books and training material from Africans and for Africans to understand exactly what is going on. And for that, next slide and the last. And for that, next slide, please. And for that, we need to have all these initiative organizations coming together for the benefit of the community. Thank you so much for your kind attention. Thank you so much.
Thank you very much, Professor Felix, for that overview that you have given. And indeed, what has come out is that there is a need to collaborate and put organizations and people together for sustainable, again, what Professor Adi was talking about, for sustainable development of instrumentation. So Professor Felix has described the African situation, the East Africa, Central Africa, and I think what is very clear is that the unprecedented loss of biodiversity happening in these biodiversity hotspots. And unless we come together as Africans who are living and working in these places, we are actually going to leave a very poor legacy for our children. We have seen the very unique plants, the unique animals that are living in the Central African forests. And we have to start asking our questions. What is the genetic makeup of these plants and animals? How do they survive in these very unique ecosystems? And how can we be able to characterize them and document them before they are lost? And at this particular moment, I'd like to invite questions. Are there any questions for the three speakers? We've listened to Professor Adi, Professor Felix, and Professor Avana in the morning, who also talked about the biodiversity and forests within the African continent. Any question? I can see a hand there. Yes, please, and use your mic.
Thank you. My comment or probably question would go to the first speaker on the non-timber forest products. My name is Eunice Machuka from ILRI. I think it's very interesting and dynamic. For me, I would look at it as, you're using timber and also gaining from any other non-timber products that are found on it. But then again, how does this impact on sustainability, considering the commercialization aspect, overuse, and eventual loss of diversity and potential extinction due to demand for these products? Thank you.
Professor Avana?
Thank you very much, Eunice, for that very good question. Yes, how to better align sustainability and profitability when developing value chain for these forest resources. As I've mentioned, the best way is to create that alternative source of supply through domestication, and also ensure that when the alternative source of supply has been created, the genetic resource base is still being preserved and conserved through these protected areas or the live gen banks and so on. So yes, the best way is mainly to ensure that we can create alternative source of supply, because we need to really tap into this the existing product all those natural resources provided by our forest, but we also need to conserve forests. So the best solution is domestication, but also to ensure to characterize, as indicated by the chair of the moderator of the session, is also good to know what are the diversity existing within each of the species being domesticated, and which one is preferred, and how can we ensure that even the one which are not being preferred, they will still be conserved. Thank you.
There's a question online on the connection between microbes and climate change. I think this is a question for Professor Adi. The connection between climate change and microbes.
Okay, thank you very much. So when we talk about climate change, we are talking about a complex situation that is factored by many factors that contribute to climate change. So one of the effects of climate change is, for example, drought. And then drought has its own impact on the plant and animal and human life. It can be short-term, it can be a long-term one. So when we talk about microbes, they usually make sure that, for example, the nitrogen cycle, the carbon cycle that we know of, the sulfur cycle, the phosphorus cycle, you can name any geochemical cycle. These cycles are maintained by microbes. So for example, the carbon cycle. When we talk about carbon cycle, we can look for the greenhouse gases, for example, methane and carbon dioxide as well. So usually the emission of these gases is an indication of the disruption of this elemental cycle. And to maintain the cycle intact and to minimize, at least not to avoid, but to minimize this disruption and emission, which results in emission, we need to take care of the microbes that take a good care of the cycling of these elements. So this is one example on how microbes contribute to the effect of climate change. So there are so many other connections, but this is one important example that I can think of now. Thank you.
And thank you very much. A final question from the floor before we move on to the next sessions. Yes, there is. Okay, I see two hands, two quick questions. Please unmute and ask.
Okay, good morning, everyone. My name is Vanessa from Cambridge. So my question is to Professor Adi. When you're talking about the microbiome, African microbiome, do you also go to the intricacies of the bacterial and gut and the phages interrelationship, because all this play a part in how we feed and the drugs that can actually be used in the gut? For example, Clostridium difficile has been seen to have a huge reaction when you take antimicrobials. So do you also actually put in place, or do you factor in the relationship between these microorganisms, bacterial organisms, and the phages that are in that gut?
Okay, and the other question quickly.
Thank you so much. My name is Wanja Levons, work with the IT here at ISIPE. One is a concern about, from yesterday, we have been told that there is a need for Africa to write its own story. So where is my place as a student who is working in IT at my master's level? What should I do? How can I contribute to the dreams of the African BioGenome Project? And secondly, what is the place of GMO products on biodiversity? Thank you.
So Professor Adi, I think the first question is yours, and any takers for the second question, feel welcome.
Thank you. Thank you, Vanessa, for a very interesting question. So when we talk about microbiome, so the very term of microbiome encompasses bacteria, viruses, phages as well, fungus, and protists altogether. So, for example, the gut microbiome usually refers to all these categories of the microbial world. So for example, usually phages are an indirect indicators of that particular bacteria. If you can't find the bacteria, for example, if you can't culture the bacteria, if you find the phage, that is an indirect indicator that the bacteria is there, because the phage cannot survive without the host, that particular bacterial species or strain. So they are included, but the example which was mentioned, the Clostridium difficile, we also have to consider, when we consider gut microbiome of African population, we also have to consider our feeding style, our nutrition. You know, C. difficile, where is it very prevalent? We know where it is. I mean, it's usually out of Africa and in the more industrialized nations. Also, the cancer that's associated with C. difficile is more common. I'm not saying that it's not present in Africa, but the food that we eat, the feeding habit that we have, is good enough to be maintained so that we can be prevented from such imbalances of the microbiome in the gut. So this is just a comment. We have to look at our food and our lifestyle as it changes. And of course, when we talk about microbiome, the phages are also important component of the microbiome, and especially those that are not very abundant can be tracked by their phages as well. Thank you.
Dr. Thank God, if you're online, you can answer what is the role of a student in a project like African BioGenome Project. And I don't know whether Professor Felix would like to tackle the one of GMO and the whole conversation around the GMO. Thank you. Professor Felix, you can go first. Or Professor Adi?
Yeah, Professor, and if I may react on the role of students or data from students. Actually, the data that I presented earlier are all from students, PhD students, master students, those who are brave enough to submit their culture, their genome data to the databases like NCBI and others. Those are the ones that popped out as part of the African data. So you already have, as students, you already have a major place, a major contribution to the data that has been generated in Africa so far. Also from yesterday's presentation, as you remember, most of the studies are done here and there by students in a fragmented way. That's why we need more, organized connection, organized collaboration. We need to move beyond the students, but the students are very basic, workforce for generation of data, for submission, for contribution to the information that is available worldwide. That is undeniable. So keep on, working what you are working. Make sure that you are an important player of this by submitting your information. I just want to ask, how many of you know, students working on various isolates, maybe plant root associated isolates, mycorrhizal, this rhizobium species, but very few are there in the databases. If you just Google and find which are there, how many of them are entered and being curated and having a proper ID, very few. And the rest are there in our, in the labs, in the freezers, facing power interruption and, all these challenges. So let's be vigilant as students and as supervisors and as also collaborators. Let's be vigilant in communicating whatever we have on hand, because we need them eventually for sustainability. So thank you so much, and you are a very important part of these things.
Indeed, I want to reiterate that students, graduate students, undergraduate students are the power engines of any research project. They're the ones who are on the bench, they're the ones doing the work, and powering their hard work together with mentorship is key so that we can be able to position the students in a position of growth for their upward mobility. So I want to encourage all graduate students, all undergraduate students to continue and further your education until you graduate with your PhD. And at that point now, you're ready to become a young postdoc or a young scientist, an early career scientist. And that's a focus for African BioGenome Project, to see how we can be the stepping stone to open up opportunities that are present on the African continent in collaboration with others to grow our own capacity locally and also internationally. So don't give up because your project has run out of money. Let's see how, as Professor Adi has said, to partner, to increase collaborations in order to be able to provide access, because one of the things, we have the capacity here, but maybe what you're lacking is access or a connection to a lab or to an opportunity elsewhere. And we hope that by you staying in touch with African BioGenome Project, we will be able to show you what some of these opportunities are. And we are already beginning to pass out our own fellowships. But I think it's not, their fellowships are already there. It's the lack of information that is flowing that we need to enhance on the continent. And I urge all of you to subscribe, go to the website, our website, and ask to be a member, because then we'll put you on the mailing list. And from that, information will flow. And I want us to give a rounding applause to the three distinguished professors, Professor Felix, Professor Adi, Professor Avana, who have given us very, very insightful presentations in different aspects. And we are really, really indeed glad and grateful. So a rounding round of applause. Thank you very much. And now we move on with our program to listen to Dr. Amel Tango. I'm sorry, I don't know the correct pronunciation of your last name, Ningetem, from the University of Xi'an. He's an assistant lecturer in bioinformatics, biostatistics, animal breeding, and genetics from the Faculty of Agronomy and Agricultural Sciences from University of Xi'an in Central Africa Republic. His work focuses on the development of eco-friendly practices for sustainable animal productivity, health, and safety. I think you're based in Cameroon, and we're really, really looking forward to hear about what you're passionate about and comparative metagenomic studies along the gut biogeography of indigenous chicken. Dr. Amel, if you're online. Are you able to unmute and present?
Hello?
Yes, we can hear you. Please go ahead and welcome.
Okay, thanks very much, Prof, to have given me this opportunity to showcase this research, who was part of my PhD down there at Pausti, Kenya, joined with Jomo Kenyatta. My name is Dr. Amel Tangumo, and I'll be delighted to take you through this one of my research output, which is entitled Comparative Metagenomics Along the Gut Biography of the Engineer's Chicken. The presentation will be started from the background up to some research perspective. This work started, the story with the behind this work started down in 2015 when I graduated from the MSc. So I was doing some businesses around poultry production and some consultancies. And through my work exchange with farmers, it has been about the different challenge that farmer was facing was having as a challenge, like technical part, and also the effect maybe of the diseases. We also realized that so many farmers their productivity, especially of their local chicken, was really low. And the low productivity of those chickens was due to several factors. Amongst other factors was, as it is pointed in the literature, the gut, this part, as you can see on the left hand of the screen, many scientists have said that this organ, as Professor Adeli have already said, the gut microbiome is really considered as a forgotten organ. While many researchers have said that this organ can really play a very important role in stimulating the immune system of the animal, and can also help the animal in breaking down certain nutrients that it cannot break down with his immune system, with other communities. And other thing is that this gut microbiome, through the evolutionary history of through the life of the animal, could co-involve the animal and allow the animal also to adapt to certain environmental conditions, and also to be resistant to certain disease. And if this gut microbiome is well monitored or is well managed, can also be one tool to reduce the effect of greenhouse gas emission, which is one way of climate change, which is one of the causes of climate change. And it can also be very good to reduce the effect of antimicrobial resistance, which is actually one of the worldwide health threat. So, as I already says, it has been observed that African indigenous chickens gut microbiome, there's very limited research or very limited result regarding that gut microbiome. And one of the factors who limited the availability of the result of that gut microbiome is that conventional culture methods allow us to only isolate around 40% of the gut microflora, which could be attributed to the fact that it's very difficult to reproduce the gut microbiome habitat. And also, even I myself, I found it really fastidious when I was trying to isolate some of those gut microbiome. And there could be also a very high risk of contamination when doing those processes. The reason why, if here in Africa, if we are able to quantify all those indigenous gut microbiome from chicken, it could be really possible for us to formulate a specific host, a specific nutrition requirement, which is really adapted to chicken, and also formulated a specific prophylaxis program adapted to our chicken, and also help the chicken to well adapt, well cope to environmental conditions, and also advocate for precision breeding of the animal and optimize the productivity of the animal. And among the different techniques which have been proposed so far as a way to quantify that gut microbiome, yesterday we talked about the nanopore sequencing for long read, and we talked also about the SMRT. But the one conventional method, the method which is actually more classical, which is more used, is this technique called the 16S ribosomal gene high throughput Illumina sequencing, which is that markers is a marker which is found across all, the bacterial community, which is highly conserved, but which is used for identification of those all of bacterial community along the gut of the chicken. So if this was done elsewhere, like in Europe, in America, in China, we're still really low. We are still behind, especially in Central Africa, as very little research have been done here. And even there in Kenya, there's very few research on using these techniques to harness the microbial diversity. The reason why the objective of the part of my PhD study was to characterize the microbiome, the metagenomic diversity, let's say the bacterial community diversity along the gastrointestinal tract region, composed of the crops, the gizzard, the jejunum, the cecum, and the other additional organ, which is the liver in the gut microbiome of the indigenous chicken. In attempt to answer this question, we purchased 16-day-old chicks back then in Cairo, Navasha. Those chicks were brood, as you can see in this image. Then they were brood for a period of 21 days, and throughout that period, they received the same prophylaxis program, the same amount of feed, and they were placed in individual cages, as you can see here in this experimental design. And then after putting after transfer them in the individual cages, they received the same feed from the starter, the grower, and the finisher phase. And throughout that period, we were able to monitor their feed intake, their daily weight gain, and their life weight. And also at 14-week period, the chicks were euthanized to collect the organ weight and also to store the intestinal sample, such as the crops, the gizzard, the jejunum, the cecum, and the liver in the cryovial. And the total of 80 biopsy samples were then stored in liquid nitrogen and later transferred then in the and transferred in -18 then for that transport in Baker Erie Herb, where the sample were used for genomic DNA extractions using the GAIM DNA tool mini kit extraction kit. Then the genomic DNA sample were then used for DNA library preparation and following by the sequencing. And all the 10 million sequence, around 10 million sequence generated were then stored in Baker Erie high performance computer for bioinformatic analysis, and then following by downstream analysis and graphical visualization using the RStudio version 3.69, as you can see. Regarding now the first result, which is to first of all have a proof of concept. This table just gives us the phenotypic characteristic of 14 weeks indigenous chick fed under the same regimen. And as we can see from this table, the feed intake was around 3,000 grams for an accumulation daily weight gain of 992, with the feed conversion ratios of 3.15, and the life weight of the animal was 1,399. Which is compared to what was observed in the literature was nearly what is expected from the breed that were used. And the breed that were used was the dominant black, especially the male. And regarding now the intradiversity, this figure just give us the box plot of some richness and evidence indexes along the gut of the local chicken. As we can see, if you see the three first box plot, you see the observed value, the Kao index, the ISE is the eveness index, while the Shannon, the Simpson, and even Simpson is the richness index. But as we can, what take home message from this is that we do not see any significant difference between and amongst the different metric index that was used. However, the overall diversity richness, as you can see, tend to increase, as you can see here, tend to increase along the different organ with a peak noted in the genome. And this peak noted in the genome could be explained by the fact that that organ, especially the genome, is just a region where the absorption of nutrients is starting. Regarding now the beta diversity, which is the difference between the different organs, this was assessed through the non-multidimensional scale based on Bray-Curtis and Jaka dislaminarity index. As you can see from from both the left and the right table, or the left and the right figure, the second tend to form a particular ecosystem on its own. So it's shifted, it's forming like somehow a particular ecosystem separated from the other organ community. So the second sample, as is forming a separated ecosystem on its own, could be explained by the fact that as is already could be associated to its function, and because of that, associated to its function. And the second, as is already reported by many researchers, is an organ where there is a high absorption of the nutrients. And this high absorption of the nutrient could be associated to the high microbial load. And not only that, the gizzard, we can say that the gizzard and the genome really overlay each other, or are merging each other, which could be explained by who can suggesting that they may have, let's say, the same community structure and maybe somehow sharing the same function. So this, regarding now the taxonomy dynamic pattern along the different organ of local chicken, we can see that on the figure 5, we try to put together the phylum signature across the different organ, the class signature of microbial community across the different organ, and the order. As we can see from this figure, we can see that as we are going at the level of the phylum, we can see that the signature is not well, there are not clear differentiation between the different organs. But as we are going to the class level, up to the order, to the family, we can see that there's a clear difference between the different organs, as we can see that the second still forming a particular different organ from the other community. And other information that we can take from this figure is that the dominant phyla is constituted by the Firmicute community, following by the Bacteroides, the Proteobacteria community, and the Actinobacteria community. While at the class level, we have the Clostridia community, the bacilli, the Bacteroidia, the Gametoproteobacteria, and the Negativicus community. So from this study, the take-home message that we can take with us at the end of this study is that 16S ribosomal RNA gene high-throughput Illumina sequencing have shown us the suitability in quantify the different gut microbiome in local chicken. And there's also a high diversity of microflora in chicken gut, which could be consistent mainly with the Firmicute, Bacteroides, Proteobacteria, Actinobacteria as a dominant phyla. And also, the microflora could vary across the different parts of GIT, which may imply or suggest that they may have different function between different organs. So another thing is that the non-invasive, we can also conclude that the non-invasive sampling strategy from a single part organ cannot give us a clear map of the gut microbiome diversity, because we have found that in many studies, most researchers are focusing only on using second. to highlight, to study the microbiome diversity, which is not, it is good, but it could be also limited somehow. So it's always good to know, to have a clear map of how the other organs are behaving. So the study also shown that the FGHT ecosystem could be considered as a particular ecosystem on its own. So as a research perspective, we would like, if possible in the next future, to study the metabolic function of each microbial community along the gut of indigenous chicken. To know, to enable this will allow us to know what could be the function, what is the metabolic function that could be associated to each particular microbial community. And that will allow us maybe to formulate a specific nutrition or specific feed formula, which is really adapted to local chicken. And we will also like to evaluate the immune response and microbial signature across African indigenous chicken. How they respond to, how the microbial community could respond to certain infectious disease of zootechnical importance, such as salmonellosis, Newcastle, and Marek disease. We would like also to explore the efficacy and suitability of candidate probiotic microflora isolated from local chicken gut. And we also like to screen the feasibility of using certain novel probiotics, nanoparticle compound to improve the gut health and productivity performance of indigenous chicken. We can't finish this presentation without thanking the Pan-African University and the African Union who gave us the scholarship in those days for us to complete these studies, and the collaboration with our JQuat supervisor, such as Dr. Maina Kagera and Dr. Matthew Gisheha, and also the ABCF program who give us the fund who funded this project, the molecular part of this project, through the Baker ILRI. And we can't finish without thanking our ILRI supervisor, Dr. Christian Kiambu, really guide us and support us throughout this study. And also the help of Dr. Eunice Mashuka, Mrs. Bernice Waweru, Dr. Jean-Baka Dumeno Enfener, and Dr. Roger Pele, who train us throughout and who take our hands on this new field, exciting field, with metagenomics of livestock species. Thanks for your kind attention. Hello.
Hello, thank you very much, and a big hand. We have just given you a resounding applause here. And a take home is the study of gut chicken microbiome and the potential that it has, not only to improve the chicken health, but also to understand the feeds that would be suitable for use to improve their health. This is an excellent presentation that we have received from you, and we're delighted to also note that this high throughput sequencing was done in an African institution in Kenya. I think we can give you another resounding round of applause. And now I'd like to invite Dr. Patrick Baenyi Simon, who is with us online, to share his presentation. And yes, I think Dr. Simon, you have your presentation. If you have, you can share. So he's a Congolese citizen with a PhD in animal genetics and breeding from the University of Nairobi, based in Kenya. And right now he's a lecturer and researcher at Université Angélique en Afrique. And he's currently involved in a project on comparative genomics of local goat breeds in Central and East Africa, under the supervision of Dr. Christian Kiambu of ILRI, with an ultimate goal to develop molecular markers for improvement in breeds. Dr. Patrick, the floor is yours.
Non, vas-y, tu peux travailler.
Dr. Patrick, please go ahead.
Mais j'aime bien.
Can you hear me, please?
C'est mauvais, dis-moi. C'était bon.
Can you hear me, please?
Yes, we can hear you. Please go ahead.
Okay. Thank you very much, dear Prof, for giving me this opportunity to present this part of my research that was part of my PhD research 2 years ago. This is a very meaningful for me to share what I've been done so far. So I'd like to share with you some results on one part of my research. The title for this presentation is just the genome-wide analysis that reveals positive selection signatures candidate genomics regions in local goat population in the Democratic Republic of Congo. So as I said, this work was part of my PhD student, and I worked under the supervision of some professors at the University of Nairobi, but also Dr. Christian Kiambu, who is a scientist at ILRI, working with the CTLH. So what motivated us to do this research? Don't know. Hello? Why? Okay. So what is the background? First of all, DRC is one of the biggest country in the Africa and located in the Central Africa. And in this country, most of the population stay in the rural area. So in the rural area, the population of the DRC can represent more than 75%. And the economy of this country is mainly based on the mineral resources, but also on agriculture and livestock. And considering the livestock, I can say that the goats are one of the species that are domesticated in this population and contributing to their household income. The benefit of goats in DRC are so many, because you can see that goats can contribute up to 72% to household income, but in rural farmers, and but also it can be raised for meat, for reproduction, for milk, but also for commercial transactions. But also in some areas, it's raised to produce organic manure that is mostly used in land fertilization. But also it has an importance on social and cultural situations, because it, as you can see on this presentation, sometimes in some areas it can be used to manifest some cultural in some areas. But also there's another advantage of these animals. It's because it can be adapted to different agricultural zones of the country. But there is still a problem with this animal, because there is no sufficient information on the genetic structure, but also the genomic performance of these animals that can help to establish genetic improvement program in the DRC. And that can lead to the low productivity and low production of this animal in DRC. That is why we've decided to conduct this research, so that we could be able to generate some information that could be considered as the basic to do what, so to establish the goat productivity program in order to improve its production, but also in order to participate on the household income of farmers. So that is why we say that the assessment of positive selection signatures of candidate genomic regions in this local goats in DRC would constitute an opportunity of breeding program through markers assisted selection for economic traits. So that is the goal or the objective of conducting this research. So what is the methodology that has been used in this research? This work was conducted in 3 agroecological zones of the country, representing the whole country. As you know, DRC is a big country, and we could not collect samples from all the regions, 26 regions of the country. So that is why we've selected only 3 regions that represented the 3 major agroecological zones in the country. So at the total, we've collected some samples, 320 samples, blood samples, that was used for other stage of this research. So after collecting the samples, we extracted DNA from those samples, the genomic DNA, and we checked the quality of the DNA that we have extracted. And after checking the quality of the DNA, we've sent our samples for the genotyping. So we've used the Illumina for the genotyping of this DNA sample. So after having our result, we've started now for the with the data analysis. But before doing the analysis, we go through the SNP filtering and the data melding. So under Linux, we've used the software that is called the Plink to do the QC, and we have fixed some conditions to get some information that, or some samples that could be used for downstream analysis. So from the 320 samples, and we remained with 259 samples for the downstream analysis, and for the from the 53,347 SNPs, we remained with 45,335 SNPs, and that is the SNP that we use for the data, the downstream analysis. So for us to assess the signature of selection, we've chosen one method. We know that there is many methods that can be used to assess this signature selection, but we've decided to use this cross-population extended haplotype homozygosity to do what? So to extract or to know, to assess the signature of selection that could be associated with some candidate genes, and that could be in association also with some traits of economic importance. So to do that, we've compared our samples with some other samples from Eastern Africa, but also from Central Africa. And these samples that we used, they are samples that were already available online, and we've compared our samples with the Cameroonian goat population, but also one Ethiopian goat population that is called Kefa. And this, after doing this, after the cleaning of the sample, so we've used a R package, which is REHH, to now analyze, to do the analysis of our data. And as you can see here, this is the Manhattan plot that it's just an example of the Manhattan plot that was generated, demonstrating us or showing us some SNPs that were significant. And this is the SNP that some of these SNPs that we've chosen to do what? To do the gene annotation, but also to know now what this SNP could really represent. So for the gene annotation, we used some genomic, some software, online software, which is called David, for this part of this stage of the work. And the goal of this gene annotation was just to investigate the key EGG, but also to do the gene ontology by considering the biological function, the functional process of the gene, but also the molecular function of the gene. So after doing all this, we got some results. Sorry, I don't know what is happening here. Sorry. Yeah. Hello? Hello?
Hello.
I'm sorry.
Yeah,
Yeah,
we
okay.
have the fire alarm has gone off here, so we have to pause a little bit and exit until we are cleared to return.
So should I wait?
Yes, please wait.
Okay, thank you.
Our online audience, we are exiting the conference room because the fire alarm has gone off.
Okay.
Hello. So I'm wondering if the current presenter can finish up, because we don't know how long the fire drill will take. Then we can carry on with the rest of the meeting online and maybe the next speaker. So can the current presenter please finish up, and then I can moderate. So what's the name of the speaker, please? Patrick Banye or Okanya? Which of the Patricks? Banye, okay. Okay, I'm going Patrick Banye. Okay. So Patrick, you are co-host now, and you can unmute. Yeah. Please just carry on.
Okay. Yeah. Okay, thank you. So I was presenting the result after collecting the data, after doing the DNA extraction, after doing the QC of our data, and after doing the data analysis. So we got some genes that really could be really important according to their association with some phenotypic trait, as you can see there. Yeah.
Okay, please carry on.
Yeah. Just about another one. The PowerPoint is oh, sorry. So let me conclude this presentation by saying this: between these genes that were assessed in some genomic regions in our gut, some of these genes were associated with this was observed after doing the discussion of our results. Some of these genes were associated with some economic traits like growth, but also like reproduction, the prolificacy, the size of the litter size, but also the disease resistance, for example, the salmonella resistance. So for us, we were really happy to see that we could find some genes that could help us to now to go ahead with our program of goat improvement. So that is why you can say that this result is just like a basis on which we can now refer to go ahead and to for further research. So as our perspective in the future, we are trying to now to select some of these genes, because we did also the GWAS, the genome-wide association studies, after doing this, to know really which can be the phenotypic trait that can be associated with the presence of some genes, so that will allow us to select some of those animals that can be now kept in a farm, a station farm, where we can now be following them by knowing how they can be raised, but also this will allow us to improve some traits that we found that could be necessary in enhance, but also to improve the productivity of this species that is very important in DRC. So thank you very much for your kind attention and for your understanding. Thank you.
Yeah, thank you so much, Patrick. So does anyone have any question for Patrick, please? Any questions? So let me go to the chat. So does anyone have any question for Patrick? So I think Amel presented earlier, right? Okay, so we'll just wait and maybe we'll just merge all the questions together. Thank you, Patrick. Please just wait because I can see it's a panel discussion that's going to be. Can we have the next presenter, please? Joel Agwang and Joan Rokani. Is Joel on the call? Okay, I can say Joel. I'm gonna make Joel a co-host. So Joel, please unmute, and I will also make. So please unmute Joel and Joan Rokani.
Yes, hello.
Yeah, please unmute. You are now co-hosts. Share your screen, your slide.
Hello, can you hear me?
Yeah, we can hear you. Please carry on.
Okay, thank you. Right, so Joan is gonna go first, then I will follow.
Okay,
good morning, everyone. Can you hear me?
Yes, please go ahead, Joan.
Thank you very much for all the presentations that have been presented. We want to thank you for giving us this opportunity to be able to present during this conference or workshop. Once again, my name is Joan Rokani. I work at National Animal Genetics Resource Center and Data Bank in Entebbe. So I'll be taking you through the sample collection technique. Next, Joel. The next slide, please. As we want to process and conserve this animal germplasm or the plant germplasm, we have to make sure that sample collection is done very appropriately because it lays a foundation for accurate, dependable, and reproducible results in both the scientific research and analysis. We also have to ensure that it is done appropriately to ensure that we obtain the best result. The sample request forms should be filled appropriately as well, and then accompany the specimen to the lab. It should also include the name of the collector, the time of collection, and the type of analysis that is going to be done on it. Next, Joel. Next slide, please. Next slide. In the laboratory, we usually have a laboratory handbook, which contains the policies and the practices, as well as the standard operating procedures that contain all the information that we need for the different samples that we are going to collect. Usually it contains all the information that is needed, the collection needs and the preservation, labeling procedures, accessing, processing the samples and tracking, retention, storage and disposal, as well as transporting the samples to the lab. All that information is contained in the laboratory handbook. Still in slide, we are talking about the same thing, but also putting in practice the fact that all samples are seen as potentially infectious. So when you're working on them, all laboratory personnel should always have protective equipment to navigate through such that they are not affected. Next. These are the materials that we are going to use. Since we are going to hold a practicum session here at National Animal Genetics Resource Center and Data Bank, these are some of the things that we are going to need while we are collecting our samples when we start the practicum session tomorrow. The scrubs, the boots, disposable gloves, blood collection vials, the needles or vacutainer needles, the restraints, antiseptics and gauze as shown in this particular slide. And one of the techniques that we are going to do, we are going to collect blood from cattle. And one of the techniques is the jugular vein blood collection, where the animal is restrained in a crush or in a chute. And then it is cleaned. You use the gauze to clean the area from where you're going to collect the blood. Then you occlude the jugular vein by applying pressure at the base, just like it is shown in this second figure. So you apply pressure at the base, and then you insert the needle. You aspirate to collect the blood. If you're going to collect more than one vial, then you use the vacutainer needle. Then you discard the needles in the Sharps box. We can also collect that blood from the coccygeal vein, where still we restrain the animal. Then we raise the tail vertically. We shall see that when we do the practical bit of it. And then we locate the groove, which is lying in the ventral midline of the tail. We swab the area, and then we insert the needle perpendicularly to the surface of the skin, and then we withdraw blood. We shall then discard the needles in the Sharps box, and then we shall apply pressure to ensure that the animal does not bleed. The other technique, or the samples, we usually collect many samples. It can be blood, it can be semen, it can be tissue, it can be hair tissue, hair follicles. But now, in this particular instance, I'm going to take you through semen collection, where you prepare all the materials that you need for semen collection, which include the artificial vagina, the cone, the test tube, and then the jacket that houses the tube, because we don't want the semen to get exposed to light. And then after that, we shall get the bull, the teaser cow, and then we shall put it in the crush. And then the stud bull will be brought after it is aroused. As it tries to mount the teaser bull, then we put the artificial vagina just below, such that the semen is dissipated into the test tube, and then it is taken to the lab for analysis, for other analysis. Next. We can also use the electroejaculation method, where the probe is used. The probe is first lubricated, and then it is inserted in the bull. But we have to make sure that we look at the intensity of the probe, because it has to be controlled and monitored. And when the bull is aroused, then we place our cone and then collect the semen in the funnel, and then we take it to the lab for analysis. Thank you. Joel will take it up from there.
Thank you very much, Joanne. Now that we've shown you that we engage so much in sample collection, however, all sorts of sample acquisition should be done in compliance with the ABS policy. Therefore, I'm going to take you through the procedures of being compliant with the ABS policy of Uganda, and you can relate with your institution accordingly. So as it has been discussed yesterday already, the ABS policy is a policy which provides guidelines on access to genetic resources and how benefits arising from their use may be fairly and equitably shared between the users and the providers of the genetic materials, as provided for in the Nagoya Protocol to the Convention on Biological Diversity. The genetic resources may include associated traditional knowledge obtained from the local people. Use or utilization in this case includes research and development on the genetic or biochemical composition of genetic resources, as well as subsequent applications and commercialization. Complying with the ABS policy is very, very important for an institution like NAGRIC, which plays a very important role in the animal breeding value chain. For instance, NAGRIC engages in introducing new genetic resources in the country for the purposes of breeding. At the same time, it serves as a national gene depository and examination center for genetic materials, and accordingly hosts the National Animal Gene Bank, as well as the African Union Animal Resources Seed Center of Excellence. That is the Eastern African Regional Gene Bank. Also, the institution engages in collaborative research in the areas of animal breeding, conservation, and genomics. All these require acquisition of samples and moving it within the country or outside the country, which definitely require clearance and compliance with the access and benefit sharing policy of Uganda. So there are steps that you as a researcher can take if you're a Ugandan researcher or if you wish to do research in Uganda. And again, I want you to relate with what you can do in your country, where to go. So I have 8 points that I want to share with you. And the first is you need to know who the focal point is in your country, which could be the Convention on Biodiversity focal point. In Uganda, we'll talk of the National Environmental Management Authority. However, it could also be a body that registers research. And for sure in Uganda, the focal point for ABS is the Uganda National Council of Science and Technology. And it will give you guidance on which research ethics committees are accredited and who are eligible to review your protocols for sample collection, for research, and so forth. Accordingly, UNCST has published, as of April 30th this year, it has published 33 accredited research ethics committees, all of which have been guided to review protocols from any field of study. The next thing you need to be aware of is to know the minimum documents to prepare. Your research ethics committee will guide you on the documents to prepare. They will tell you whether they are templates or you need to develop your documents de novo, and if any of the documents will require translation. They will also advise you of the prevailing global scenarios. A very good example in our time is the COVID-19 pandemic. So you need to prepare protocols for mitigating COVID spread. So if you're unsure of what additional documents or what criteria, you should consult the REC Secretariat. Then the third thing you do is you prepare the documents, and be careful to be really meticulous, because the documents will be reviewed by very competent scientists. For research using animals, we have the School of Veterinary Medicine and Animal Resources, Institutional Animal Care and Use Committee of Makerere University, serving as the Research Ethics Committee. They have time templates. For example, the IACUC form. This form, you fill this form, making sure that you address the 3 R's of using animals for research. Number 1, do you need to replace the animals with something else? Or you need to reduce their number and still achieve the desired outcome of your research? Or if you cannot afford both the first 2 options, you should refine the methodology that you're going to handle the animals with, so that they experience the least pain in the most ethical manner. The second document is the full proposal, and then the third is the research summary. And then another important form is the consent form template. Here, you are expected to agree terms in advance with the provider of the genetic materials, and these are the mutually agreed terms, and you know this will form the PIC, or the prior informed consent. So you will fill in this template based on the mutually agreed terms, and then translate it into local languages, especially of the provider of the genetic materials. Next, you'll proceed to pay the fee for ethical review. You need to be aware of the different types of reviews and their fees. Again, ask your Research Ethics Committee Secretariat if you're unsure, because there are many modes of review. We have the regular, we can have speedy, we have expedited, all of which attract different fees. Pay the fee in the bank and keep the receipt. And number 5, submit your documents for ethical review. You'll be required to submit hard copies, soft copies, or both. The templates, including the data sharing agreement or the material transfer agreement, in addition to the receipt that indicates you have paid for review. Again, always call to inquire, are there any additional documents, and then follow up the review process. Then once you're done with the REC review, you now go on to register your research with the national focal point, in this case, the Uganda National Council of Science and Technology. You create your account and profile at the web link given, submit the documents indicating that the REC has cleared you, submit all other relevant documents, and then the fee for registration, which is also paid in the bank. And if you wish to export material, you need to prepare data sharing agreements and the material transfer agreement. Be sure to have a checklist to make sure that you do not miss any document. These are some of the critical contents of the material transfer agreement. Well, when we engage this process, we did not encounter standard data sharing templates or material transfer agreement templates. However, we were guided by the ABS guidelines and regulations of this country. And again, if you're not sure what to include, you should ask the national focal point. The DSA and MTA drafts should be reviewed and approved by the Research Ethics Committee prior to project or research registration. These documents will mention the provider of the genetic materials and the recipient, as well as the terms of use of the material. It will specify benefits the provider is entitled to and how any future benefits, for example, resulting from commercialization, arising from future use of the genetic material, are to be shared. They will also specify how material and subsequent data will be handled. For example, by the sequencing company, what happens to the materials after sequencing? Are they returned to the provider or are they destroyed? The protocols of return or destruction should be specified in the respective templates, either the data sharing agreement or the material transfer agreement. Again, the next step is to be aware of the different reporting points that your national focal point will require. So they will guide you because you are normally required to report progress of your project to the compliance enforcer. As general advice, you should understand the documents that are required in advance. If the material transfer agreement or the data sharing agreement is required, prepare drafts in advance and submit to the Research Ethics Committee for review. Sorry. Inquire about additional documents in advance. For example, COVID mitigation protocol. Start the registration process early enough. Give yourself enough timeline. Inquire about the different types of reviews and their fees. And again, ask the Research Ethics Committee Secretariat if you are not sure what to do. Be meticulous with your documentation. Keep your documents safe and enjoy the learning process. Sorry. With this, I hope that people who wish to engage in research are guided of the steps to follow, either in your country or here in Uganda. Thank you so much.
Dr. Thank God, you can carry on with the question and answer session. Thank you.
Thank you, Joel. Yeah, thank you, Joel. So let me wait for Anne. I can see that they are coming back to the hall, so she can now coordinate the moderation. Is that okay?
Yeah, sure, that's fine. Thank you.
Okay, I think because what is remaining now is okay, I can see Anne coming. What is remaining now is a panel session between Joel, Patrick, and Amel. Hi, Anne.
Hi, thank you.
Hi.
And thank you. We had a small interruption. Fortunately, no fire here, but we had to obey the fire alarm. And thank you very much. We were able to catch the last of Simon's presentation and also yours, Joel, and thank you very much. And now we are ready for some questions from the floor and also online. I can start with Joanne, to asking a question that is directed towards Joanne. And it says, during your sample collection for germplasm conservation, is there a way to determine whether you have duplicates of the same sample so that you don't end up with, such that you only end up with a core collection that comprises genetically different samples, and avoid wastage of germplasm resources? I think there's a question also for Patrick regarding the criteria that was used to select the different ecological zones. I think we can start with those, and then I will come back here to ask one of our panelists here to ask a questions. Go ahead, John.
Thank you very much for that question. Usually when we set out to go and collect samples, we make sure we collect sufficient sample and make sure that we have duplicates of the sample. Then on those duplicates, we keep part of the sample, and then we use one as a working collection. Thank you very much. Because it is also not usually okay to only have one sample just in case anything happens or you lose the sample. That means it can affect the whole process. So we usually collect a sufficient sample and then split it, and then we keep part of the sample, and then we use one as a working collection. Thank you very much.
And the question that was asked directed towards the ecological zone and the criteria for selection.
Okay, thank you very much. As I said, DRC is really big, and we could not attend all the different corners of the country. That is why we selected some regions for our data collection. So the 3 regions that were selected, they were selected according to some criteria. And the main criteria was just the representation of the main agroecological zone of the country. Because in the country you have 3 main agroecological zones, and the 3 regions, because one was in the central part of the country, one other was in the eastern part, and one other was in the western part. But also we focused on the economic importance of goat in those goats in those regions, but also the diversity, the genetic diversity that would be observed phenotypically on those animals. So those are the 3 main criteria that allowed us to select the three regions. Thank you.
There's a question for Dr. Armel. How does the difficulty in reproducing the gut habitat conditions impact the biodiversity of the chicken gut, and what potential consequences or challenges does this pose for maintaining a diverse, healthy microbial community within the chicken digestive system? Dr. Armel, are you online? While we're waiting for that, you can ask your question. Sorry? Okay.
Hello?
Yes. Thank you.
Thank you very much, Prof, for the question. The question is, how does the difficulties in reproducing the gut habitat condition impact the biodiversity of chickens? Isolating the microorganism, isolating the gut microbiome was really difficult, because in our study, we try, first of all, we wanted to isolate all the microorganisms, let's say characterize them through culturing first. because this was one of our research questions. But during the isolation, we found that especially the Clostridium community, with our basic lab setting, it was really, really difficult because those was among the anaerobic microorganism. So we couldn't get the let's say the appropriate lab setting to be able to isolate and characterize all those community. So it could be really, really difficult if maybe you are having maybe a new strength of let's say a new strength of microbe that you like maybe that could be maybe a new probiotics. So we found it very difficult to really isolate those new communities. So and the reason why we feel like we saw in the literature that molecular assay would be maybe a huge opportunity for us to screen, to know all the diversity of the microbial community that is found in the gut. But in terms of reproducibility, as I've already said, it could be really, really difficult. Yes, we need a lot of time, maybe a lot of studies to be able to test different media, different enriching media, to be able to isolate microbial community. But I think with bioreactors, people are doing it with bioreactors. Actually, just recently I've seen maybe by reading one paper, I saw that it's possible to isolate microbial community by using bioreactor. But that time in my lab at Bausti, we didn't have possibility or facility to isolate chicken gut by using those bioreactors. But I'm open if it's possibility to collaborate by using those tools. I'm really open to learn. And what are the potential consequences and challenges does this pose for maintaining a diverse and healthy microbial community within the chicken digestive system? As I already says, if you are able to use those bioreactors to isolate those or to mimic, let's say, the physiological environments of the intestinal gut, I think it will be really, really, really huge step on isolating and conserve those important microbial community, and that could be even applied to feed animal production industry, and also or even in human nutrition. I hope that I've captured all your questions. How do you handle sample from remote area where infrastructure and accessibility? So there in JQuat, that time in JICA, there was having an animal house there where we built the cages in order to limit the access of people. So we was really were like 2 to 3 people to work only on those chicks to limit access of people. And during the slaughtering of the chicken, we used I don't know, we used oh, I forgot the name, maybe in French, laminar flow, sorry. We slaughter the chicken under laminar flow to limit the contaminations or any contamination that could arise from the air or even from us. But when collecting each organ, after one chicken, you had to change the glove to limit the contamination. I don't know if I capture all your question. I capture all your question.
Thank you very much. There's also a question from you, for Dr. Armel, for you here from the floor.
Okay, thank you, Dr. Armel. My question is that during your study, did you consider like gender in characterizing the gut microbiome? Because like in some studies, like for instance in cows, we've seen like the hormones can play a huge impact on the gut microbiome. So in your study, I really don't know if you did or why you didn't. Thank you.
Dr. Armel, the role of gender on the influencing gut microbiome. You can unmute and answer.
Yeah, thank you, Prof. Yes, during this study, from the beginning of the project, we thought of also studying the effect of sex on the microbiome. It was one of our research question, but due to the constraints of the limited funds, we had to focus only on male. But we acknowledge that not studying the gender effect could be one of the limiting factor of this study. But we work in this study, we work only on male. But we would like, if it is possibility or any possibility of collaborating with you or anyone interested about this study, to work not only with male, to work with female, and also looking how the other genetics type of chicken behave, what could be maybe their let's say their microbiome signature. So we also look about, we know that the gender was limiting factors, but if it's possibility, we are open to test how the other breed, how the female could behave, and how the other genetic type will also behave. Thanks. I hope that I capture your
Thank you very much. Thank you very, very much. Yes, you have captured the spirit of the question and you have answered it very effectively. Ladies and gentlemen, this particular session that we have had through this morning was to capture the unique diversity of East and Central Africa in terms of species, in terms of organisms that are available, and also to appreciate some of the challenges that are present with some of this. So this brings us to the end of this morning session, and I'd like you to join me in giving a round of applause to all the presenters, especially those who are presented in this particular panel. Thank you very much, Dr. Amel, Dr. Patrick, and Dr. Joel, and Dr. John for your very, very effective presentations. And we look forward to seeing and reading your papers online as we go forth. And now I'd like to hand over the workshop moderation to my colleague, Dr. Catherine Ziomo, to come and take us through the next session, which is the gene technologies sessions. Thank you very, very much.
Thank you, Prof. Ann, for leading the morning session. Welcome back, everyone. We are moving on to the next session, which is on genomic technologies in agriculture. Yesterday, we heard about how we can advance infrastructure on the continent, how we can develop that. And for me, it's not just a technology push, but more of applications of this technology in Africa BioGenome or for biodiversity, as well as for agriculture and increasing our food security. So our first speaker is the head of emerging solutions for genomic applications at Illumina. Is going to talk to us about what they're doing and the technologies that are available and how we can also use those for the AfricaBP and the whole community here at large. So I would like to invite Xavier David from Illumina. Okay, sorry for that. So we're going to, okay. And the presentation is loaded.
Should I bring my PC up? Should I have my first?
Oh,
The name of the presentation.
they're asking for the name of the presentation.
Oh, should have AfricaBP. There we go. Fourth from the top.
Oh, okay. All right.
Thanks, good. Do I open it up? All right, well, hopefully that can be seen by those remotely. Brilliant. Well, good morning, everyone. My name is Michael Abdo, not my colleague Xavier. It is without question a pleasure to be here today. I have joined the previous workshops remotely from my home in Western Australia, and I would have loved to have been here yesterday, but for the fact that there is actually no quick way of getting across the globe. Well, not quick enough, as it were. So my role within Illumina is slightly different, and it does actually support Catherine's introduction in that I don't focus on product. I don't actually focus on geographies even. I can work anywhere between Cape Town and Tokyo. What I do focus on are those applications listed of infectious diseases, microbiology, and agriculture. So effectively trying to bring together genomics in that One Health realm. Now, my presentation will be quite different to this morning's session. It's not going to be particularly technical. What's more, I do have colleagues who are presenting during the course of today, and of course will be participating in and hosting the workshops for the rest of the week, which will give you an opportunity to delve into much greater technical detail. What I do want to do, though, is use this as an opportunity to address those in the room, those online that are perhaps early in their scientific careers, perhaps not that familiar with Illumina and genomics technologies, and give them some ideas for what is possible in this genomic era. And for those of you that have a few more years under your belt, hopefully give you some ideas as to how it might be applied differently to address some of the challenges that you all face in your day-to-day activities. For that, I will divide the presentation loosely into four subject areas: a very brief, simplistic introduction to Illumina, highlighting how Illumina has played in the genomic era, and then segueing to examples of our technology, sequencing in particular, at work in the field. Now, Illumina, if you look at our advertising, is typically aligned with improving human health through unlocking the power of the genome. But to state that really understates the opportunity of genomics and the responsibility that that plays in human health by actually addressing the inputs. And this is where the adequate supply of nutritious food, sufficient quantities of nutritious food, and then a safe and sustainable environment in which we live and the animals and plants around us also survive, particularly in an era of climate change, become integral to our mission as a company. What that means is there are, for the most part, no corners of the life sciences community that aren't impacted, that don't use, don't want to use, or don't benefit from genomics. And so from a commercial perspective, we address these markets of oncology through to agriculture and molecular biology. But then within those markets, the individuals, the customers, you in this room and online, are also addressed by all of the different sectors, from universities addressing basic research through to hospitals applying now precision medicine in the treatment of oncology. But if I extend some of the conversations and the presentations this morning, also precision nutrition through clinical analyses of the microbiome in patients, both healthy and sick with other related disorders. Now, for those particularly early on in your careers, you may not realize Illumina is 25 and has only been around for 25 years, and 25 years may seem like a long time. But in terms of genomic milestones, it is actually all but an adolescent. So in 1998, Illumina was started. It actually started as a microarray company, not a sequencing company. Massively parallel sequencing, as we now know it as next generation sequencing, didn't exist in 1998. And so with time, Illumina acquired sequencing technology, brought that into its portfolio, and has continued to evolve that and the use of NGS and microarrays through to the present day. Now, Illumina is very proud of that 25-year history, and it makes it one of the oldest genomic technology companies in the industry. But we need to put that into context. Now, the scale doesn't represent how big a divide there is between that 25 years and what evolution and nature has been doing for billions of years. So on the far left of screen, as you face it, about 4 billion years ago, life on this planet started. It's got a head start of 4 billion years. That's 4 billion years to refine what it's doing and why it does it. About 2 million years ago, Homo genius existed, started on this planet. And we've been refining what we do for that period of time. Fast forward to about 70 years ago, and finally we worked out the backbone of this thing called DNA. Fast forward another 40 years, and effectively in the 80s, this thing called PCR, polymerase chain reaction, was invented as a technology, which really accelerated our ability now to look at the genomic data, the genomic evidence that supported 4 billion years of evolution. At the turn of the century, massively parallel sequencing. And so this is why I say we really are at our infancy in regards to the use of genomics. And to the question around the relevance for students, I think this is perhaps the most exciting time to be starting your career in life sciences. I consider myself reasonably young, but even during my PhD, I poured my own sequencing gels and labeled the nucleotides with radio labels, P32. Developed those on film, and then read the film. Consider that, compare that to what you have access today on this site and elsewhere, both in Kenya and across the continent of Africa. Now, let's talk some practical terms. What does that mean? What can we do with all of that opportunity? The reality is, it tends to boil down when one is considering genomics era to opportunities to assess in parallel a number of targets rapidly. Originally, as per Illumina's evolution, this focused primarily on SNP genotyping using microarrays. And there is still a very valid place for genotyping based upon microarrays in today's application. But it has evolved, primarily driven by the reduced costs of sequencing, to now include next generation sequencing. And I don't really distinguish between whether you're approaching it from a long read chemistry or a short read chemistry. Fundamentally, the evolution of next generation sequencing has followed the same path. And access evolution driven by reduction in cost really has facilitated that. However, The key is actually knowing the question, the challenge, the biological hypotheses that you want to address in the first place. And this then becomes far more important than deciding long read versus short read, cost versus value, speed, time, flexibility, and the likes. So for example, if we started in the middle and we were actually addressing biological complexity, we might take vastly different approaches. Now, for example, if I was to be interested in an already domesticated species, there's probably no point in me focusing on sequencing. Rather, using microarrays, I can actually now analyze a far larger cohort and actually define perhaps traits that are going to, markers and traits that are going to be more relevant, traits that might define, I beg your pardon, the indigenous species versus, or breed versus a different breed, etc. Alternatively, though, if I'm actually approaching a species for which there is no known information, then, rightfully so, define your scaffold using long-read chemistry. But that alone may not actually also answer the questions that you need and generate the information you want, because nature has a 4 billion year head start on you. And so you might actually have to incorporate high C. You might actually have to incorporate a degree of epigenetics, short-read chemistry, to fundamentally define and curate an accurate representation of your species genome. Of course, access could be an issue. The sequencer that you might, or the microarray scanner that you might be wanting to use might be across a border, might be in a different country, a different city. And so that alone might mean tailoring the style or the scale of your experiment. Cost is inescapable, but here too, one can leverage the platform being a massively parallel sequencing to reduce cost, either by processing more samples simultaneously or actually looking at targeted regions as opposed to whole genome sequencing. Sample number might be your challenge. If you can't collect a large enough cohort to represent the populations, perhaps you need to look differently whether or not there are related species or breeds that you could leverage. Time. Perhaps you need the answer instantly. As we were aware during the COVID pandemic, time became critical in terms of managing new variants of concern. And so waiting until you accrued enough samples to run on a larger sequencing run and the likes over several days really was counterproductive in terms of managing the disease. Sample quality might be a rate limiter. Often the species you'll be interested in aren't going to be conveniently located in a sterile laboratory. We actually have a researcher we work with in Australia who is genotyping wild horses in the Australian Alps. Now, these animals are notoriously difficult to take DNA samples from. And so as a consequence, in the last 6 years that they've been doing this with the Illumina microarray, they've managed to collect only 400 samples. And even these, a good percentage, have actually come from carcasses. The reality is they will not get sufficient quality and quantity of DNA to do a long-read sequencing experiment with those samples. And so microarray really was the only platform for them. And then there are aspects of data analyses. The tools that you have, the need to share your data across borders with your collaborators may dictate the need to access local onboard computing power or cloud-based computing, and then all the requirements and the logistics and the compliance around sharing data on that scale. The bar has been set quite high, however. If you're familiar with the news of the last week, the UK Population Genomics Project released the data of 500,000 genomes that it has sequenced to those for public analyses. So the scope, the opportunities that you now have, for example, to cross-reference your species of interest to such a large data set are immense. And it might mean, if I come back to sample number, that perhaps you don't need an experiment of hundreds of samples. You can do much fewer, but then leverage publicly available data sources. A few examples, of course, are always great for a reality check. One such example for here in Africa has been Illumina's collaboration with the H3 Africa Consortium since its inception. And this really leveraged that idea of the right fit technology, starting with a microarray built specifically for the initiative, and then added to with content generated by sequencing or through sequencing. This being made accessible to a range of academic institutions and academic studies. And as a consequence, there have been more than 250 publications. Now, if you think about the author list around those 250 publications, that's an enormous community that have benefited from this single initiative. Part of that has actually been building the infrastructure. And I deliberately actually distinguish between capacity and capability. And I think this relates to the comments this morning around sustainability of sequencing and genomics technology. Capacity is one thing. It's very easy to place a box. In fact, the last thing we want is just to place a box. Capability, however, is your readiness, your preparedness to actually use that box. And that means taking and collecting samples, purifying the nucleic acid, processing the sequencing, which is actually, dare I say it, the easiest part, and then analyzing the data to reflect utility in that data. Otherwise, it is just a number. And if we think about just numbers, the number on the left, 0.2%, that is the percentage of animals that have their nuclear genome characterized and made publicly available. So to all you students, the world is your oyster. There are 8 million species on this planet. We've done 0.2%. 0.16%, the number of plants that have had their genome sequenced. And 90%, it's the percentage of marine species that are undescribed. That despite the fact that marine protein, seafood, is now a key component of most diets the world over. If we bring that relative to the framework of AfricaBP and much of the research that you do day in, day out, we can see that on the far right-hand side, animal health, biodiversity studies and the likes really benefit directly from genomics and its application. The utility of that, though, is much more than just a number, just a data set. And that's where it segues to food security, making sure that there are sufficient quantities of food in the right areas, that that food is of the appropriate nutritious value, and that the host's microbiome is such that that nutrient value can actually be realized. But it also goes beyond that. Looking to zoonotic diseases, reservoirs within the community, as the climate changes, as urban and rural areas interplay and interact on a far greater scale than ever before. And then our management, remembering that as humans, we've only been around for around 2 million of the 4 billion years, and yet our impact is quite profound. We haven't yet predicted the mistakes we've made today, but analyzing things and managing things like antimicrobial resistance through genomics technologies gives us a chance to avoid and avert disasters. If we consider that example of aquatic species, I'd mentioned that 90% were undefined genomically, and yet when you look at what is used for food and agriculture, there are more than 5,000 wild fisheries. There are more than 500 aquaculture institutions or activities the world over, all providing alternatives, sustainable alternatives to terrestrial species for protein. And in some instances, mechanisms by which you can actually increase the sustainability of a terrestrial farming practice. Seaweed supplements, for example, have been directly linked to reducing methane production in livestock. And there are now whole industries in Indonesia and even in Australia that produce seaweed exclusively as a food additive for livestock. When we think about plant-based crops, we can see from this illustration that in reality, we actually have a number of near monocultures that we are dependent upon. Now, some of those are highly diversified and resistant to climate change. Others are not. And in that instance, the opportunity with genomics is to actually address climate change, to adapt these rapidly, to ensure that yields without further damage, without further dependence upon fertilizers are maintained. And we can see this through a simple example of the chickpea, a key component in many diets, where it is actually in this study leveraged the very scenarios that I mentioned about the right size tool for the project. And so here, balancing a combination of both sequencing and microarrays, they were actually able to identify a number of appropriate markers for drought and heat resistance. Cattle, a key component of most agricultural industries the world over. There are about 40 different species. There are about 8,000 or more different breeds. Now, these have both economic and cultural significance and represents another opportunity to apply genomics to address both of those sometimes competing interests. And here too, a combination of sequencing and microarray technologies allowed this particular institute in in Victoria on the east coast of Australia to develop the genomic information which they've now applied to microarrays to routinely screen the herd in Australia. Indeed, the herds in Australia, the dairy herds in Australia and New Zealand, every head of cattle is genotyped. That affords those countries an incredible premium on the open market. Most of New Zealand's milk is exported to China for that reason, because of the quality, because of its quality assurance through genomic technologies. And more recently, if I think of this purely from a product perspective, and I should reference at the top right, this work was funded through an initiative of Illumina's, which the AfricaBP workshops were very, very helpful and prominent in promoting. It's the Greater Good initiative. This year's winner will actually be announced in January, so please stay tuned for that. But the previous winner utilized this opportunity again through the combination of a relatively small cohort of sequenced data to combine that with microarray technology and produce a product which is now sold by Illumina, but also utilized by researchers the world over to manage camel herds across the globe. And so, in summary, really, the key points that I hope to leave you, whether you are early in your career or more established in your career, is the reality of agrigenomics is that it is a commodity of the industry. Whether your focus is on biodiversity, whether your focus is on pure commercial output and the likes. And one doesn't have to be a large multinational institute to benefit from that. This can happen at any scale. And therein lies the opportunity for Africa as a continent to really leapfrog much of the time and investment that has happened in the Western world in managing and utilizing genomics in its agricultural industry to accelerate the industry, whether it's for conservation, whether it is for sustainable practices or otherwise here locally. And so to close, I really just summarize my my presentation, I should say, with this slide. Look at genomics technologies as a continuum. And the continuum is really around finding the tool that addresses your hypotheses. What's your greatest challenge? What are your rate limiting steps? What are the answers that are going to give you the greatest benefit? Whether it is that first publication, whether it is the final chapter of your dissertation for your PhD, or whether it is to actually manage your herd at a commercial level. The combination of sequencing and arrays allows you to address a variety of novel variants. It allows you to focus on known variants and traits. Combining that data really then allows you to manage and show utility to those efforts and yield a return. So with that, I do thank you for your attention here and online. And if there are any questions, more than happy to address those.
Thank you, Michael. I think we are just going to have questions all at the end for all the presenters. Let's give Michael another round of applause. And our institutions like Illumina and all the presenters at this session really give us the sense or contribute significantly to technology and also to improving the efficiency and effectiveness of how our genomic data is generated and also analyzed and handled. So our next speaker is Dr. Esther Kiragu, who is the Managing Director at Change Biotech, a company that is based here in Nairobi. And so it's good to also have that global experience and also a company that is based here in Nairobi. With that, Dr. Esther can have the floor. So your presentation here.
I think it's so many.
It's this one.
I just go to the. You can see my name. Just okay.
Okay. Do we click from here?
Yeah. Good morning. Still morning. Good morning, everyone. So. Okay. So I'm so grateful for this opportunity to be able to talk to you and interact with the scientists present today. And I'm really grateful to the AfricaBP organizing team for the opportunity also, and even for this great initiative where we get to interact and chart our path as African scientists on how we can be able to manage the genomic diversity that exists in our region. So I'm representing Change Biotech, and just a brief about Change Biotech. So it's a relatively young company in East Africa. Probably some of you have heard about it, some of you haven't. And it was founded in 2020, and we really are key on bringing positive change in the life science industry in East Africa. So we envision that the young scientists here are the people who will bring the next technological breakthrough, whether it's in agriculture, whether it's in clinical, whether it's in life science advancements. We know that you people are the ones who are going to birth that next big thing, maybe even a Nobel, why not? We can dream big. And we hope that that will come from East Africa. And that's why we want to be part of that big story in the life science industry in East Africa. So the company is led by a team of young directors who have worked on the bench, who have experience in doing science in the bench, and also who have been in the industry. So we understand the challenges that come in when you're running your samples in the lab and also accessing all these things. So we really want to be part of the change that ensures that going ahead, we are going to ensure that the industry grows as we grow together. We are a company that's based in Nairobi, but operating in East Africa. So if you're from East Africa, definitely we can serve you and we can work with you. So again, we work with partners who have the same vision as us. Change Biotech wants to bring technological advances to East Africa, and we've partnered with two major companies who are in the genomic space. So you might know of QIAGEN. QIAGEN has been a leader in extraction and PCR solutions for a long time. So we partner with them. And if you visit our table outside there, you'll be able to see, especially now we have the digital PCR machine, a technology that I believe that scientists should be able to use for the increased level of sensitivity and accuracy in PCR assays. So please visit our stand, we'll tell you more about QIAGEN. So for today, I wanted to talk about our other major partner, who is MGI, who's critical to this Africa BioGenome conservation discussion and what we want to do in the coming few years. So engage me as I tell you a little bit about MGI. And maybe as I start, who might want to hazard a guess on what the three initials MGI stand for? Just a guess, maybe. Someone, anyone, a company that has just three initials, MGI. Who wants to hazard a guess? Okay, so MGI, I don't know what the official, we've never known exactly what the official, so just like you, but in my discussion with the colleagues in MGI and AfricaBP BioTech, we thought it stands for making great innovations. And MGI as a company, that's what it has been doing. If you look at the history, so established in around 2016, mainly to develop sequencing technologies. So MGI is a subsidiary of BGI Genomics, and BGI has been known to produce sequences en masse. So some of you might have sent your samples for sequencing. When I was working in the lab, we used to send samples to BGI. So BGI acquired a technology called DNB-seq from a company called Complete Genomics. And since 2013, it has only been an exponential growth in terms of providing not only sequencing technology, but the machines, the technology to ensure that everyone is able to access sequencing products and services where they are. And previously, as I've said, we used to send samples outside. But right now, that's not the case. Whether you're a scientist doing high throughput sequencing, whether you're a scientist having few samples, you can be able to get the right solution for your needs. And that's why I say MGI is making great innovations, because as you can see with their history, they've just been making great innovations that you definitely need in your lab. So now I need to talk about the title of my presentation was Bringing Choice to Sequencing and MGI DNB-seq. So I've been asked by many people at a stand, what does DNB-seq stand for? So as a sequencing technology, as a next generation sequencing technology, the first step, as you all know, is library preparation. So you do your fragmentation, then after you do your fragmentation, the next step is usually adapter ligation, adding, doing end repair and all those things. But the end goal of your library preparation is a double-stranded DNA library. Most often, it can be different, but most often. But then that's where we come in now with our unique DNB-seq technology. So what is it about? So after you get your double-stranded library, then the next thing in the DNB-seq technology is that we usually separate the double-stranded fragments and work with a single-stranded fragment, sorry. And this single-stranded fragment is circularized. Then after it's circularized, we use an amplification technique called rolling circle amplification. And basically, this is very different from the routine PCR technology we know, because unlike the routine PCR where you make copies of copies of copies, here what you're doing is a linear amplification where you're copying the fragment, the original fragment that you had, you're just making copies of that in a rolling style. So you're not making a copy of a copy. So you continue making, in a rolling technique, you keep increasing the original template around 300 to 500 times. And if you think about that, the end result is a ball. So like if you have a ball of yarn, so you have just the same string turned around many times, and that's exactly what we have. So this is the DNA nanoball, and thus we call it DNB-seq. So then the DNA nanoball is what we load onto the flow cell or the array, depending on how you know it, and that's what we sequence. So what's unique about the DNB-seq technology? As I said, you're not doing PCR. So because of that, then you end up having higher accuracy, you have low duplication rates, and then you do not have the high chance of index hopping. And this technology has been utilized, as you saw in the history of MGI, it's been there for quite a number of years and is continuing to grow. Just an illustration of how we make the DNA nanoballs. So if you can see, I don't know if, okay, so so the single strand is separated, then circularization happens, then you make the DNA nanoball, which is just a long continuous ball of the fragment that you intend to sequence. So then the next thing is we load the DNA nanoball on the flow cell, on the patterned flow cell. And this is where the goal is to now sequence and make sense of the nucleotides. We use a patterned array on our flow cells. And this is to ensure that, as I said, you have less index hopping and you have that a precise location loading on your flow cell. Then after that, it's the CPAS sequencing, whereby now you flow your nucleotides. We use fluorescent nucleotides and your terminator enzyme, and then you get the sequence generated. And this is what the machine will read and give you now the sequencing report. As you can see, the sequencing output is the same across all sequencers. Even for us, it's an MGI FASTQ format, which you can now go downstream and use it for your bioinformatics analysis. So as a summary on what DNB-seq technology is, as I said, you first make a DNA nanoball. So a DNA nanoball, you make it using the rolling circle replication, just copying the original fragment many times. Then we load it on the patterned array, and the patterned array, the MGI patterned array is a proprietary technology where it just allows you to have reduced index hopping and even reduce the chance of getting duplicates. Then after that, it's the sequencing, where now we make sense of the nucleotides that are being synthesized. And in summary, that's the technology. So just a brief, I mentioned it, but I'll just repeat briefly. I said the advantage of DNB-seq technology over using conventional PCR amplification is that in PCR, you make copies of copies. But in DNB-seq, remember, you're only copying the original fragment that you got during library preparation. So that ensures that you have low amplification bias and you do not have an accumulation of errors that can come as a result of PCR artifacts. So yeah, so just to say that the key advantage of the DNA nanoball is the increase in signal. So the increase in correct signal. So when you create the ball, the massive yarn of the nucleotide that you fragmented, then the signal is enough to be able to be detected accurately when you do the sequencing. So we can also do the long fragment reads. Some of you have used the long fragment sequencing technology on Nanopore and the likes. So we can also be able to use to create a short read long fragment, and we currently can go up to 40 kb. And this one, as you've seen, it's important for when you want to do SNP detection, when you want to look at indels, copy number variations. So we have a technology still on the short read platform, but able to combine and create a long read fragment. And we can tell you more about it if you come to a stand. It's actually possible on our sequencers. So the DNB-seq technology has been growing by leaps and bounds. From around 2016, where we had only 2 publications. Currently, the publications are at least at around 2,000, and the publications cover all applications and across different species. So you have RNA-seq, you have metagenomics, you have, as I said, the short read, long fragment read technology, reproductive health, oncology, we cover across all. So the DNB, you have the confidence that the DNB-seq technology can be able to answer your question. And you can be able to refer to publications, peer-reviewed publications, which are already there in the databases, to be able to even give you more confidence if you want to utilize it. So MGI does not only produce sequencers, but it caters for the whole workflow, meaning we have solutions for library construction, then we have the sequencer and the sequencing solutions, and even the bioinformatics solutions. So if you want to do library prep, we have library prep solutions to cover all the different applications you're interested in. If you're doing whole genome sequencing, if you're doing a single cell sequencing, we have library kits that can cater to that. And then we have a range of sequencing instruments that cater for low throughput and high throughput needs. And I'll tell you more about it in another slide. Then our bioinformatics solutions are catered to both very experienced bioinformatics scientists or even beginners. We can be able to give you a platform that allows you, if you're not very good at bioinformatics, and also if you're an experienced user, we have a solution for you. Again, MGI has automated products especially for library preparation. And one unique thing about our automated lib prep solutions is that we not only have solutions that work with MGI library kits, but we also allow third-party kits. So if you have a very nice kit that you think works for you and you want it automated on the MGI platforms, then it's very possible. We'll only make a script, and that script will allow you to be able to utilize that your library on an MGI, either the SP100 or SP960. So if you want to increase your throughput and you want to do automation, please don't hesitate. We have a solution that can work for that. Then, as I said, I'll talk about our sequencing, our high throughput sequencing technologies. But again, if you want to move from low frequency, low numbers, and you want to increase the number of samples you're sequencing, we have solutions for that. So let me show you a bit about our genetic sequences. So I've said that we have sequences that cater for all throughput. And for the high throughput needs, we have a sequencer that can give maximum output of 72 terabytes per run. Can you visualize 72 terabytes? That's a lot of data. So that in the TX series, those are the sequences that are used for population genomics and will be actually appropriate for the Africa BioGenome Project needs, because these are huge population studies looking at many species, and these are sequencers that can be able to deliver quality data for that need. Then we have the T7, which is 6 terabyte, again, a very high throughput platform. Then we have the medium throughput needs, the DNB-seq G400. This one, the data output ranges from around 300 to 800 GB. Again, can cater for all sequencing read lengths, and has been lots of publications on the G400. So it's a machine you'll find in clinical, in agriculture, in all applications. So has been used in most of the publications have been done on the G400. It's a beloved sequencing machine. Then we have the G50, which is now in the low throughput, where we have at least between 30 to 150 GB output. And then we have the E-series. So the E-series is where we cater for portable mobile sequencing needs. So if you want to go and do sequencing in the field, maybe do rapid pathogen identification, then the E-series is the solution that we have for that. So the previous presenter mentioned about how the cost of sequencing has been coming down, and I fully agree with that. We have been moving from 3 billion cost per genome during the days of doing the slab gels. And then we had the Sanger sequencing, a good technology, but still quite expensive. Then I think I should. Oh, oops, okay. Okay. So so then we moved into the sequencing synthesis technologies, and the cost really came down. So now we are in the range of sub $1,000 per genome, and we are confident that it will keep going down and down and down. And MGI DNB-seq technology has a key role to play in that. If I can show you, so currently we are around $500 per genome on some of our sequences, and we believe that it will keep coming down to even maybe $100 per genome. Then we have two new kids on the block for sequencers, and this is a new low throughput sequencer that is very fast. It's an ultra fast sequencer that has an output of at least 8 to 48 GB per run. It can accommodate 2 flow cells, and it can accommodate quite a number of read lengths. And this is very good for applications that focus on maybe oncology for clinical researchers and pathogen identification. So for people who are starting in their sequencing journey, this sequencer released actually for our market in June this year is a sequencer that's going to be a game changer. So fast, and it also has integrated bioinformatics. If you want to make it easy for you in doing the analysis, it has an option to incorporate your bioinformatics. So this is a new sequencer, and we can tell you more about it if you come to our stand. And as I mentioned, we envision the cost of sequencing going further down. So in the TX series, currently in some regions in the world, they're able to achieve at least approximately $100 per genome, especially for population genomics. So it's not crazy to say that in the next 10 years, we'll be sequencing at $100 per genome, even in our region, because the sequencing technology keeps on improving, the outputs keep on increasing, so we can be confident that everyone will have a choice and be able to achieve, massive output of sequencing data. So finally, I'll just say as I said, we are very happy to be in this forum created by AfricaBP. And we, especially in their goal 2, they want to increase, output and sequence over 105,000 species in East Africa. Of course, massive sequencing technology will be needed. We are here to support you. We have the team of scientists ready to work with you, support you both in the lab work and also in the bioinformatics. And we are confident that together with you scientists, when you go back to institutions, we'll be able to come and visit and give you more about how we can do this and achieve sequencing all these species, and even contributing data to the public databases. So we are confident that we're on the right path. So when you go back to your lab, whether you want to do your PhD, whether you're looking at your postdoc and you want to do some projects, please talk to us, come see our team, and we'll be able to see how we can further work together. So I'm really grateful, and I'll end my presentation there. If you have any questions, please, you can raise them or you can come see me after. Thank you.
Thank you, Dr. Esther, for your nice presentation and for giving us a perspective about what's possible and what options are available for sequencing. Indeed, the AfricaBP is an ambitious project. We'd like to sequence 105,000 genomes. And so with that, definitely it's important to have options in terms of where we can do that work and also who we can work with to be able to do that work. I was really impressed by your company, fairly new, but really having a regional presence and wanting to build capacity for African scientists. So we're going to move to the next presentation. Our next presenter is Dennis Kivuva from PacBio Biotech, another sequencing company that is also here in Nairobi. So. Is it here or it should be?
Good afternoon. Yeah, sorry for the delay while we are trying to load the presentation. So I'm Dennis Kivuva. I'm the Sales Operations Manager for Inkaba Biotech East Africa Limited. And I'm just going to briefly try to demystify sequencing NGS to those who are new to sequencing and the aspiring students who are looking to go into sequencing. So at Inkaba Biotech, we supply NGS services, genomics products, and among these products, we have universal products which are for universal applications. So when we are talking about sequencing, when we are talking about NGS, to some people, it sounds like that you need to get a full solution from one company and apply that. So we are here to tell you that we have third-party products which can be used with most of the mainstream sequencers. That can be maybe, for example, maybe more accessible and cost-effective for those who are resource constrained. So, for example, I'm going to briefly touch on two products. The first one will be more of a kit. It's from the NEB profile. We do have the NebNext Ultra Express library preparation solutions. So NEB basically manufactures reagents. They do not have their own sequencers or equipment. They mainly focus on reagents. So they have this NebNext portfolio. In the NebNext portfolio, we have library preparation kits which cater for such brands as the Illumina brand of sequencers, as well as the Oxford Nanopore brand. So I'm going to introduce to you the NebNext Ultra Express Library Kit. They have both for the DNA and the RNA. So this kit is an ideal kit for most of our market segment because it touches on the most important aspects of NGS application. So the first thing that I need to point out is that the NebNext Ultra Express library kits have been streamlined for the fastest preparation from the genomic DNA to get a library. They have reduced the number of steps that you need to get a library from the genomic DNA, and the reduction of the number of steps implies that you need to use fewer consumables. That's the tips and tubes. So that also has an implication, the cost implication. So you'll need fewer. You'll use fewer resources. You'll save up on the money that you have spent for that. The Ultra Express kits are also very flexible with different sample types and different protocols, and they can be used both on manually, and they're also automation friendly with most of the mainstream automation systems. So just to touch a bit on the points I listed out, the NebNext Ultra Express kits, the library preparation kits, they are faster in the sense that for DNA library preparations, it takes less than 2 hours, while for RNA, in about 3 hours, you get your libraries which are ready, including the poly-A enrichment and rRNA depletion. So for both DNA and RNA applications, you can get your libraries within a day. In a single day, you can do the all workflow. So this is achieved with the fewer steps that I said before, and with fewer steps, with fewer consumables, you're also saving up on the investments that you're putting in that. And they have been designed for a wide input in terms of the DNA quantity that you're starting with. So for example, for DNA, the NebNext Ultra Express kits, they accept an input of from 10 to 200 nanograms of pre-shared DNA. And for RNA applications, the input range is between 25 and 250 nanograms of total RNA. So the NebNext Ultra Express kit features a single protocol for all inputs. So this also simplifies the use, even for those who are new to the NGS world. It's a very easy kit to use. As I mentioned, this portfolio is automation friendly, so it can be used with most of the mainstream automation. So any bid they do not have a preferred system, so it's open. I would say it's an open kit that can be used with most automation platforms. So focusing specifically on the DNA library prep from NebNext Ultra Express, you can go from pre-shared DNA to a complete library ready for sequencing in under 2 hours. I think I mentioned that. So the libraries that you make with the Ultra Express DNA kit, so they can be achieved with a single protocol, which again is touching on the simplicity of the kit for those who are new to NGS. So the workflow for this kit is in a single tube. That's meaning you are using less consumables, and you're also cutting out on the possibilities of contamination when you are switching off over consumables. And the flexibility that comes with the kit also accommodates for customized protocols if you desire, but it's also a complete kit if you do not need to customize it. So the workflow for the DNA kit, you start from the shared DNA. From shared DNA to the DNA tailing, adapter ligation, PCR enrichment. The PCR cycling, you go for 8 cycles, and then the cleanup. So the total workflow is roughly 110 minutes. And with this workflow, you get a very robust library with uniform GC coverage and insert size of a wide range of input. So for example, you can see from the graph for the input of with an input of 10 grams and 200 grams, the GC content on the second graph, you see that the GC coverage is consistent over all the input sizes. So yeah, the NebNext kit will give you that kind of uniformity over the different ranges of input. The workflow for the RNA kit from the Ultra Express portfolio, it's also so the RNA enrichment, the poly tailing, and the rRNA depletion, that takes roughly 1 to 2 hours, and then the fragmentation, first one synthesis, second one synthesis, and cleanup, the DNA tailing, adapter ligation, that will take roughly about an hour, so giving you a total of 3 hours at the maximum. So the RNA library prep kit also produces high library yields across a different range of inputs. So you can see from the graph the yield from 25 nanograms, 75 nanograms, and 250 nanograms. So we have NebNext also, they did a comparison against other currently available options in the market, like for example, the Kappa hyperprep and Illumina stranded, and you can see from the output of a different input ranges. So secondly, so I've basically touched on the manual library prep solution, the kit. So I'm also going to introduce to you the automation side. We have a platform from Mira, from BMS. So it's called the Mira. So BMS biological systems. BMS. So they are the manufacturers of Mira, and I'm sure most of you are already familiar with the smallest qPCR machine, the MIG. So the Mira is a liquid handling system, which is the first to have an integrated camera, which ensures precision pipetting, and it is a very compact system. It's a very small and compact system, which weighs a mean 10 kilos and a footprint of less than 0.2 meters square. So it's basically half the size of the other currently available liquid handling systems in the market, which have the capacity to do NGS library preps and such other applications as PCR setup, sample pooling, cherry picking, and basically liquid handling, even for 384 wells. So the Mira is ideal for NGS library preparation applications. It's also ideal for cell media washing and for determination of minimal inhibitory concentrations in antibiotic susceptibility or resistance applications or projects. So just to go a bit more into detail about the Mira. So as we all know in science, we are very keen on the precision and accuracy of systems to achieve quality results. So we all know that what precision is and what accuracy is, and the Mira has been tested to attest that it provides the highest precision and highest accuracy possible for an equipment that is to be applied in the NGS world. So, for example, I should mention that all the data that is provided here is tested with the Mira and the MIC. So, to show the precision of the Mira, 48 replicates are done. Extraction is done from 48 replicates on the Mira, and amplification done on the MIC. And you can see from the output that there's a lot of repeatability. You can see all the 48 samples fall in the same range, with a variance of 0.05 from the CT value of average of 19.23. So that's very high precision, considering that the starting template is of 1 microliter in a reaction of 25 microliters. Even at a low copy number, so the previous one was a reaction of the copy numbers was 10,000 per reaction. So the reaction was repeated with low copy numbers of 100 copies per reaction, and still a starting template of 1 microliter in a 25 microliter reaction. And you can still see that there is a lot of repeatability. The repeatability for 48 samples is quite noticeable. These are quite a robust system. You can see that the variance is 0.11. So that's quite high. And it proves that the Mira is very precise. So 48, another reaction of 48 replicates, repeated 3 times on the MIC again, and we still get such kind of repeatability with a variance of 0.11. So all the 3 MICs were analyzed during using the same feature, but on different machines, on different MIC machines, but we still get the same kind of repeatability. So about accuracy. So a 7.10-fold dilution series of a template was done in 4 replicates. Still, again, extractions done and dilutions. The dilution series done on the Mira, and then detection done on the MIC. And you can still see the accuracy is high, and at a starting concentration of the starting template was 1 microliter in a reaction still of 25 microliters. So accuracy and precision has been tested over several trials on the Mira and MIC, and it has been proved that it can provide, even at the extreme concentrations, it can provide consistent results. And it's an ideal system that can be used for NGS preparations without doubt of getting any biased results. So a complex pattern of various dilution factors from a 10-fold reaction to as low as 1.2-fold, it's achieved on the Mira setup and on the Mira extraction and the MIC detection setup. So just to mention about a bit about the Mira. So BMS provides very good support and very good facilitation for using the Mira, especially for NGS applications, with some common scripts available on their website. For example, for pooling, for Illumina DNA library prep, for serial dilutions and all that. But still, it's open to coding, whereby if you are familiar with Python and a bit of coding experience, you can even input your own scripts and apply it on whichever application you have in mind. So just to give a brief of the specifications of the Mira, as I mentioned, it's a fairly small equipment with dimensions of width 36 centimeters, 46 centimeters long, and 31 centimeters of height when the lid is closed. It weighs 10 kilos, and you can see the precision and accuracy down there. And yeah, so it's a very robust system, which is ideal for liquid handling and NGS applications in the context of library preparation. It's also an open system, so you can bring in all the automation compatible kits, like for example the NebNex that I talked about previously, and from other manufacturers. Of course, there are many library prep kits which are compatible with automation. So yeah, just as I finish, I wanted to say that we are here as in Kaba Biotech. We are here not just as suppliers. We are here to support you. We don't just stop at where we drop the kits or the equipment to you. As a service provider, we also give the support. So if you have any challenges, even in designing your projects, in starting your projects, we are here to help. We are not biased. We will not restrict you to what we supply. We offer you the support, and then for products that we supply, we'll work with you until you get the results you're looking for. And with that, I want to say thank you. And if you have any questions, you are free to visit us at our stand. We discuss more, and yeah, feel free to interact with us. Thank you.
Thank you, Dennis, for the very good presentation. Indeed, now we have the technologies here. We now have the sequencing machines here. And it is now a question of the support that is required after you sequence the data, after you have the results. And it is good to know that institutions like Inkaba do offer that support post the sequencing, post the work in the labs. And also the issue of the reduced timing, the efficiency that your work, that your systems or the infrastructure has is really impressive. So we are a bit behind time, and we're going to really move through the last presentation so that we can take questions and also have the last presentation for this session. Our next presentation is from Dr. Sean Edwards from Illumina. We had the first presentation from Illumina about advancing agri-genomics technologies, but now we want to understand the power of the genome from Illumina again. Oh, okay. It's okay. Thanks.
Yeah, good afternoon, everyone. So yeah, unfortunately, I'm not Sean. I'm here presenting on his behalf from Illumina. So my name is Peter Ndega. I'm a field application specialist working with F&S Scientific. F&S Scientific is the channel partner for Illumina. In other words, we work very closely with our clients, with our Illumina customers, to ensure we provide them with an end-to-end support when it comes to genomic solutions. And this is why we considered to partner with Illumina towards this regard. So the title of my presentation is Unlocking the Power of the Genome, an overview of Illumina sequencing technology. So personally, I'm more of an application guy, so expect more of the application and techniques more than sales. So I'll be delving more into what makes Illumina tick? Okay, so I thought I should start with this particular slide and state that Illumina has been a very key driver in two key technological evolutions, that is genome sequencing and SNP genotyping. And as it was stated earlier by the earlier presenters, the goal is essentially, or we envision, to reduce the cost of DNA sequencing and accelerate genomics globally. And in terms of genotyping, we have more than 55 arrays that we've developed for plant and animal sciences, animal species, and there are many more to come. So we have more than 20,000 systems, Illumina systems installed globally. So this is a testament of how dominant our sequencing platforms are. So this slide categorizes, or rather shows you the different platforms and how we've categorized them in terms of the applications that our customers are getting into. So we have low throughput systems such as the MiSeq, and I should state that the MiSeq is one of our most popular platforms. We have the MiniSeq, the iSeq. These fall into the low throughput category. We have also the mid throughput, that is the NextSeq 550 and the NextSeq 1K and NextSeq 2K. We also have platforms that serve the diagnostic applications such as the MiSeq DX, the NextSeq 550 DX, and the NovaSeq 6000 DX. So for high throughput applications, we have the iScan system, the NovaSeq 6000, and our newest baby, the NovaSeq X and NovaSeq X Plus. So when it comes to our customers, we ensure that we have them covered in all aspects. And as F&S Scientific, the channel partner, when it comes to decision making into what platforms to go for, we are always eager to support and advise accordingly, so that you go for the platform that truly serves your needs. So on the continent, this slide was earlier shared by Michael. We have quite a heavy presence on the continent, and this shows the impact that we have in the African continent. So you can see we have over almost 130 MiSeq instrumentations, 25 NextSeq 2000, and 35 MiniSeq platforms in the country, in the continent. So in other words, Illumina is a very reliable partner when it comes to genomic sequencing. And what I'm going to delve into is there's a reason that Illumina is dominant. And this, one of the key reasons is the chemistry that underpins the technology, the sequencing technology that Illumina employs. And just briefly, the technology is a sequencing by synthesis chemistry or technology. And basically, this technology occurs on a flow cell. So the sequence of bases is determined through a combination of colors and signals and fixed known position of each template physically attached to a flow cell rather than the transport of labeled fragments through a gel. So I'll highlight more on the SBS chemistry and why it makes Illumina tick and reliable in terms of genomics. So in a nutshell, this is the Illumina sequencing workflow. It has 4 basic steps. That's library preparation, cluster generation, sequencing, and data analysis. And just to go deeply into each of these. So when it comes to library preparation, essentially before you sequence, or rather before you sequence your DNA, you need to have it prepared. And the starting materials need to be fragmented. So essentially, you want to fragment your DNA into manageable pieces. And then from there, you add adapters. These are small synthesized pieces. And then you give them to each of the DNA fragment to make libraries. So in this slide, essentially, you want to end with a complete library. For clustering, you want to ensure that you have the P5 and the P7 oligos that will complementarily base pair, or rather bind, hybridize to the oligos that are on the flow cell, on the sequencing flow cell. As I highlighted earlier, the sequencing occurs on the flow cell. You also want to ensure that you have the sequencing primary region, binding regions, which I highlighted here as RD1 and RD2. And also because in most cases, you'll be batching your samples, you need to index them or rather barcode them so that you can multiplex them later and identify them. So in terms of cluster generation, so you've prepared your libraries. Now you want them to hybridize onto the flow cell, and then you amplify them through a process called branch amplification. So what branch amplification does, it generates for you these clonal clusters that enable the massive parallel sequencing of your DNA libraries. So what is a cluster? A cluster is a group of DNA strands that are positioned closely together. So each cluster represents thousands of copies of the same DNA strand in a 1 to 2 micron spot. And as you can see, so essentially what you want is to ensure that you have multiple clusters that are massively parallel sequenced. And this improves the confidence of the base calls that you have at the end of the day. That is the accuracy of your sequence data. So the flow cell, this is where all the magic happens. So the cluster generation occurs on the flow cell. And this is usually a thick glass line with channels or lanes. So for the different platforms, some utilize different flow cell technology, something that I will highlight later on. So each lane is coated with a loan of oligos, which are complementary to the library adapters. So once you've generated your clusters, you proceed to perform sequencing using the sequencing by synthesis chemistry. So here you use fluorescently labeled nucleotides, which are incorporated. So these bind to the complementary DNA bases in the clusters. So what happens is that the instrument images the flow cell, and then the image information identifies the first base in the cluster. And then this process is repeated to determine the sequence of the bases on the clusters, one base at a time. So this is a closer look at the 4-die chemistry that is employed in the MySec platform. So you add your fluorescently labeled nucleotides, and then these nucleotides are excited, and then they are imaged, and then the terminator and the fluorescent dye are cleaved from the fluorescently labeled nucleotides, and then you proceed to synthesize, or rather to sequence the fragments, as you can see there. So sometimes if you are like me, I'm more of a visual person, so I understand things better when I sort of visualize them. So I thought it was important to include this to explain the sequencing by synthesis workflow.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of data analysis. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in Base-Space Sequence Hub. Discover the possibilities of next-generation sequencing.
All right, so yeah, so Michael mentioned that Illumina has been here for a while, and what has been happening over the years is Illumina has been evolving its SBS chemistries to ensure reliability. Because as I said earlier, at the end of the day, you want to ensure that whatever instrumentation that you're working with, you can have reliable sequence data. And with all the innovation that has been happening over the years, Illumina is a definitely reliable partner in that regard. So you can see there are different channel chemistries that have been utilized or employed for the different platforms. There's the 4-channel chemistry that utilizes all the 4 dyes. We have 2-channel chemistry and also 1-channel chemistry. So what I should mention here, despite the different chemistries, the quality of the data is not affected, okay? Now, recently, a new chemistry has been introduced, referred to as the XLIP SBS chemistry. And this is a revolutionary way to achieve the SBS with higher accuracy and faster spins and greater stability. So in this particular chemistry, what has happened is the polymerase, the XLIP SBS polymerase has been engineered in such a way that it ensures high fidelity and spin. So essentially, the fluorescent, or rather the XLIP nucleotide, contains a new 3' X block that terminates the strand extension. And then the fluorescent dye is attached by the X-linker chemistry to the remainder of the nucleotide. So after one incorporation step and imaging, both the block and the dye are removed in a single rapid cleavage step, facilitating the next nucleotide addition during SBS. So what are the improvements that as this chemistry brought to SBS? So one of them is the stability of the nucleotide. So XLIP SBS nucleotides are more stable to thermostress than the standard SBS nucleotides, as you can see in the figure here. And also, using this new chemistry, there's a decrease in the SBS cycle time. So the polymerase enables 3 times faster incorporation and fast deep lock of the XLIP nucleotides, contributing to a 2 times decrease in SBS cycle time. And the other thing is about improved sustainability. So this chemistry has enabled improved reagent stability and ambient shipment to the customer. So ambient shipment removes the need for dry ice and reduces the size and weight of the delivered consumables. So this particular chemistry is going to be supported on the next 2000 and 1000 as from next year. So at the end of the day, you generate your data. So data analysis can occur in different phases. There's a primary analysis that occurs in real time while the data is being sequenced. And then you have the secondary analysis. This gives you information about the alignments and the variant detection. And then you have the tertiary or data visualization, where you can be able to visualize your data, your reports, and annotation as well. So how do you maximize genomic insights? Yes, you've generated your sequence data. How do you get the maximum out of that data? So bioinformatics is a challenge that cuts across board, regardless of whichever sequencing platform that you're using. But what if you could detect variants regardless of type and the tolerant to population bias? Or what if you could access comprehensive pre-configured pipelines in a single platform? Or what if you could accelerate secondary analysis beyond the primary and the data is turned efficiently? So these are some of the questions or some of the issues that Illumina has tried to cover by developing the Dragon for secondary analysis. So this is a collection of bioinformatic pipelines that support data analysis beyond the generic data that you get. So this is a collection of the different bioinformatic pipelines or applications that are being supported by Dragon as of now. So the reasoning behind this is to ensure we provide our clients or our customers at least with data that they can proceed to a certain level, that they can proceed to get more insightful information from. So, and the advantage of this Dragon platform is that it is accurate. So it is updated in real time with several collection of databases globally. It is comprehensive. It measures the full complexity of the genome, and it is very efficient. It accelerates analysis and lowers the cost. So with all these things, so you can leverage on our Illumina technology for its simplified library prep. You can customize content if you're working with panels, for instance. Depending on the application that you're working with, we have versatility and flexibility of the application that we support. And also we do have integrated analysis that comes on board on some of the instruments like the NESTSEC 1000 and 2000, and the high throughput platforms like the NovaSeq. So just to conclude, so for those who are lucky enough to be selected to participate in the workshop that is beginning tomorrow, so you'll be learning more on sequencing on the next SEC 550, and we'll be working on this protocol, the Illumina DNA PCR-free protocol, that essentially incorporates or rather includes the following steps. So we're looking forward to your participation tomorrow. Yes, and thank you very much for your attention.
Thank you, Dennis. We've come to the end of this session. And this session has been very, very useful in terms of learning about the technologies, what's available, and also at what cost. So the exhibitors are here. You can have a chat with them outside and for further information. But I'm going to take a few questions from the room, and also, okay, there's one here. You can go ahead.
Okay, good afternoon. I'm Kimani from Iriri. I wanted to ask the people who are maybe the companies themselves or the people who are delegated to supply reagents and equipment, why don't you start offering services instead of selling equipment? These technologies are changing very, very fast. Within 5 years, you find even if it is not completely obsolete, there are new technologies. Why don't you offer services instead of selling the equipment and the reagents? Also, for us here, we've been having challenges. You want to sequence, you find a company in Singapore is cheaper than a company that is in Africa, because they'll also even facilitate the shipment. If I want to ship RNA, I'll be required to ship RNA using some of these companies, and it will cost me more than $1,000 just for shipment. But if you have the company here doing the sequencing, I'll just use dry ice to transport my samples. Thank you.
Thank you, Wilson. Any particular company you'd like to address this?
Okay, there is the new, Illumina and also the MGI. The MGI house, they are very new. They can take this opportunity to, instead of selling us the machine is very, very expensive, they can provide that service, and it will be so cheap, and also the time that it takes. I think like 1.5 years ago, I took more than 3 months to have my samples after I've prepared. It took a lot of time to set the samples because I need clearance from CAFIS and no other companies, other organizations. Sorry.
Okay, thank you. Dr. Esther, you want to take that one? You can use this.
Okay, thank you. Thank you for your question. So, and thank you for raising the concerns you have about the time it takes to supply reagents, the cost about shipping. So yeah, as a new company, we are really trying our best to address that. And one thing I'll say is that for us as MGI Change Biotech Partnership, we understand that technology keeps on changing. So one way we are addressing that is saying that we want to place the equipment with you. We're not asking you to outright purchase the equipment, so that in case the technology has changed and your throughput needs have changed, we can then take the old machine and give you a new machine. So try us for that one. And then in terms of supporting the scientists to have the technologies here. So our business is mainly in terms of providing the solutions that you need. So what I would throw the question back to the young scientists here and say, we want other young scientists to now start a sequencing business locally. Then we are very willing to provide you with the technology and the support that you need. So let's get into this as scientists. I've talked to a few people and said, let's get into this business of commercializing the knowledge we have. And together we provide the solutions, they provide the services, and we grow the industry. Yeah, so please come to a stand and I'll talk to you more about it.
I will attempt to unpack some elements of your question. I guess fundamentally, and it's interesting that I should follow MGI because MGI is a subsidiary of BGI, which became famous for purchasing 256 high-seq sequences. They were the first factory-scale service provider in the world, enabled with Illumina technology. Now, from Illumina's perspective, your specific question was, why don't the manufacturers become service providers? In simple, it's because our expertise is in the technology of sequencing, refining SBS chemistry. So not changing, but refining. Our expertise is not in understanding the possible 8 million different species that might be your focus. And that's where the challenge in service provision comes. So service providers, whether they are institutional, whether it be based here at ILRI, or factory scale, Novagenes, BGIs of the world, will all have specialties, and their prices reflect that when you deviate from their specialty. The other aspect then comes down to cost. And there, again, I would encourage you to, for all aspects of your project, look at different tools available to you to address costs. So if it is the challenge of shipping fragile RNA, consider doing the cDNA conversion yourself and ship the cDNA instead and reduce the costs there, potentially accessing DNA sequencing prices as opposed to RNA sequencing from the service provider. There are many ways that one can address this, and here it's where you can call upon the companies, Illumina, channel partners. I'm sure the same applies for AfricaBP Biotech as well, to really unravel what's going to be the most appropriate library preparation, what's going to be the best experimental design, and then make your choice from what is a very competitive market. Because fundamentally, one aspect of that reduction in price is actually competition. Competition is healthy for all of us, whether we are competing with our peers to be the first to publish, or whether we are, in fact, the sequencing companies trying to be the one to produce the box that produces the largest amount of sequencing capacity, or the data analysis solutions that allow you to generate utility from that data sooner rather than later. So unfortunately, I don't have a specific exact silver bullet to address your answer, but hopefully that's at least alluded to some of the facets of what you asked.
Thank you, Michael. I'll take another question from the room, and then I don't know if there are any online. Any other? Yes.
The question goes to Peter. Yes. My question is about, first, my name is Kennedy from Medical Research Center. We are located in Thika. So my question was on more on the application. So I just wanted to know, there is part of the sequences that you said there is that of enzymatic activity. I don't know whether there is specific enzyme for polymerase that you are using, like whether it's TAC or any other, and which one specifically, why did you choose that? Is it TAC polymerase, or what is it? Yes, yes. Okay.
Yes, we use the transposase enzyme to perform enzymatic fragmentation. So, and what happens can add more onto that is most of the newer library prep kits that you're using for sequencing, to enhance the fragmentation process, we are linking the enzyme to the beans. So you find that most of the library prep.
There's an advance in the technology. Is it possible to have some of the solutions which are adaptable to the new consumables already in the market? I guess it's just food for thought, and something that can be addressed, I guess, with the increases in efficiency and reliability of the technologies. So our last presentation for this session is coming from Dr. Shem Kuya, who is a senior lecturer at the University of, and the Director of Research and Innovations at Jomo Kenyatta University of Agriculture and Technology, based here in Nairobi. He's a research fellow at the Swedish University of Agricultural Sciences, and also regularly partners with CG centers, C4 ECRAF, on various research activities. He holds a PhD in plant science from JQuat and also a postdoc from the University of Bonn, with the support from the Alexander von Humboldt Foundation. So he's going to speak to us about, his topic is scientific collaboration and communications, which is a very important aspect of the work that we do. If we don't communicate clearly and effectively, all the science that we do can become a waste. So with that, I will hand over to you, Dr. Shem.
Thank you so much. Good afternoon. Great, it's been a nice session of learning, and particularly happy to be here. The last time, and maybe the first I was in ILRI was when I was undergraduate looking for an attachment, and this is my second time when I'm an old person. Tells you that it's a precious place. I'd like to talk about scientific communication, and in a brief time to allow a question or two for us to engage and discuss. We know that science produces information that is really useful to the society. And this information can be used either immediately or eventually. For example, when we do our fundamental research, it can form a foundation for other activities. And so this information is supposed to create some kind of impact. And mainly we do this by writing. So we're communicating for us to bridge the gap between science and the public. But then there are also other gaps between practice and policy. And so because we're saying that writing is one of the most common way of communicating our science, one of the things that I can ask us, and because I know we are a little bit, we've been sitting a lot, just look at your neighbor and ask them, why do you write? Let's see. Demand an answer, just demand an answer. Can you get one? Did you get an answer? If you just got an answer, tell me. I know a PhD student might have said, I write because I've completed data collection. I write because I want to submit this thing. I want to start sleeping. For those of us who've been in the research for some time, sometimes we write because we're told it's a way to prosper or to avoid perishing, right? And so there are many reasons why we write. I also looked around why people write, and I got 5 reasons. One is to be read, okay? I got a nice story and I'm writing to be read. One is to be understood, the other one is to be followed, to be cited. But I like the aspect of writing to influence attitudes, influence practice, somehow influence policy or inform policy. So I came up with 3 things that I am confident that should motivate us to write our science. Otherwise, if we don't, then we'll do the science And the gap between science and the public, the users of science, will continue to exist, or the gap between policy and practice will continue to exist. So I feel we write to create awareness. We're writing to create awareness in its holistic way. I feel we're writing to raise interest. And I feel we're writing to contribute to changing the narrative through influencing those attitudes and practices and also informing policy. And so there are two things that can help us in writing, for us to write for impact, because we write every time. One is the story that we're writing about.
Close to mic.
I do this? Okay, sorry. One is the story that we're writing about. The other one is the way we transport our ideas. When you talk about the story we're writing about, this will be like the message. It's the ideas that we're conveying. It's the content that we're talking about. And for a PhD work, this mainly comes from our research, which means that at one moment we have to be able to know when to stop data collection and start writing. Of course, there are some schedules that say data collection, writing. From my experience, you write continually. If I were a PhD student, by the time I finish lab work, I would be through with writing my materials and methods draft. And then when I'm doing now the results, then I can revise based on what I found, okay? But then also for those who are in science, maybe the story is about just sitting down, listing ideas, brainstorming with groups, rearranging and organizing those ideas to come up with a meaningful structure and meaningful outline that you can generate into a publication. Sometimes it may come from your research, which comes after you've written your proposal, or sometimes you may just be tired and want to review the state of literature. That can build a story. I think the question here is, what is the story? One of the challenges I look at when I was doing my PhD and also when I interact with PhD students is a lot of interesting ideas. You'll agree with me, when you're doing your PhD, that is the time you encounter a lot of new, novel, interesting ideas. And sometimes you are unable to package it into a story. And so you have it all over in those chapters. You have everything is new and everything is interesting. And then now it becomes a little bit difficult to create your story. And so the message today is, so what is the story in what we are writing? Beyond just for, because you see, we're writing either for enrich within the scientific community or also outreach. The many people who do not know the really nitty-gritty of the science. Regardless of what you are doing, the reader should be able to tell the story. The reader should not strain to fish out, to zoom in, or to write and ask at the end of the day, so what is the story? But then the other thing that we're looking at is how do we package the message? Which channels do we use to create impact? So I'm thinking the way we move our ideas can contribute to impact. And I think two things here matter. One is timely dissemination. Timely dissemination will bring impact. And timely dissemination is affected by two things. One, how quick we make our submission. For example, if it's a manuscript from your work, and this one you have control, because you have to decide enough is enough, I want to submit. It depends, one, with the number of authors, how you agree, how the work is flowing. And so you have to reach a place where you submit your work in time. But then there's another way that we delay. The review process normally delays our research process. And so you submit a manuscript to a journal and the journal takes long. I do acknowledge that sometimes it's difficult to get reviewers. Most of the time, reviewers are just declining. And currently they say lack of time, lack of time. Initially I used to see, they say, expertise is outside, is not really aligned, but mostly people are saying lack of time. And so I think if I were writing a paper, one of the things I'm interested with is how quick the journal publishes. And we're not talking about questionable journals that really publish within few days. We're talking about quality journals that take all due procedures to publish. And this is simple. Just look at their submitted, revised, published. It will tell you information. I know some journals may not, they can revise 2, 3, 4, 5 times. But then if they give that information, then you are safe. When you submit a paper to a journal that tells you that it takes 6 months to publish, then what you are sure is that the people you are working with will send out the story earlier than you. Even if it's not the same information, their work will have higher impact compared to yours. I had a story at a time when in 2016, I submitted a paper to a journal, and it stayed for 1.5 years before review. And when it came back, they said they have not found a reviewer. I felt like, wow, this is another one. So I made up my mind that I would select intentionally where to publish, because I have invested time and therefore I don't want to do such gambling. But then the other way of magnifying impact, I would say, is the use of the social media. I have noted that most of the papers that are shared on social media links, they achieve greater circulation. All of a sudden, someone is telling you, I've seen your paper, I've seen your paper, I've seen your paper. It means there is greater circulation, okay? They also achieve more citation. They get cited more than those that we just rely on. It's in the journal website. So they will be cited many times. But also what I've noted is that you can get connections and interactions that can lead to collaboration or clarifications for that work, so that you get immediate feedback that I've seen your publication, it's suggesting this. Is this something that we can discuss, a collaboration? Is this something that you can guide me, clarification? So using also social media can really amplify the impact of our work. I think what we need to be a bit judicious is which content do we share on social media? Because I've seen some journals also do not accept publications that preprints. Are they called preprints? Where you first submit, it's in the public, and then now under review. So some journals would avoid that, but generally many journals are allowing preprints. I'm thinking of a situation where you submit for a preprint and then it's not published, you submit to the other one. So that one you decide. But I think the point here is we have to know that there are many ways of pushing our message across. Some methods are slow, others are very quick. Rapid dissemination of results is very important because people need these results to make decisions. Otherwise, we will continue making decisions from rumors that also get propagated through social media. One of the limitations also of social media is that it can be used to convey information that may not be verified. And so, because I know once you are given a card, your mind will shift. I want to promise you some hints so that I can get your attention. I want to give us some practical hints for ensuring that we write. And this is the reason, because we assume. The reason is that we are all busy. And sometimes people say that they are waiting for time, ample time to be able to write. If we continue waiting that way, we will not write. I've seen executives who are really busy and they are also writing. I've also seen those of us who are not busy like those executives, but we are not also writing, but we are able to say we are busy. And so one of the things that we can do is schedule specific times for writing. And the trick here is not to put writing at the periphery of your time. It's actually to block certain times when you are highly productive and block it, that this is my writing time, okay? Once you have blocked that, the next thing is for you to be consistent. And one of the challenges of consistency is how much you chew. If you chew a big chunk, you can just bite once and choke. Okay, so the trick here that I've learned over the years is to set small, manageable or achievable targets. And this could be in terms of time. I'll write for 2 hours. It could be in terms of time. I'll write 500 words. It could be in terms of subheadings. I'll write the study site subsection of my methods. And so you stick to that and then you become consistent. And the other enemy of consistency is boredom. It is possible that when you are writing, you get bored, especially if that is the only thing you are doing. And so you can spread the activity in a number of ways. One, you can play within the manuscript. When you are bored with this one, you go up. There's a danger with that. Two, you can stagger manuscript. You have one manuscript you are writing on, focusing, and the other one for collecting ideas when you're bored. Or if you are busy and executive, you can call your juniors when you are bored, and then also you get to your manuscript later. You can also switch between projects. The idea is to manage that aspect of boredom so that you don't give up. But then the challenge of moving across manuscripts, and this one I've noted through the people you interact with, is getting disrupted. Going to search for small details. You are writing the methods, and then you see a point for discussion. You move there, you start building a discussion, and now it's difficult. And so you avoid that. But I want to get you to bullet 1, which I find is very nice. Start with the easiest part. I know if you have attended workshops and write shops, you'll be told, start with the methods or start with the results. It works in certain settings. But for me, the trick is start with the easiest part, especially if you are at the beginnings. The only thing I will tell you is don't start with acknowledgements. That one don't. But it's possible to start with references. We can discuss. It's possible to start with references. Popularize the key references into your reference management software. Because if you don't start with the references, then you leave out important key references in your work. So you see, you can start with references, okay? Then you annotate them, and then they build the story. Anyway, so as you write, explore internal review. This one tells you that all drafts are not good, and so you need to work on them, and they only become good by revision. But as you continue to revise, know when to stop, like data collection. Just know when to stop and submit. And this one also depends on the objective, the scope of the work, and the target journal. A tip I would mention is let's avoid redundant publication. You know, a situation where you want to have 10 or 15 publications from the same research work. So you're doing sequencing, and instead of comparing this method with this one, then you are publishing this method alone, and then publishing the other method alone. And then for someone to be able to have a clear picture, they would have to tell you, so-and-so wrote about this and highlighted this weakness. So-and-so wrote about and highlight this weakness. And then you have to deal with all conflicting ideas, ideas that don't agree, or other that are contrary. So let's avoid called Selani publication, either where we duplicate or we self-regulate. Now, the other aspect that I think would be very important for someone who's writing is selecting a journal really in advance. Reading will tell you select one choice journal, but I found that to be really impractical. Have a number of journals. One way, if you are lazy, just try their journal finder. For those publishers, just throw in your abstract. But you see, when you are throwing your abstract in, it means you have written something. So it means you did not select your journal in advance, okay? But then when you select your journal in advance, you get an opportunity to look at the format. What is the format? This one can be really a drawback. One of the things that I don't like is transforming my one-piece abstract into subsections of methods, you know those things. It can really be annoying. But if you select your journal earlier, 2 or 3, and then it gives you that opportunity to read before and know how to format your work. But also as you write, you want to consult, you know, for those who read the Bible, it's like your Bible during writing, you know. the Christians say, read your Bible every day so that you grow. Here you read those instructions every day so that you'll be accepted. You know, it's like a gate. For you to go through, you have to obey certain rules. I would suggest, and this, the fourth bullet, I've found it to be really, especially for those who are starting, to be really important. I've had a student who gave me a budget and told me this 100,000 is for the open access publication. And then I'm asking, where do I get this 100,000? Said, Dr. Changa, you see, like you give this amount. Then I'm like, no. So where the project does not provide funding for open access, I specifically go for subscription. I'm not getting into a lure of cheap pay open access, which now brings us to bullet 2 about questionable journal. But there is also the other trick. There are journals that are giving discounts. Recently, I wrote another paper. I think it was the fastest to write, either within 3 or 2 months. And so we sent to some journal. And then we were looking for the money. We wrote with colleagues at World Agroforestry Center. So when we were still planning to pay, I saw that there is a discount. So I'm not promoting Research for Life, but they gave a discount for that paper. It's in that public space. So there are really publishers or journals or entities that can give discount. Why not look for those discounts as we are scouting for journals? There are many, I have seen many when they are giving issues, they provide those discounts. The other aspect that can help us when we are writing, I think we need to capitalize on our complementary competences. One of the tricks I've noted is where you have people working in different areas. For example, this paper, I wanted some guy who can analyze the work. So if I were the one to do the analysis, I would have run the same things in Excel and maybe 1 week. That guy did it in 30 minutes. He just wrote scripts in some minutes and then did the writing in 30 minutes. So you see, there we have there is synergy. I'm able to do that introduction and the methods. And then where I would have taken longer, he has just run his scripts in Python and then he has analyzed the data. So capitalizing on other competences available with the peers. And when you seek those collaborations, there's also another advantage, opportunities for open access publication, especially if you are collaborating with the universities in Europe. Majority of them have, subscription or they pay? And so that if you're the first author, immediately you select your paper, it brings that opportunity for open access. So I would strongly suggest that collaboration, it helps. It helps to improve the quality of the manuscript because of the different competences we're bringing on board, but it also helps for other added value such as open access. Now I have to start summarizing. And so if you have been writing and you feel that now it's not possible to write, you have submitted your thesis, and there is some hints that can help us continue writing, okay? And one of the things is becoming a peer reviewer. I selected these points deliberately. Becoming a peer reviewer. What I would say is that in your career plan, provide an option for you becoming a peer reviewer, okay? Becoming a peer reviewer has a number of advantages. One, it helps you know the scientific writing processes, and it also helps you know the mistakes people are making. It's easier to criticize other people's work, especially when those mistakes touches you. You see them loud in the other people's work. And so you want to, because you see you're assessing quality and suitability, and you read the journal requirement, you feel like, this is qualified, this doesn't qualify. So you tap on that knowledge. But also as you do peer review, you broaden your knowledge scope and you stay updated, because in most cases you are reviewing work that has just been completed. And as you review, you get to know people in your field. And so if you are looking for collaboration, it's possible to network and to link. The other aspect that I've found that can keep you publishing when you feel like you have submitted your thesis and you don't want to publish, is for you to explore systematic reviews and meta-analysis. I find this interesting in a number of ways. Majority of the approaches we are using to write our literature review is biased because most of it is narrative review. And it depends on the references you are able to access. It depends on the references you know. And so most of our reviews are either a praise of technology, a praise of research, or a serious critique to create a scare and show that my work is important, okay? And so what I've found with the systematic review is that they provide a systematic way that is unbiased for you to coalesce contradicting views and summarize. And so it is really possible to do a semi-systematic review even for your thesis. Even for that paper that you are writing an introduction, you need to consult databases and get out with a number of publications, even if it's within a year, so that you have both sides of the story. Because you see, primary research will always be context specific, and they will always report results that are either contradicting, inconclusive, or there are gaps. And the other beauty of doing systematic reviews is just helping you to get to know what is happening. Other than just broadening your knowledge, you really get to know what is happening. But it also boosts citations. One of the papers that I have a lot of citation is where we have done systematic reviews. They get cited a lot. That's not the motivation, but the learning that I got by interacting, because it's rare to do a systematic alone, but by interacting with other people, it really pays. You get to learn a lot. You learn to manage a lot of data. So I mentioned a number of what I consider as some of the advantages that once we have done our PhDs and we want now to really contribute to this impact, we can explore systematic reviews. As much as we're saying all these technologies are good, one says this better than the other, one says this better than the other, one says this has this limitation. Why can't we do a systematic review that synthesizes results from different analyses and comes out with one conclusion? It is good by how much, you know? Both the direction and the magnitude. Now, the other ones that are friendly, and I found this to be very helpful, just join a journal club. How many of us are in a journal club? Wow, that's fantastic. Yeah, join a journal club, challenge each other. You will find that you're motivated to write. Write-ups are really phenomenal, especially those that requires you to go with a draft manuscript. My first paper in 2011, 12, actually, there were 3. I go pop, 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 pop. I went with the drafts in a write shop, and they happened to be the only one. So people used them. The comments, you can know how they were harsh because they were my first draft. But people gave a lot of comments, submitted, they were accepted. Other options are just attending conferences, seminars like these ones, where we tap on knowledge. I've seen things that I haven't seen here, and I'm like, wow. My students should know about that. My students should know about that. Why are we struggling with this? Yeah, the other one is joining grant writing teams. If we don't have money, somehow our science will fade, and we have to raise money for us to do the science. And research projects are a bigger source of information, especially in scientific communication. And so here I mentioned briefly about grant writing, and this is my last slide. Yes, I confirm that's my last slide. One of the things that I've seen that is a limitation for our contribution to science is really knowing the type of projects that are funded within our area. And I'm going beyond a thesis-like project, which is a bit narrow, to a multidisciplinary project, because most of the funding options immediately after your PhD are likely to take a multidisciplinary angle. The other aspect is that some of the problems require a multidisciplinary approach to solve. And so we are better positioned to know those types of projects that are funded. And so once we have that idea, what do we do? Because writing a proposal or a grant is not something that you just start now and apply in February. So we need to start early. So how do you start early? I think we can develop a list of calls that identifies the focus area and the dates. This helps you if you are out of time to target it for next year. It helps you to do number 2. And number 2 has to do with a database of ideas or concepts. It helps you to start brainstorming. What are the problems in our area? What are the problems that we need to solve? And number 2 helps you in number 3. Who can I reach out to that we can build this proposal? Okay? And so if you have a list of calls, identifying their focus, the start dates, the donor priority, then you are able to look for complementary skills and expertise. But then also sometimes we may not be at a level where we join consortium as participants. It's really also to join through an established scientist, trying to provide whichever role that you can provide at your level. My challenge is to try to talk to the professors we are working with, the researchers we are working with, so that we are part of that proposal writing grant. If you are in a PhD, you write focusing for your postdoc. If you are postdoc, write focusing for a scientist, so that we're part of that to gain the valuable skill from those who already know. But then the last thing is always to align our work with the donor's interest. The donor has the agenda. That is not debatable. However much problem you have, they don't give you money because you have a problem. They give you money because they have the priority area, and your research must fit in that priority area. So thank you so much. It was a pleasure talking to you. I come from JQuat Research. So I put there an email for research. If you want to collaborate with JQuat on research, reach us to us so that we can create awareness, raise interest, and influence some of the things that have not been influenced, and at least inform some of the things that need to be informed. Thank you. God bless you.
Thank you so much, Dr.
I stay for questions, or people want to go eat?
No, you can stay for questions.
Okay.
I think we can take some questions. Thank you for giving us a guide on how to influence and how to write very, very well. What stood out for me was to not start with acknowledgements. Yeah, but this was really a very good guide on how we can write to influence attitudes, to influence policy, and to influence other researchers as well, and how we can align our writing with what the donors needs or what the funders needs. So are there any questions from the floor before we? Yes, 1, 2.
It's not a hard one.
No, they, you can ask from there.
Thank you so much, Dr. Shema. My name is Kennedy. You talked about questionable journals.
Oh, yes.
Yes, I'm just asking, how do you identify, is there a difference between questionable journals and predatory journals? Because there are some journals that have high impact, others have low impact at the same time. Yeah. I don't know how do you identify the predatory journals?
I find using the word predatory is very strong. And so I try to use questionable to include even some of the journals that we cannot categorize as questionable, but I create it to push my own agenda, and then drop it at some time. So for predatory journals, there are lists. Earlier on, there used to be the BALS list. If you are in some institutions, their library provides links to check, the whitelist ones. How do you identify? There are just a lot of list of criteria. One is the review time. One is massive campaign. They tell you, send us an abstract, send us a paper, within 4 working days, we publish. That's questionable. And so it's possible there's a lot of criteria. My approach has been to stay on the mainstream publishing approach. There I will not go wrong. How do you identify their criteria? I could send links to the organizers and then they will share. I don't want to start sharing some criteria and then you say, this one. About high impact, low impact, yeah, there's that narrative. My approach is let's create awareness in whichever journal, because some of the higher journals requires a little bit more experience. Some of them may require you negotiating with the editor if you want to publish certain work. And so I rarely go for high, low. I just prefer, is this journal the right channel to disseminate my information to the audience? Yes. What is the turnover time? For example, for my PhD student, I don't want to submit to a journal that takes 6 months, no, or that takes more. I look at that, and then I stay within certain range.
So on the last one, about the time frame for the publication itself, you like you said about your journal, your manuscript took like 1.5 years.
1.5 years.
So, and at the same time, you're talking about like 6 months. Okay. So where you reach a point now, you feel like you want to withdraw from the earlier journal. Are you able to withdraw it so that you publish it in another journal?
I'll give them a maximum of 3-4 months. A maximum of 3-4 months. Because you see, most journals will send to a reviewer and ask for 14 days, okay? With the intention that if 14 days it's not been accepted, and the reviewer, they recall and send elsewhere. So I work within 14 months. But generally, if you reach out to the editor and ask out, we submitted this manuscript in this time, kindly tell us, whichever language you use, an update. They will tell you your manuscript is under review, so they give you hope. But beyond 6 months, unless the science would have changed, the tools might have changed, right? Someone would have published. For example, the one I've shown you, we were publishing equations that can be used, just simple mathematical formulas that can be used to estimate carbon. I knew another person who was publishing, I knew another person who was publishing, and we were discussing, and we were saying, like, let's see which one comes out first. So if we wait for more than 6 months, yeah, you can reach out and ask to withdraw. It's allowed. I think what is wrong is now having two parallel manuscripts under review. That bothers in ethics.
Last one now, sorry for that. Concerning the databases for references,
yes.
You know, there are some databases that have literature reviews that are not indexed, the papers that are not indexed. So probably you may help us concerning those databases that have papers that are indexed, because at times you find people citing papers that are not indexed, and that raises issues. So you may help us on that as well. Thank you for that.
Yes.
I think I'm Shadrach. I think my question touches on what he's talking about. I need to ask, what's your opinion on preprint culture that is currently ongoing?
It's a good one. I really don't know much about it, but it's a good one. I haven't tried preprint. I just prefer submit, go through the process. It's a good culture, it's encouraged. Just find out whether your journal target allows that. Regarding databases, I think at this moment, we cannot say that there are papers which are not indexed. Even those questionable ones are now being indexed. So indexing will not really be a criteria. Because if you go to Google and someone published something somewhere questionable, they are there. The question about databases is, does your institution have access? That's where collaboration comes in. Collaborate with ILRI, collaborate with other institutions, so that you can access major databases are known. That's where Research for Life, Agora, and others come in, so that you can be able to access. I think most universities in Africa are able to access those through those provisions by Research for Life and others. Citing a paper that is not indexed, to me, citing is like a like. So I just won't cite you anyhow. I have to read your work. I can't read in her work, your work, and then cite your work, because it's like I'm approving. Citing something that you have not read can easily translate to perpetuating error in literature. It's endorsing work. So for me, I have to read, approve of that content, and then cite, because I'm giving you credit. Yeah. So why cite something that is not indexed? Yes.
I'll speak up taking time. Thank you. Fabulous advice. One piece, if I may add, as an addendum to your hints for writing, if you feel that your work shows innovation, has relevance to your peers within the published realm, don't be afraid to reach out to the urgent entities. Those customer testimonials, white papers, webinars, like social media may give you another forum to further publicize your efforts and increase the reach. With that, I have one question. 2023 has been the year of ChatGPT and AI. Where does that sit in writing for the younger scientists?
Well, it's a difficult one. One time I was submitting a paper and then I got on the portal. Was this paper generated through AI? So I said, because I had written, I said, let me say yes and see next step. Out of curiosity, and it didn't go further. I don't know why. But traditionally, as teachers and lecturers, we have been on the receiving end of discouraging the use of that. This is one of the reasons we have sometimes we are unable to do these things on ourselves, having certain weaknesses. And so when you bring another tool that helps us, it may bring a kind of laziness. There's that debate. But then also there is this observation that AI is here with us. It's not going anywhere. In fact, it will advance. And some people are giving an example where we used to go to exam room and do mathematics off head. And then there come a generation that was given a calculator. And we felt like they are doing exam with calculator. We thought they are weak, okay? Because they can't do off head. But then there are people who are managing our money in the banks, okay? They used calculator. And so I really don't have a strong position, but I will encourage people to use. I haven't used it, but I will encourage to try. Actually, not I haven't used it. I tried it with my children and ChatGPT, it gave some information that was a little bit not accurate. And so what I did is now I had to train them. When you tell ChatGPT to do this, get other information to verify. Because I found the lack of a human approach to writing. There was a disconnect of information. So I really don't know how someone can use that for a paper. I saw a paper that was generated through AI, submitted, accepted, recalled. Then I was like, wow, people have gone that far. But I would say, because technology is here with us, people can try to use it, but also, how do I say, ensure that it's serving the purpose. Because however much you refuse, it will be there. And for example, let me tell you from a university point of view. And so there's this lecturer who say, we can't use AI. Don't use ChatGPT. And because he doesn't know what it produces, the student uses it, and the lecturer is like, wow, this student is, wow. He knows how to organize points. This one is factual. But if you have ever used it, you know the limitation. You really know this is not from Paul. This is not Paul's mind. Okay? So I would say I'm limited in that area. Maybe she has an answer.
I really don't have an answer, but just on what you're saying, I was imagining Africa BioGenome that you have a lot of scientists, students, it's cutting across. And I know we are going to be generating data that needs to be published. So I was wondering, within the Africa BioGenome Project, has there been a consideration to build a community of mentors when it comes to writing, so that we have students, young scientists, young researchers being molded so that the data we generate across Africa, we have a pool of people who can write and mentor other people when it comes to writing. Without going the AI, we maintain the innovation and we maintain our community of writers. So we don't have data generated and just sitting. I was just wondering.
Okay, I'll take.
Someone from African Genome to answer
Okay,
that.
I'll just take that and then maybe Prof. Ann, you can add a few things. Yeah, so capacity building is really at the core of what the AfricaBP is about. It's not only capacity building for technologies, for genomics, for bioinformatics, but also for scientific writing. That is why, and also communication, and that is why we have Dr. Shem here. So since we are in the initial stages, we're still trying to build this community. And so questions like that or suggestions like that are really very welcome on how we implement it and really do it in a very sustainable and a way that continues beyond the time that we are in. So thank you. Let's give Dr. Shem a round of applause and then and all the presenters for this session. Thank you very much. It was a very, very informative and highly engaging session. So we have come to, yeah, we were late for lunch, but we're just going to take a 30-minute lunch since we are behind. Today, lunch is being served at the tent. There's a white tent out there. And I want to believe you have all received meal tickets. So you just present your ticket and let's try to be quick so that we're back in here by 20 minutes after 2, which is 30 minutes from here for those who are joining us online. We're going to take a 30-minute break for lunch. And housekeeping, this room is safe. You can leave your stuff here, but if you wanna carry your stuff, you can. Thank you.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makkelneck, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of Zymo kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
Ngaba BioTech has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. Inkaba Biotech has promoted research in the region by providing quality products to researchers. To many more years, Inkaba Biotech, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At Ngaba BioTech, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the
genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good Initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have zero DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of Zymer kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At AfricaBP, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DMB-Seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good Initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have zero DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of Zymer kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At Ingaba Biotech, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good Initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have zero DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of Zymo kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At AfricaBP, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have 0 DNA sequence in them, and we're able to do that quite efficiently.
the major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of zymer kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At AfricaBP, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DMB-Seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have 0 DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for a Greater Good Initiative award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of zymo kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes that happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At AfricaBP, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies, including library preparation and automated sample processing. Expanding into new business, MGI has developed multiple innovative life science tools and technology, including portable single-cell systems, remote robotic ultrasound systems, and BIT products. With a strong focus on the upstream of the industry, MGI operates in more than 80 countries and regions, serving more than 1,300 customers to date, empowering the downstream customers in reproductive health, precision cancer diagnosis and treatment, infectious diseases, food security, agriculture, forestry, fishing, and consumer genomics. Since the establishment, MGI is driven to disrupt the status quo and accelerate the industry innovation. MGI aims to build a suite of life science core tools to address the fundamental questions, push the limits of science and technology, and lead the innovation in the era of life.
So the Greater Good initiative is really going to open us the door of the genetic diversity of camels at the worldwide level, which really we cannot attain with our efforts alone. This will be a great opportunity to hopefully find new variants and variants that can be useful for the community, for livestock traits of economical interest.
We were doing work with sheep and goats. We had a relatively small sample that we genotyped initially, and the grant Illumina provides will allow for additional genotyping, so we almost tripled the number of animals. And awards like this allow us to do genetic and genomic discovery, and the discovery will hopefully lead to ways to feed people better.
Cowpea is a legume, so it produces a very protein-rich grain. So it's very important for food and nutritional security, especially in sub-Saharan Africa. We needed to improve the genetic knowledge of cowpea, and that's the essence of where the Illumina iSelect fit in, which was supported by the Agricultural Greater Good initiative.
We're focusing on crops that are important in African diets. Sometimes you just need something to jumpstart you. For us, it's made a huge difference. We're creating sequence on species that have 0 DNA sequence in them, and we're able to do that quite efficiently.
The major emphasis of our research has been on enhancing the crop productivity in chickpea, pigeon pea, and groundnut. The most satisfying thing for me is when we see that these technologies, they are being used in developing better varieties, which will be helpful to farming communities.
One of the things that should be said about the award is that it's really a big contribution in helping researchers both in plants and animals with reagents and activities that are associated with developing countries.
So my experience working with Illumina has been fantastic. Anyone working in a kind of omics-enabled capacity on a crop that has any relevance to the developing world should absolutely apply for Greater Good Initiative Award.
Welcome to the 20-year anniversary video of AfricaBP, Africa's genomics company, here from our headquarters in Makelnek, Pretoria in South Africa. I'm Dr. Oliver Preissig, a co-founder and the executive director of this unique company with currently around 150 employees in different countries in sub-Saharan Africa. The idea of AfricaBP emerged not far from here in Pretoria in the year 2000 while I was doing research at the University of Pretoria and saw the need for this specific type of company in South Africa. It took 2 years for the venture team of 5 to officially start the business in August 2002, as it was a major struggle to find the funds to do so.
When AfricaBP officially opened its door for business in August of 2002, we primarily offered primer synthesis as well as single DNA sequencing services. In 2007, AfricaBP became the first company in Africa to offer NGS services based on the 454 sequencing platform, the GSFLX. This allowed our customers across the continent to be able to do whole genome sequencing for bacteria and many other microscopic organisms.
Many experiments require high-quality DNA or RNA, especially for downstream analysis. For example, SNP genotyping on the mass array system requires high-quality DNA. At AfricaBP, we use a variety of Zymer kits for DNA or RNA isolation, as they are not only simple and reliable, but effective and efficient, making your experiments hassle-free.
AfricaBP has a footprint all throughout sub-Saharan Africa, and the way a company makes it happen is by having a very good logistical department. We have a dedicated team of staff members that does imports from all around the world and exports into sub-Saharan Africa.
The service department started in 2016. We service, maintain and repair all the instruments that we do supply. We have field service engineers that can go on site and do our services, and we've been growing ever since.
I joined the company 9 years ago, and as we celebrate 20 years of existence, I'm glad that I chose the best path for my career. AfricaBP has promoted research in the region by providing quality products to researchers. To many more years in AfricaBP, your African genomics.
Science is our passion and business, and so we strive for perfection. But it does not end there. At AfricaBP, we also care about our communities. And for some years, we've been supporting the Mukondeni pottery in Limpopo. And also in the same village, we've been supporting a young entrepreneur to advance his business services. And recently, we've acquired this farm in Hammanskraal to develop an agricultural community project. We also extend our community support in sub-Saharan Africa, such as in Kenya, where we support the Gogo Boys Football Club in Kibera slums in Nairobi. We hope our support makes a difference.
Human beings have been using astronomical tools to adventure into the infinite universe, to explore the infinitesimal by making use of life science tools and technology. Founded in 2016, headquartered in Shenzhen, China, MGI is committed to building core tools and technology to lead life science through intelligent innovation. MGI has established 7 international R&D and production centers and is mainly involved in 3 key business segments, including genetic sequencers, laboratory automation, and new business. Continuing heavy investment in R&D, MGI has developed a full range of patented technologies, including DNB-seq sequencing technology, patent array technology, and sequencer optoelectromechanical system technology. Gene sequencing has a very high barrier to entry as it involves multiple fields, including optics, mechanics, electronics, fluid dynamics, molecular biology, chemistry, nanoscience, and other disciplines. MGI is one of the few companies in the world that has the ability to mass-produce clinical high-throughput genetic sequencers. Besides gene sequencing, MGI has also developed a series of core technologies.
Good afternoon and welcome back. We are now ready to start the first afternoon session. My name is Professor Anne Muigai, and I'll be chairing this particular session. We're going to start with a short presentation from Roland, who is from INCABA. Roland, are you online? Dr. Roland, if you're there, you can unmute yourself and share your presentation. And we'd like to kindly request you to make a presentation we're going to cut your time short in 10 minutes, if you can do it. Let's try and see whether it will work. So while we're trying to locate Dr. Roland, is Professor Leibert Tonfak online?
Hi, Prof. Good afternoon. I'm here. This is Roland.
Ah, excellent, Roland. So I don't know whether you had the interesting interlude that I had here with my Congress or workshop participants, that we are cutting everybody's time. So can I give you 10 or 12 minutes to do your presentation for us?
That will be fine with me, Prof.
Are you able to share from your screen?
Oh, sure. Let me try that now.
So as he's sharing his screen, this is short session this afternoon, just to give you a highlight of the practical sessions that Africa BioGenome Project has planned for the next 3 days. So the providers and the technology companies who are going to be taking us through will tell us what they are going to be presenting, and we will be there able to see what is on offer. So please go ahead.
Thank you very much, Prof, and welcome everyone to my presentation. Our practical workshop will be on Sanger DNA sequencing for species identification. And like we know, Sanger sequencing is a method that was developed by Frederick Sanger in 1977, and is built on a technique that incorporation of a modified DNA base onto a DNA fragment that is being synthesized terminates the synthesis. And so with the help of gel electrophoresis, these fragments are being separated. And because the modified DNA are labeled, radio labeled, the fragments are being detected on the gel, and the positions of the fragments kind of tells us the position of those bases on the template DNA. And so originally, Sanger DNA sequencing would involve 4 sequencing reactions, with the main difference being the addition of the modified bases, that is the DD, ATP, CTP, GTP, and TTP. But with technological advancements, the bases are now being modified with fluorescence dyes of different colors, meaning that instead of using 4 different sequencing reactions, a single sequencing reaction is used. And also there's advancements in terms of use of capillary electrophoresis, which has a higher resolving power, making differences of 1 base pair. And so the detection of the fluorescence from this gives the sequence of the template DNA of choice, and this is detected in automated genetic analyzers. So, like we mentioned, the modification of the base is at the 3' position of the base. So we have the dideoxy nucleotide triphosphate instead of the deoxy nucleotide triphosphate. And with this, the DNA polymerase is unable to incorporate new bases onto the growing strand of DNA, so they're leading to the termination. And because this base is labeled, now this time in modern Sanger sequencing fluorescence labeled, the fluorescence is being detected in the capillary electrophoresis, and we can detect the positions of the different bases. So final data, we have the chromatogram and the text version of the DNA sequence. So some of the precautions to look out for would be the template DNA quality, look out for contaminants, and also excessive reagents. And to check for this, the agarose gel electrophoresis will actually help eliminate DNA contaminants or RNA contaminants, while spectrophotometry can help us to check protein contamination. Also, another aspect to take a look at is the template size. That's looking at the quantity of DNA to submit based on the template size. So some of the commonly used genetic markers for species identification using Sanger sequencing are the bacteria 16S ribosomal RNA gene, in fungi, in parasites like 18S ribosomal RNA gene as well, and then for some vertebrates, cytochrome B gene, and there are quite a couple of them. So the intention of our workshop was to have all the participants coming with their samples so that we take them through the process of DNA extraction, PCR amplification, and gel electrophoresis, some preliminary QC to make sure that the downstream sequencing process goes well, and then amplicon preparation and shipment to our facility for sequencing. And also, once we have the sequencing results, we have some post-sequencing workshop for data analysis. Unfortunately, we had just 2 submissions from the participants. So we have Plasmodium parasite samples, and we have some fish parasite samples. So we've synthesized the primers for this, and they will be used by all the participants during the workshop. So some of the members who are making this possible are the Prof. Chuku who is the director of the BTC lab, that's our host institution, and other members of the laboratory. So thank you very much for listening.
Thank you very much, Dr. Roland, and thank you to INCABA for being a host of one of the practical sites. I'm aware that Professor Leibert had prepared a presentation, and I just want to ask whether you would like to make the presentation now. And Roland, if you don't mind, you can stop sharing your screen. Thank you very much. Professor Leibert from the Biotechnology Center, University of Yaoundé. Are you there? You can unmute yourself. Professor Leibert, I can see you online. I'm seeking your guidance on whether we can load your presentation and you make it, or whether the one from Dr. Roland suffices. So no response, but we can come back to you if you're still online with us. We will be able to invite you. And next, we have Professor Mafwila from University of Kinshasa. Let me just say a little bit about Professor Mafwila. He has been able to get a room there in University of Kinshasa where participants like yourselves are gathered, but not only are they gathered, Prof is also doing the translation from English to French. And I just want to thank Professor here for making this particular workshop even more accessible to non-native English speakers. Are you there? I don't see him online. Professor Mafwila? Okay, then I think we can move on to Helen Gusise from Gus. It's a tongue twister. Dr. Helen, are you online? Yes, I can see you online. Please unmute yourself. Yes, thank you very much. Prof, please unmute Professor Nigusi.
Okay, thank you.
Trying. Forgive me, the pronunciations can be difficult even for us.
In Swahili.
I'm sorry, yeah. Oh, niseme kama, I say it in Kiswahili. That one I can. Thank you so much. And the floor is yours.
Thank you.
So I'm assuming that you will share your slides and your screen from your end.
Yeah, I'm trying.
So there's a green share screen button at the bottom of your screen.
Did you see my screen?
Not yet. Yes, now it's there.
Well, okay, thank you. I'm Helen. I'm an assistant professor at Addis Ababa University and national node representative of AfricaBP. And I'm also a co-host of this practical session together with the Bio and Emerging Technology Institute. And I'm really excited to be part of this initiative. Our practical session focus on next generation sequencing, data analysis, and pipeline. As you all know, next generation sequencing have been 3 major sequencing techniques, but our practical mostly focused on the second one, Illumina, which is more than 90% of the current market dominated by Illumina sequencing machine. Why we focus on this NGS, or sometimes we use it high throughput sequencing technology, because of 2 main reasons. Cost of sequencing. As you have seen from the graph, cost of sequencing become decreasing and become flat nowadays compared to the early before 10 and 15 years ago. Again, data generation, cost of sequencing per data and amount of data generation, because before 10 or 15 years back, it took year and months, but nowadays we can generate a whole genome sequence less than 24 hours. So because of this reason, nowadays, large amount of research project, genomic research project and data have been generated. Most of our graduate students also generated same type of data, but they are in the how to analyze and how to interpret this large data sets. That is why we focused in this practical session in our site. As I've told you, this is a different next generation sequencing machine, specifically from Illumina. We have in the NovaSeq era, have generated large, probably more 400 gigabytes per day, which is a large amount of data. When you see application of NGS nowadays, yesterday in the morning, there are different research have been presented, and this NGS can be everywhere, can use it for DNA sequence analysis, RNA sequencing, and even for transcriptomics, which has in different living organisms based on the research objective of the scientists. When we see in agriculture, NGS can be applied in different scope, both in livestock and crop, in different sub-specialization, different cereals, crop and animal livestock species. So it has a wide application, including genome editing recently. NGS could be applied in biodiversity conservation. We have seen some data in this morning. So it can, the scope is very high. It can apply in everywhere, in every discipline. As already said, it could also applicable in forensic science, clinical application. So that is why I focused. And as a 3 days workshop will focus on this workflow. We'll focus on the quality control of the reads, trimming, and most probably focused on mapping of or genome assembly using reference genome. We're going to use the small data size that is Arabidopsis, and then we'll visualize those mapping. We'll try to give them detailed practical and hands-on training for our participants, both in person and virtually. The pipeline will focus on this, how to manage the quality issue, how to map, how to assembly using different mapping tools. We'll also introduce them some command line, because this big data will not be used as normal Windows system, computer system, and we'll use high performing computer, which is in Bio and the Emerging Institute technology. So the participant need to know some command line, especially the Linux Unix programming. We'll introduce them, some basic command line first, and then we'll move to the assembly and quality analysis of the short reads data, since this Illumina sequencing generated short read data, and genome assembly a little bit difficult having this very small fragment. So that is why we still focus on this. The organizer of the Ethiopian team will be me and Dr. Kassaun, another who was presented yesterday, the Director General of Bio and Emerging Technology, and there is also Dr. Zodu from BETIM. He's a director acting as a director of Bioinformatics Directorate. The trainer will be both from Addis Ababa University and Betin. My colleagues, Dr. Ade also presented in this morning, and my colleagues from Betin, Hermias Asafa and Jonas Gebrenike, will assist in this practical session during the 3 days. This is a practical session. The venue in the Betin, we have HPC facility, our computing facility. There is online well-organized practical session room for in-person participants, and we'll also broadcasting via online for our online participants because there is a limited space and resources to host all the participants. We received more than 120 applicants. Unfortunately, we can manage it only 30 of them. This is the facility we're going to use in the next 3 days. I would like to acknowledge African BioGenome Project to give us such opportunity to contribute the betterment of our graduate student and researcher in the country. My institute, Addis Ababa University, to help me to host this workshop, and Bioemerging Technology Institute to host as a co-host and facilitate all things to make this practical session on board. Thank you. This is what I have, a very short introduction about our practical session. Thank you, Asante Sana, and Amasadinari.
Thank you so much, Dr. Helen, for taking us through what will be part of your presentation. 120 applications from which to shortlist 10 to 15 applicants. That's a tall order. And that just shows you the demand that we have for training.
So, sorry, Professor Apollonia, Professor Anne, sorry. I wanted to ask Ellen and also Anne, with regards to those shortlisted, I mean, what's the spread like? Are they all in Ethiopia or also some other neighboring countries? Because in North Africa, we got, lots of, in terms of, applications and people from different things. So Helen, do you have that, the demography of the top of your head or something, or?
Yeah, actually, we only received the applicant for only practical site in Ethiopia.
Okay, yeah.
There are 120 applicants. From there, we shortlisted 30 of them.
Okay.
22 something for in-person based on our facility we have, and the rest of them will follow virtually.
Yeah, absolutely. So do we have a link for them to, I mean, do you send them the, like, okay, that's fine.
I have sent them and everything is ready. We will start it tomorrow morning.
Yeah, because we've been receiving several emails and chats on the social media asking us, can people still register? So I've been passing those conversations to you and Terefe, and you guys will take it up because we want, one of the things we are doing with this practical workshop is that we want the host institutions to, take more responsibility, and, given that we help to, launch this call. So I think with the any future questions we have, we'll now direct it to you, and then you can interact with the applicants, and maybe you can try to connect them to the live streaming on other platforms within your institution.
Okay, yeah. Even me, the follow-up of the post last week in the LinkedIn, I also received lots of emails from Paul, but this showed us really there is a strong demand, so we should think about the future. We should organize a type of workshop in the near future together with another organization.
Yeah, so
from where this participant is based. Now I'm thinking in that direction.
Yeah, absolutely. So I agree with you on the future. So because this became clear to AfricaBP after the workshop in Pretoria. So we have I have spoken to majority of the partners and the sponsors, and they are happy for us to do this on an annual basis. At least the ones I spoke to. And we hope we'll return to Nairobi, we'll return to East Africa and Central Africa again. So Professor Anne Mugai, I think I will leave this to you to, if you want to help make the commitment as well. But I think for the continent, this workshop is very helpful, and hopefully we'll return to East and Central Africa next year, and hopefully we can do this every year. And the format will be the same, it will be a distributed format whereby across each country, each institution that want to host, they can then host people, a few people here and there. But going forward, what would happen from next year is that we would have, it will be more of a conference style. So people would have to submit rather than have a serialized day 1 and day 2 where we invite speakers. We'll only just invite keynote speakers and invited speakers, maximum around 8-10. Then we'll allow people to compete via submission of abstract for oral presentations and posters. So that will kind of focus around the day 1. And of course, we'll kind of devolve the way we've done the practicals from day 3 to 5, hopefully, because we've also been receiving several contacts from several institutions across Africa saying that they want to be part of this. So I think for us, this is just a test run. And yeah, we also need to think about how we manage, like 10 institutions or 12, because at the moment we have 6 institutions across 6 countries or something. We have in Ethiopia, we have in Congo DRC, we have in Kenya, we have in Uganda. So yeah, 6 institutions across 4 African countries. Okay, thank you, Dr. Helen.
Thank you. Thank you so much.
Thank you, Anne. Yeah.
Thank you very much. And a big thank you again to all the institutions that are hosting practical sessions for us and technology companies that are providing the support for the full successful launch of those practical sessions. We do not take it lightly. The demand has been tremendous, and it just shows us that we have to do more to make these workshops and information more accessible to wherever we are. Now we will listen to Professor Mafwila, who is at the University of Kinshasa. So if you can unmute University of Kinshasa. Yes, you can be able to share your presentation, and feel free to go ahead. Thank you.
Yes, thank you. Can you hear me?
Yeah,
yes, we can hear you.
Okay, thank you. So, thank you for the organizer of this conversation. So, here in the University of Kinshasa, we commit to organize.
Sorry, Antoine, could you please maybe come closer to either the laptop or the phone, because we can hardly hear you on the call here, please. Maybe if you come closer to it and speak into the mouthpiece of either the laptop or the phone, that'll be better. Thank you.
Okay, can you hear me now?
Yes, this is better.
Okay,
Yeah.
okay, thank you. So, just saying, as we are in a French speaker country, so even for this presentation, please, I will do that in French. Donc, les objectifs de notre formation, le biologiste capable de décrire le Linux en tant que système d'exploitation. Également, il doit être capable d'installer le système Linux dans Windows. Linux. Et une fois que nous avons appris avec le biologiste comment le faire, on devra ensuite appliquer les lignes de commande de Linux pour faire l'analyse des données issues du séquençage génomique de la nouvelle génération. Donc, on va pouvoir, avec le biologiste, voir comment on acquiert les données provenant du séquençage génomique de la nouvelle génération, comment évaluer la qualité de ces données Et une fois que la qualité des données est évaluée, comment procéder à l'amélioration de la qualité de ces données. Et ensuite, une fois qu'on a fait procéder en utilisant l'INIX à l'assemblage du génome. Et une fois qu'on a assemblé, évaluer la qualité de l'assemblage et procéder à la notation. OK, donc on va discuter sur utiliser l'Linux comme système d'exploitation, étant donné que c'est un système qui permet l'utilisation de programmes qui ne disposent pas d'interface utilisateur graphique au niveau de Windows, mais l'analyse des données issues du séquençage. Alors, Linux, comme vous le savez, c'est un clone de Unix à partir de zéro qui a été construit pour servir de système d'exploitation pour la plupart des serveurs à travers le monde utilisent Linux ou un de ses variants. Et la plupart des téléphones Android fonctionnent avec le kernel de Linux. Donc, ça offre l'avantage d'être un système d'exploitation open source que les gens peuvent améliorer. Donc, c'est ouvert à l'amélioration, ce qui offre la possibilité d'un développement exceptionnel et même une rapidité dans son développement. Donc, c'est un système qui est aujourd'hui, on a rendu possible son installation dans Windows. À partir de Windows 10, on peut installer le système Linux dans Windows. Et nous allons utiliser, bien sûr, ces manuels d'installation pour pouvoir installer Linux dans pratique. Et donc, on va discuter sur la structure de répertoire dans Linux, où vous avez une structure qui est caractérisée par un ensemble de chemins menant à plusieurs niveaux de directoires placés à des niveaux hiérarchiques, où le biologiste a la possibilité de stocker des éléments qui sont issus de son analyse. Alors, l'interface ligne de commande Linux, ça c'est un élément important qui va aider parce qu'en l'absence d'interface graphique pour utiliser le programme, Linux a prévu un terminal qui permet à l'utilisateur de parler de communiquer avec l'ordinateur. Et c'est par cet terminal que le biologiste va introduire un certain nombre de commandes qui vont lui permettre d'interagir avec la machine. Donc, voilà, ça c'est un modèle simplifié d'une ligne de commande de Linux où vous avez une commande qui est suivie des arguments. Donc, c'est l'exécution de ces commandes qui va permettre à ce que l'ordinateur puisse analyser, puisse traiter les différentes entrées.
Excuse me, Dr. Antoine, your
Yes.
slides are not moving. We are still seeing only your title slide.
This is...
People online can only see your title slides. You're not in presentation mode.
Yeah, so Dr. Antoine, I think what JB is trying to say is that you need to move your slide from slide 1 to slide 2 to slide 3 to correspond with what you're saying, because it's still in it's not in slide mode, in presentation mode. Could you please?
Okay, let me correct that.
Mm-hmm. So if you go to the top and just go to slideshow.
Okay, slideshow.
Yeah, so go to the top. Yeah, go up, 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 up.
Yeah, yeah. Is the same?
Yeah, so you go up to the left. Go to angle 90 of your screen. Angle 90 of your screen, please. Just at the very top, angle 90. Yeah, okay, here. Yeah, there. Yes, yes.
Is that good now?
It should work, but it doesn't look like it's working.
So if you just press F5, Antoine.
No, let me continue.
Just press F5 to go to diaporama mode.
F5, F5.
Yeah. Ou alors tout en bas.
Is that working?
So another option is, if it's going to take time, what you can do is each slide you want to talk about, you just press the slide number so that you may not be on slide mode, but let's say what you're talking about on the left.
So let me go like this.
Yeah.
Like this.
Yes, exactly. Please carry on. Yeah.
Okay, thank you. So ça, ce sera le lien par lequel nous allons procéder à l'installation de Linux dans Windows. Donc, comme vous voyez ici, Linux va se présenter dans une structure particulière de directoires qui sont placés en ordre hiérarchique, qui permettent aux et de les stocker. Donc, j'ai parlé de la ligne de commande de Linux. Donc, nous allons faire des exercices pratiques sur comment écrire des lignes de commande de Linux pour pouvoir avoir une réponse de l'ordinateur, pour pouvoir exécuter un certain nombre de commandes. Et ça, nous verrons à partir de demain. Ça, c'est l'exemple d'une ligne de commande. L'option et les arguments. Et une fois que vous mettez cette ligne de commande dans l'interface, vous aurez une réponse de la machine. Donc, notre exercice va consister à analyser les données avec Linux que je viens de présenter. Et ces données vont provenir du séquençage de la nouvelle génération, bien sûr, des données provenant du séquençage de la nouvelle génération. Et comme vous savez, ces données se présentent en fichier FASTQ 8. Et nous allons procéder à l'évaluation de la qualité de ces données en utilisant un programme. Plusieurs programmes sont disponibles pour l'évaluation de la qualité. Il y a FastQC, Prinsec, NGS Toolkit et les autres. Donc, nous allons utiliser FastQC pour pouvoir évaluer la qualité des données ici du séquençage. Une fois que nous avons évalué la qualité des données, nous allons corriger les problèmes de qualité en utilisant issu pour améliorer un programme de timing pour améliorer la qualité des données issues du séquençage. Une fois que nous avons amélioré la qualité, nous obtenons des fichiers FastQ, donc de haute qualité. Nous allons procéder à l'assemblage du génome en utilisant un programme. Plusieurs programmes sont disponibles. Il y a SPAD, Velvet, CLC, SOAP et les autres. Nous allons utiliser SPAD. SPAD pour nous aider à faire l'assemblage des données à partir des reads. Alors, une fois que nous avons fait l'assemblage, nous allons obtenir des contigs, des scaffolds ou des génomes circulaires. Et nous allons évaluer à la fin cet assemblage en utilisant, bien sûr, le programme QUAST pour évaluer la qualité de l'assemblage. Et une fois que nous avons du génome en utilisant le programme POCA. Donc, à partir de demain, nous allons commencer avec des exercices sur les lignes de commande Linux et le jour suivant, nous allons utiliser les commandes Linux pour procéder à notre exercice d'assemblage de données issues du séquençage de la nouvelle génération pour aboutir à un génome complet et à un génome annoté. Voilà, merci beaucoup pour votre attention. Toutes mes excuses pour les désagréments techniques que vous avez connus. Merci beaucoup.
Thank you very much, Professor Mafula. I think we had a sincere appreciation for the French language, and we are directly, we appreciate the effort that you have made actually to ensure that there is direct translation for your audience that you have there with you, and we thank you very much. So right now, I'd like to introduce Dr. Jean Bakker from ILRI to come and tell us and give us an outline of the practical workshop that he will run here at ILRI.
Thank you. Thank you. Okay, so it's too low for me, this mic. I hope you can all hear me well. So good afternoon, everyone. I could continue in French if you want. All right, so, okay. Ah, sorry, we need to share, okay. So I'm going to present quickly the training workshop I will deliver tomorrow and day after tomorrow. It's just a 2-day workshop. On the bioinformatics of Oxford Nanopore. So it will be mostly about the bioinformatics workflows for genome assembly using data from Oxford Nanopore technologies. But we'll start with an in-depth presentation of the Nanopore technology as implemented by Oxford Nanopore Technology, the company. And then we'll focus on the bioinformatics analysis of sequencing data for genome assembly. So we'll have no wet lab activities. It will be in DAMA here, and also a few slots are available online for attendance online. I will first present quickly the Nanopore sequencing technology, and then give you the outline of the 2-day workshop. So briefly, and I do this because I know you've been having presentations on Illumina technology, and PacBio is also here. Unfortunately, we couldn't get ONT to avail reagents, and for other technical reasons, we have no wet lab hands-on this year on Oxford Nanopore technology. So I just wanted to present quickly the technology. So at the core of the technology is those macromolecules that are called pores that are inserted in a membrane. And these pores are engineered so that they are very sensitive in a way that can be interpreted. They are sensitive to the translocation of analytes through the pore. Those pores are mounted on a membrane, and therefore, when you have analytes that are attracted by an electric current to move into the membrane, there is an electronic sensor that records the changes in the conductance of the pore. Okay, so in steady state, without any analyte that goes through the membrane, you have ions that are in the solution into which this system is, or the solution that is above the system, above the membrane. So these ions are naturally translocating through the membrane because they are attracted by the difference of potential that exists between the cis side and the trans side of the membrane. And therefore you have a current. But when a certain base goes through the nanopore, this current is disturbed and is modified. Okay, so you have to imagine that a single-stranded piece of DNA goes through this pore, and as it goes through, you have those squiggles that are the measurements of the electric current, the conductance, the electric current that goes through the pore. This is measured and then is deciphered so that we understand what are the bases that were on this single strand of DNA. And that is called base calling, this last part of transforming these graphs of picoamperes, very small amounts of currents. This graph of picoampere over time, transform this into base pairs that have translocated because these base pairs are part of the single strand. So unlike other technologies, this technology is not based on synthesis of a complementary strand. It's the direct analysis of a strand of DNA that translocates through a pore. Key advantages of the technology, we have just 2 slides about this. Nanopore sequencing is very flexible and versatile. The run can be interrupted at any time. It's real time sequencing. So as soon as you start the sequencing, in real time, the translocation of, or the current resulting from the translocation of single strands of DNA is analyzed and is deciphered into base calls, so you have the information. So as soon as you think that you have harvested enough gigabase pairs of data, then you can just stop the system, flush the flow cell, wash it, and store it in a fridge for future reuse. MinION, the small device that is the most common sequencer using this technology, is powered from a USB port, so it's very lightweight and it doesn't require a strong source of current. The temperature requirements are relatively loose, room temperature basically. You don't have to have your sequencer placed on a slab of concrete, extremely stable. You don't have to have very good air conditioning and humidity control, etc. There is no optics, no microfluidics inside the sequencer. So the device is quite basic and sturdy. So it doesn't require a maintenance contract. If you don't use the sequencer, you don't pay for anything because you don't have maintenance contract. And in Kenya, we have some good supply chain logistics. Be aware, there is one issue: flow cells must not freeze. So sometimes in the logistics chain of shipping to you, if the people are not very aware and if they think, oh, it's like any other biological material we are asked to supply, we just freeze it as low as possible and it's the best. No, that's not true for Oxford Nanopore flow cells. That they must not freeze, because freezing destroys, shears actually the membrane. High flexibility, as I said, you read the native strands, you do not do anything, no on flow cell amplification, no synthesis, you just read what is there. So you read the native strands, you read long reads because there is no limit to the length of a read that can translocate through a pore. Reads of several mega base pairs have been achieved. Methylation detection is also possible. You have live results, as I said. real time. You stop, you wash, you store the flow cells, and you can even do adaptive sequencing based on your live analysis of what is sequenced. You can decide to reject a strand and to push it back out of the pore if it's not a strand of interest to you, because you have been able to base call it in real time. And also you can monitor your system, and let's say you're in a diagnosis application, you want to see if a virus is present. As soon as you have sequenced that virus, you have the information if you're able to do real-time analysis, and you can stop the run. Okay, this was just a very, very brief overview of the technology. Now into the workshop agenda for those 2 days. So tomorrow morning, we'll have a more in-depth technology overview, and we'll do a bit of new Linux literacy. So in-depth presentation of the technology, use the Linux command line, and also we're going to use ILRI's high-performance cluster. We have a big server here, and that's what we're going to use tomorrow and the day after. So you have to be able to understand what a job scheduler is and to issue simple commands to schedule jobs. Wednesday afternoon, we'll talk about file formats and we'll do base calling. So you have different file formats, intermediate file formats that store those graphs of amplitude of a current over time, conductance over time, and this is typically FAST5 and POD5 format. And then after that, after base calling from these, you get FASTQ file format, and you're probably familiar with FASTQ file format if you've already been doing some NGS. And we'll see what are the files and the reports that come from the sequencing software. We're going to do base calling offline using a GPU, and we'll try different base callers. The day after that, on Thursday morning, we'll look at quality check reports, and we'll start looking at assembly software. So how to check the accuracy of base calling through text or graphical summaries, and then we'll embark on using 2 workflows of genome assembly, Fly and Medaka, or Minimap2 and Mini-ASM. So that's about assembling a full genome, typically what you would do in AfricaBP when it comes to assembling from scratch new genomes. And finally, Thursday afternoon, we'll do assembly quality check, and we'll look at online workflows. So we're going to understand what's the output of genome assembly software. We're going to navigate the results of this assembly software using Quast to verify some key parameters of our assembly, and Bandage to visualize the assembly. And then we'll also quickly have a look at ONT pipelines that are published online and that you can run online without installing anything on your computer, such as Epi2Me, those pipelines being mostly used for metagenomics. And that's all for me today.
Thank you very much, Jean Bakker, for that oversight. And I have seen that Jean Bakker has written to all he had shortlisted for the practical workshop, asking for confirmation for physical attendance in order for us to avail any available slots to those of you who are here or who will be able to attend physically. Thank you very much. The next presentation is from Dr. Rushba Shah of Illumina. I believe that, yes.
FNS Illumina, same thing. No, that's fine. Can I do it in PDF format? Would that be easier? Can everyone see the screen? Maybe not. Do it in PowerPoint format if it's easier. All right, thank you very much. Good afternoon, everyone. So I do understand we're a little pressed for time, so I'll do my best to save you time, and I hope that benefits everyone else as well. So just a quick introduction. My name is Rishabh Shah. I'm here on behalf of Illumina as part of F&S Scientific, their partner here in Kenya. And this is just a very small, short presentation to sort of highlight our relationship with Illumina, but also more to enlighten you about how we support genomics as a whole, and hence why we call it the end-to-end solutions. Quick introduction. I think some of you already know who we are, especially those of you from ILRI, and possibly I see some of our customers here, which is fantastic. So F&S Scientific, we've been in the game for over 40 years. Sounds old, but I still consider ourselves young and always looking for new opportunities to tackle unmet needs. And here we are doing it together with Illumina as we speak. As a company, we offer a whole range of technology solutions, laboratory equipment, field-based testing equipment that serves all the different sectors you can think of. We're here talking about research, but we also serve the various industries. We serve food security, healthcare, environment, energy, water, and more. I'm sure you're familiar with many of these names. So putting these brands together allow us to play a part in this ecosystem about some of the solutions that you need in order for you to carry out this research. I think what's impressive about this consortium, the Africa BioGenome Project, is they're now looking to see how genomics can be used to address biodiversity in Africa. It's the same technology solutions you need, but it's a question of how you utilize them to help you in your endeavors. And we, as a technology partner, simply come in to provide you what the world has to offer. In terms of impact, so we really focus on how we can make an impact. Yes, of course, we're here for business, but we want to do good. So this is just some photos, some snapshots of some of the work we've done. For example, here we worked in Mathare slums to provide diagnostic solutions for sickle cell disease. We do a lot of work in water quality and safety. We were here a few months ago with Illumina to talk about wastewater surveillance. But remember, aside from the biology side of things, there's also chemistry when you think of water as well. And we do have holistic solutions for that. Air is becoming more and more of a concern. Air quality, again, solutions for that. And of course, food security. So I'm sure you've all heard of the menace, aflatoxin. We support the value chain, right, from farm to fork, as they call it. And lo and behold, here we are with our partners Illumina to unlock the genome. So we're pleased to be here together with them to be supporting this initiative. These are just some photos from recent events that we have also participated and conducted with universities, with research institutions. And I think the important message here is that in order for you all to unlock the genome in your different domains, we are here to provide whatever support we can on behalf of our partners. So we are the first line support. They come in with the expertise to ensure that we're able to deliver good solutions and good value to our customers. So in terms of Illumina, I think they've already presented to you, so I'm not really going to say much more. I think you've heard from my colleague Peter earlier a bit about the solutions and also what they're going to be doing in terms of the workshops. I think from Xavier more about what Illumina offers. But in one sort of summary slide, this is just an example of all the different applications where you can effectively utilize, leverage Illumina solutions for genomics. And currently speaking in Kenya, this is just from our sort of engagements at the moment. I've just highlighted some of the areas where there's already ongoing initiatives where we are actively working, supporting in discussions. So that goes down to cancer research, microbiology, agri-genomics. On the clinical side, oncology. And in terms of what they call the unique areas, there's precision health and neurogenomics. I believe we need to probably add another one here for biodiversity, or I'm not so sure where it fits in, but I'll let the experts advise on that. So in terms of a company, genomics is not new. I'm sure, as you will tell me, genomics has been there, but technology is ever evolving. And as far as the African continent is concerned, and at this point, I'll say, let's leave out South Africa, let's leave out North Africa. Genomics is still in its infant stages here in Kenya, in East Africa, in Central Africa, and West Africa. So for us to be able to provide good service to you, we also recognize and appreciate that, you know, when we talk of sequencing, that's the very last step in this, what we call this workflow. And, you know, as the saying goes, garbage in, garbage out, right? So we have to make sure that we're able to provide you that end-to-end support, right from the time you get that sample all the way through to being able to perform your analysis. There's many different ways to do it. You can walk, you can cycle, you can drive, you can even fly. What is essential is knowing where you want to go, and we're here to support you. With regards to how we operate, just to emphasize, there's a whole team of people behind this. I'm just one person here, but you've already heard from Peter. You've heard from our team at Illumina. We have a whole team of people here at FNS. So we have people in marketing, in application and technical support, which is extremely important. These machines need a lot of support. The scientists need help when it comes to troubleshooting. Also very important, as you will appreciate, these reagents are very sensitive and delicate, hence our logistics and operations play a key role. On Illumina's side, while we are the interface, we do depend on Illumina for their specialism, their team of experts. They also have a next level of support and their customer service. So with that, I'd just like to say, I hope I kept it short. Thank you very much. I do hope you guys make the most of the next few days, and looking forward to see where this takes us. So asante sana.
Thank you very much, Rushab. You must forgive me. I associate you with Illumina so much, and F&S is obviously key and part and parcel of that ecosystem. And now I'd like to invite Oliver Pressig from Inkaba, traveled all the way from South Africa, to come and give us a highlight of the practical workshop that he will be holding from tomorrow.
Do I have to do anything here? Yeah.
I think he'll share it.
Okay. Yes. Good afternoon, everyone. Thank you, Anne.
Yes.
Well, the workshop part is very short at the end. It's so small. Where is Paul? Okay, well, maybe just to introduce the company Ingaba Biotech. So it's Ingaba. The aim was, when we chose the name, to have a click loud. It's a Xhosa name, Xhosa word. So that it's a little bit more difficult to pronounce the company name. In the meantime, we are represented in quite wide in sub-Saharan Africa. We have offices, the headquarters is in South Africa, and we have subsidiaries and branch offices in Tanzania, Kenya, Uganda, Cameroon, Nigeria, Ghana, and Senegal. A few of our employees have given talks, like Acclaim, Roland, and Dennis. And well, while we're still waiting for the presentation, which obviously needs to have some pictures, but So we are 21 years old, and the idea was to 21 years ago to start the company to produce oligonucleotides and DNA sequencing services. Because I was a researcher in South Africa, in Pretoria at that time, and I saw we were waiting very long for getting primers. And, you know, if you do Sanger sequencing, especially if you walk on a bigger fragment of DNA and you had to wait like 3-4 weeks to get primers, a work that normally would have taken during my PhD, you know, a week, took in South Africa a month or 2 months. And so that was the idea. And obviously, if you have an idea, you need to get money. And so we got some shareholders. And the Swiss government also gave a loan, which was very nice of them. We also paid it back in the meantime. So we developed Ingaba Biotech. But when we started producing oligonucleotides and offering Sanger sequencing services, we also realized that the market is most probably at that time too small. So we started offering products from other suppliers, from manufacturers. So we started a distribution side of Ingaba Biotech. And the first one was the Fermentas, that was later bought by Thermo. The second one was Nanotrop, that was later also bought by Thermo. And we're still selling the Nanotrops, but Fermentas is now sold by Thermo Scientific. Then came, okay, I see it's copying. It's a very big file. In 2007, so 5 years into our, okay, I think there's now something happening. Okay, let's start and I'm making it also a bit shorter because Anne wants to finish sooner. And so actually I wanted to talk about the, you know, as a company, how we developed to actually sequence whole genomes. As I said, we started and maybe here just a quick picture of our facility in Pretoria. That's the one property. On the one on the right belongs to Ingaba Biotech as well, and then there's another one across the road on the left side. And as I mentioned, we have subsidiaries, and our regional headquarters are in Kenya and in Nigeria. So we have here recently in Westlands bought a property in Riverside. That's the one. And just now they finished a lab in it. So we will develop lab services here in Kenya as well. In Nigeria, that's the building in Nigeria. I was there last week. It's actually opposite the IITA bus stop in Ibadan. So not in Lagos, in Ibadan. Here we do already Sanger sequencing services for like 2 years. And the next thing what we want to do is offering NGS services. And I'm showing here properties, etc. And obviously there are teams, and there's a picture of our Nigerian team as well. Okay, sorry, that's the wrong side. So I already mentioned a few milestones, but I waited actually for a very important one. It was in 2007, where 5 years in our business, we got a next generation sequencer. Actually, the first one in Africa. It was a GS20 from 454 and Roche. And I will get back to that soon, because the other aim was obviously at that time we were just based in South Africa, and we always wanted to see Sub-Saharan Africa as our market. And we opened in 2010 our first subsidiary here in Kenya, in Ingaba Biotech East Africa. And I actually came here in 2009 and had actually, I think we had a presentation in this room as well. Then 5 years later came Nigeria, and then you see Tanzania, Ghana. 2020, we got our first PacBio sequel 2E. And while we still expanded to Cameroon and Uganda, and this year in Senegal, we are finalizing our registration there. And also this year, we started the Smart Farm by Ingaba Biotech in partnership with the World Wildlife Fund for Nature in South Africa. So just, I mean, we have not much time, but that's our farming project. It's a corporate social investment. It will be a community farming project, but it has also 2 pivots, each of 20 hectares. And we actually now seeing how we can farm. This is a commercial way, and this is our Sonele, our farm manager, and that's our first crop. It's maize, sweet corn. And yeah, so the aim here is obviously, it's, as I say, it's a community project, and we want to do sustainable farming. And then in some way, obviously, it will be also very interesting to see how our tools that we have, our genomics tools, how we can help a farm to be producing better. And obviously, first thing would be soil analysis, metagenomics, etc. But there are also things that I have in mind, and they come up. So, as I said, we are a team, obviously, and you have met a few. We are over 150 people now in sub-Saharan Africa, and that's a few pictures of our lab in South Africa. In the last 21 years, we have built up quite a few technologies in our laboratory. The result was that the shareholders didn't get a dividend for 19 years. So they had to wait. Oh, but obviously they were part of the decision. So we have oligosynthesizers, Sanger sequencers. As I say, one is now in Nigeria. We have a NextSeq 500 from Illumina. We have a mass array, and just recently we got a Thermos Titan mass array, microarray system, which was missing in our portfolio, microarray services. I mentioned the Sequel 2E from PacBio, and yeah, the aim is to have more service facilities in sub-Saharan Africa. As I say, Nigeria started already. Kenya, after this meeting, we will go and inaugurate it. And obviously in the other offices that we have with time, that will also come. Obviously it comes with a cost. It's not yeah, it needs funding. I mentioned distribution ship. Here are our big 5 brands, starting with Seegene, which most probably nobody of you know, because it's a molecular diagnostic brand, New England Biolabs, Simo Research, Eppendorf, and yes, PacBio. And there was a very interesting question in the morning, why suppliers don't have their own service facilities, you know, that we have changed. And we'll get to that. Now, back to a bit of history about, mentioned in 2007, we got the first GS20 system here in South Africa, in Africa actually. And one of the first project was to sequence a plant pathogenic bacterium called Pantheia ananatis. And that was done with a group at the University of Pretoria. And Peter de Meyer was the student, and you see so that was the first genome that we sequenced. And as you see, that's a part of the publication they published, and they mentioned Ingaba Biotech, which was very nice. They also said how many reads we got, like 900, nearly 1 million reads of 80 to 120 nucleotides in length. I think at that, I mean, today we don't appreciate that anymore, but that was a breakthrough in technology. That was really next generation sequencing. And yeah, so the genome was 4.7 megabases big. If you look at when it was published, it was 2010, so it was sequenced in 2027, so meaning it took a bit of time actually to get it to a publication. So sequencing at that time, the analysis was very, very time consuming and new. Peter once a few years later told me, you know, Oliver, one thing that the sequence revealed to us, you know, they bought primers from us at that time, and it seems at a certain stage some primers didn't work. And, you know, I guess they troubleshooted and they still didn't work. And normally then they say, well, they go and buy it somewhere else and they still didn't work. So what he found is actually those genes that he wanted to amplify are not present in that genome. So that was a little story. But I also want us to say that that wasn't the first genome that was sequenced in Africa. This one is the one. It's Rickettsia ehrlichia species. It was also sequenced by the University of Pretoria by the Professor Alsop. And there's also a name Etienne de Villiers, who is very familiar here at ILRI. He was working here. Why I show that is, if you look at down here in the things, also they did Sanger sequencing. So they used a lot of money and a lot of time to sequence inserts of plasmids on ABI 377. That's a very old system. And the ABI 3100 wasn't, well, at that time a bit newer. So, I mean, we have gone far with genome sequencing because in 2020, as I mentioned, we got the Sequel IIe system from PacBio. Again, we are the distributor of them as well. And in the meantime, we are also a certified service provider. So what has changed? Now we have high fidelity long reads of around 20 kb, which makes the bioinformatics analysis much easier. And so in the meantime, we have had over 100 runs with up to 4 smart cells. So we have sequenced several human genomes, animal genomes, plant genomes, some for the African BioGenome Project as well. But there is a limitation with the Sequel IIe is that, you know, first it costs, and secondly, the throughput is also not that, you know, I mean, if you have hundreds or thousands of genomes to run, wasn't really possible with one Sequel IIe. And so what brings us to 2024? So we will get the PacBio Revio system. It will be shipped end of the year. And what does that mean for the customers is, first, it has 15 times more throughput, meaning one could potentially sequence 1,300 human genomes in a year, which is or equivalent, if you know, in a genome size. And most probably much more interesting is that actually the price comes down by 3/4. So genome sequencing on a PacBio system will be much more affordable. And I think that will change also for the African BioGenome Project, things that we can do. And well, I was supposed to talk about a workshop. So we also do workshops quite a lot. On the left side are workshops were done in Nigeria. On the right side was the AfricaBP workshop that we did in our lab in Pretoria. Now we will start, or Acclaim there in the back is actually the one that organizes or will do the workshop starting tomorrow for, I think, 2.5 days. Obviously, we don't have a PacBio system in East Africa or here close by. So that's the reason why we will just do the library or the DNA preparation library, and obviously everything else that goes with getting the library sequenced. And we will then, once the data are available, get online together with the delegates to look at the data. The PacBio is sponsoring the smart cell here, and we also trying to live stream this on YouTube. We have our social media person Jovenci here. If all goes well, so I think Acclaim will share the link with everyone who wants to have a look. I'm not so sure how easy it is to live stream a workshop, because there are many different things that can happen, but we will try to do that. And I think I was told, I wrote it down, so we have a fish. It was shoemaker spine foot fish. Taffy is it in Swahili. And a weed, Streicher weed, and I looked up, it's witch weed, so I don't know if that is a good omen for the workshop, but but yeah, thank you for your time. And for those participants, maybe go to our stand just to make sure that you're here at the right time, and yeah, and also to see if everybody comes that wanted to come. Okay, thank you.
Sorry. Thank you very much, Oliver, for taking us through the workshop. And yesterday, when we were standing outside the door with Oliver, somebody called out his name, Oliver, right? And he turned around and he realized that there's another Oliver on site. And he made a very interesting comment that not many in this generation carry his name. Yeah, it seems like it's a name that was quite popular a few years ago. But that brings us to the end of the explanations on the different workshops, that practical sessions, hands-on sessions that will be held in 6 different locations on the African continent. I wanted to just spend a little bit of time to explain why this is important. This is important because on the one hand, many participants attend a conference and go home having seen videos and demonstrations of practicals and very, very few hands-on training. And this is what we want to do. We want to bring a little bit of the hands-on training closer to the people. It's not easy, especially when we had 750 applicants for the 60 or so places that were available. And that also tells you what the demand is. But we want to increase this, and therefore we are calling on institutions on the African continent who have laboratories, science laboratories, to avail those laboratories for us to be able to have workshops. And we're saying we're taking the technology at that level. If your lab is able to do DNA isolations and pipetting, that in itself is a skill that is essential for molecular biology. So you don't have to have a sequencer on your site, but if you have a nanopore, you have a spectrophotometer, you have some laminar flow, that, you know, within the value chain of molecular biology technology, there's a use for that, and there's somebody somewhere who would like to learn that technique. So we would like to close with saying how thankful we are for all the technology companies, the institutions, and the individuals who have partnered with African BioGenome Project. And I must say that this is coming at no charge for you, African BioGenome Project participants, and this cost is being borne wholly by the workshop preparers. Yes, they're being the cost is actually being met by the technology companies and the institutions that are running these practicals. We also thank you very much for availing the support. Like we've had people traveling all the way from Australia. We've had people traveling from Europe to be with us, the Illumina team. We've had the PacBio team traveling, not, from very far, but still far outside the borders of Kenya, from South Africa. And all of you, some of you who have traveled from Ethiopia, from Burundi, from Rwanda, from Tanzania to be here. So we thank you very much for the uptick.
Professor Mugai,
yes.
I think we have Laura. I don't know if we're doing the vote of thanks or we're going to come back.
We're not doing a vote of thanks. This is just to thank the technology companies. And this is the point where we'll invite Dr. Laura Okalembo, PhD, who is now giving us the next talk. So Dr. Laura, with her colleague, Dr. Peter, is giving us a presentation on sequencing for farmers in East Africa, current and future perspectives. And I think for us, what would be very, very interesting for us to hear is what is the connection between what you're doing for farmers and what the genome next-gen sequencing technologies can do for us. So Dr. Laura is currently a senior scientific consultant at BioTeam and a TED senior fellow with 22 years experience in genomics and high-performance computing research and solutions for real-world problems. Prior to joining the BioTeam, she was using portable genomics and supercomputing to help small-scale farmers in sub-Saharan Africa to control whiteflies and viruses which have devastated cassava crops. And cassava, as you know, is a staple for more than 800 million people globally. And she is doing this presentation together with Dr. Peter Seruwagi, who is a senior researcher and a consultant with 27 years plus experience in pest and disease, molecular diagnostics, surveillance, and IPM. And welcome, Dr. Laura. You can go ahead. The floor is yours.
Okay, great. Can you see my slides?
Yes, please.
Slides are there. Okay, I want to say Abari, Mbote, Apoyo, Yoga for my Soroti family in Uganda. So really thank you all so much for, number one, inviting Peter and I to talk to you about some of the work that we've done over the years, and also the organizers for inviting us, and also for changing our time so I didn't have to wake up at 2 o'clock in the morning. So thank you for that. It was only 7 o'clock this morning and 5 o'clock for Peter. So anyway, I just want to make sure Peter is on the call, and I hope he's going to speak right after. So I want to make sure he's able to be unmuted at some point, but let me see. Let me go to the next slide.
I can hear you, Laura. I hope I'm unmuted.
Oh, you're fine, Peter. Perfect. So I'm just kicking us off, and then Peter is going to take us through, but I just want to first acknowledge the team. We're presenting on behalf of a bigger team, and just, some of you know people in this photo, but, there's been an international collaboration for us to actually get portable genomics in the field with the farmers. So just want to acknowledge Monica Kehoe, Joseph Nguru, who many of you know. He is an absolute legend and has spearheaded so many amazing, amazing, amazing things. And he's the reason that I'm on the team. So he's not here with us because he's big man on campus in Tanzania, I think some sort of presidential appointee now. And then there's Dr. Titus Alachai. And there you are, Peter. Look at us in that cassava in Zambia. And on the far right is Professor Elijah Ateka from Jomo Kenyatta University. So I'm going to kick it over to Peter because there's no better person to talk about cassava than Peter. So over to you, Peter.
Thank you very much, Laura, for that wonderful introduction. And I want to thank the organizers for allowing us to add to the voices for those who have used these technologies to not only advance science, but help the end user and who is the farmer. Now, the cassava virus action project is the main focus of this work that we're presenting today. But Laura and I and the team that she's just presented there have worked on these viruses for many years, right from our own advanced degrees, but also for much of our employment. And we really are talking passionately about something that we've seen transformed for the benefits of the farmers. So in this particular slide, the Cassava Action Virus Project was really concentrating on helping the farmers. The farmers have a big challenge understanding production constraints. And when it comes to cassava viruses, they could not tell which, plants are infected, which ones are to use, and especially for viruses that do not probably show clear symptoms. And even if they did, many farmers related this to maybe nutrient, deficiencies, sometimes drought. And so the project looked to help them understand the cause of the virus in very simple ways, to understand that these diseases are caused by something that they can't see with their own eyes, but that is transmitted in the planting materials that they do share in their communities. And so it was very important to help them understand why it is important to have plant health, to ensure that the material they are using is free of viruses. So the farmer was the first, target. Next. But then, of course, we are working in an environment with different institutions, and therefore capacity building became of great urgency, especially with regard to the new technologies from Nanopore. And many of these were scientists, technicians, and students. And they were actually well trained in molecular tools. They knew what to do. But we're talking about a new technology from Nanopore. Next. Now, we all know that cassava has been previously mentioned, I think, is a disease. I think it was at the introduction that affects a crop that is, food staple to more than 800 million people globally. In Africa alone, more than 350 million. And it is also consumed, but it's also of great industrial use. Laura, next. And so we see smallholder farmers always, when you go around East Africa, West Africa, selling and earning an income. And so our own work is very much relevant for them as regards to earning an income. Yeah. But of course, it's constrained majorly by two viral diseases. One, which is DNA viruses, the DNA viruses that are cassava mosaic disease, and then the RNA, which is cassava brown streak viruses. Now, both of which are transmitted by the whitefly, Bemisia tabaci. And this, when you look at that picture of cassava brown streak, you can get total loss. This is the edible part that is attacked. When we first saw this disease, we did not understand. We thought it was bacterial because, initially we had cassava, bacterial diseases. But when we got to know that it was viral, we definitely knew that it was going to be a huge problem. Initially, constrained on the East Coast, but now is threatening to move into West Africa, Southern Africa. Of course, it's down to Mozambique, Zambia, and South Africa. And we do get total loss, but the project used diagnostics. We were using it to monitor the disease, the spread of the viruses, to understand the genetic variability in the cause of viruses, but on a regular level, using it to screen plant materials for plant health for use by farmers and multipliers. And of course, the technologies were used to, this was in University of, Western Australia, in Australian Perth. And you look at the technology we are talking about, it is expensive, it needs a lot of training, and so was not obtainable everywhere. So much of the sequencing was obtained elsewhere, abroad, in Europe, in South Africa, for people within East Africa. And sometimes when we went for cheaper services, we went to South Korea. But at the advent of the Becker, we did a part of the sequencing in Becker, in the region. So huge investments involved. And of course, the game changer becomes the mini ion portable, kit is, that, as you can see, is right in, fitting in here in the arms. And this is the magic that turned around things. And in the next slides, Laura is going to present the work we did using this kit in order to serve the interests of reaching the farmer with a technology that is highly advanced, but in a very simple way and in real time. So that will end my introduction. And Laura is going to take us through the work that we did, and we are very proud of. Thank you, Laura. Welcome.
Of course, Peter. Thank you so much, Peter, for that. Peter is clearly understating his many, many years of working on cassava disease diagnostics. The work that I'm going to show stands on the shoulders of the cassava diagnostic project for sure. So you guys have pushed us so far forward and allowed us this opportunity to use this. So as Peter said, the majority of sequencing equipment is out of touch for a lot of people, or it sits in a lab. And so at this time Peter and Joseph Nguru asked me to be on the team. And I said, oh, you know what? They're using this portable genomics thing to detect Ebola in West Africa. Why aren't we trying to use this for farmers in East Africa? Because, early diagnosis is important for farmers. If they can tell their plants are sick, they can intervene. And so we started working on trying to figure out how we can take this to the farm. And we have a project called the Cassava Virus Action Project, and we literally are we donate our time. We're not the people that have the big funding and all this sort of stuff. We're just a collective that's trying to hustle for the farmers. And so I show you this picture. This is the team in Uganda, and every single thing we need to do sequencing, from DNA extraction to analysis, is being held in the hands of the people. Okay, so this was a shift for us. We thought, let us go out to the farmers, let us walk around with them, and let us pick the samples and do the sequencing of the viruses right then and there. So this was our first, this is in Tanzania. And what we did was we sat on a blanket and we started from the leaf material. And it was wild, but we did it. And as you know, this is Brenda Muga sitting with Mama Mina. And lab work is not that glamorous, and it's really boring for farmers to sit there and watch. But the one thing that's super important about this is farmers. I think, Peter, you had said, it was taking 6 months to get back with the results, and the farmers were calling you tourists in Soroti, Uganda, because the thing is, as scientists, we go out there, we get samples, and we go back. And I watched the videos yesterday, and there was a lot of discussion about data governance and all that. And I think it's super important to remember that the farmers deserve to see what we're doing. And even though it's crazy and it looks crazy, in half a day, they're going to get the results, and it's worth it. It's really worth it to give them the respect of the results, because they're sacrificing a lot for us to be there. A lot of trust. It's a lot of trust for the farmers to let us come out there with this crazy equipment and sequence plants on the spot. So this is Brenda working on a library prep for the nanopore sequencing. So we did this in 2018. And I want to, everything we needed to do sequencing is on this blanket. I don't know if my cursor moves for you, but I'll try to point out on the top left, you'll see there's a yellow case, and then next to it, that machine is the DNA sequencer, and it's being run off of a motorcycle battery. And our colleague, Dr. Joe Stanton from New Zealand, has pioneered that portable DNA extraction machine called the PDQEX, which stands for Pretty Damn Quick Extraction. So you can look that up, or we can put it in the link, or this is being recorded on YouTube, so you can see that. And then in the foreground next to Brenda, you'll see our styrofoam chest there. You'll see the MinION being sequenced. And at the time, we were the first people to have access to what they were calling the Minute, which is the GPU compute that changes the squiggles that was discussed earlier into actual base pairs. And we were running all of that off of a laptop power bank. So now they've merged the MinION and the Minute into the new machine that you guys are gonna see in the workshops. But you can run these things off laptop power banks. And this is connected to my computer with Wi-Fi. And so we're watching the analysis. So this is the full setup. We took the leaf, we did the extraction, we did the library prep, all there on the farm. And this is a closer look at the portable extraction machine that we were using. And it's also run from a phone, Wi-Fi. And this is the person with the pipette in the foreground, or in the background there. That's Charles Kayuki, who is actually Nanopore hired him from all the great work that was happening on this project. So if he is now employed by Nanopore and he is in the region, he is in Tanzania. So we're really happy about that as an outcome. But library prep was happening in the field, and we were loading the flow cells on the drums. It was amazing because the farmers were drumming for us. So we didn't think about where we were going to put the device, did we, Peter? So they kindly brought over the drum, and Charles loaded. If you've ever loaded one of these flow cells, it's wild, and to do it in the field is even crazier. But anyway, here's another look at the power bank and the equipment that we were using to do the sequencing on the spot. And and if you know Joseph Nguru, we are firm believers, a team that selfies together will stay together. So we like to take some selfies, and this was us doing some analysis on the farm. And one of the things that was funny, Peter, about us was we didn't know if it was going to work, but when it worked, the farmers are holding the computer with the results, right? And the thing about this is we sequenced 8 or 9 of the samples the farmers were interested in, and they were all positive for virus. And you might think, why are those farmers happy? They're happy because now they can take action. And now they know that their farms need to be altered so that they have food in 9 months. So they were very happy to get the results right there on the spot. And the biggest thing about what we did was it can take 6 months with all the sending samples. Even, all the resources in Kenya are great, but Kenya is not Uganda. Kenya is not DRC. Kenya is not Ethiopia. Kenya is not, there are limitations in moving samples. So the power to the people really makes things move. And so it was taking 6 months to do this before with various things. And we did it in 3 hours. We did it right there on the farm in 3 hours. And it was a powerful thing. And a lot of people have criticized us for all sorts of things, including, why do you need whole genomes? Why can't you just do the thing is, collecting whole genomes, as we've seen with COVID, allows you to look at the evolution of the viruses, right? It allows you to be proactive and not necessarily always reactive. So anyway, we're firm believers that collecting whole genomes is really important for studying virus evolution. And if you take anything out of this, it took us 3 hours to do that on the farm. And so I have a video to show you because there's nothing more powerful than having the farmers tell you what it means. So I'm going to play. I hope you can hear it.
Nimekoswakoswa na nyoka yule mwenye sumu kali, ati yeye na kapu lake anachukua anaweka wa.
See the result right now. Yes. Asha has too much cassava. And that's a really great problem to have. Na kuzalisha mazao mengine, unaweza ukategemea kuuza kuliko, ukaenda kununua mchicha, ukanunua mbegu ya maboga, ukanunua mbolea, ukaanzisha bustani. Good quality DNA.
Okay. It's so much better to watch people's faces in the real world. Virtually, I'm not sure. But anyway, that is available other places too. Oh, how do I go to the next slide after this? Okay. I want to tell you that I didn't show you any data because I want you to feel something, but we have written a paper if you want to know all the details about the extraction methods and the analysis methods and where all the data is. You can look up the paper and it's open access, so no need to worry about that. I was also really fortunate to be able to give a TED talk about this, and that you can look at that as well. A much more polished version of myself is there. And okay, so I work now for this company called BioTeam, and there's a gentleman there who always does this trends from the trenches talk about what's going on with technology, and Peter and I have sort of synthesized, because I think we might have been ahead of our time, Peter. We did that in 2018 and we started it in 2017, and we've learned a lot. There's a lot of things that have happened, and I think I'm just going to say a few things because I know we're like a gazillion hours over time, but I think it's important to note that labs in a suitcase make it easier for people from outside to come in, fly in, grab data and run away, right? So I think we need all the discussions you all had yesterday about data governance, I think are going to be even more important now that you can go to a farm and you can sequence anywhere. I think that flying in reagents doesn't help anyone. I think that having suppliers there, and I also think that suppliers who are exclusive to one sequencing company are not helpful, right? Because I know there are politics involved in this stuff, but really we need to have these portable genomic things ready to go for people to use. The reagents not being there is a problem, and they're expensive. So one of the things that our team has spent a lot of time on is working with governments to try to get them involved early on with the help of importing and being supportive of this. So we've spent a lot of time working with borders and agents. And I mean, I've met with the president of Sierra Leone, and we are always at the EAC commissioners, aren't we, Peter? It's like governments are super important here. Yesterday, you guys all talked about the data is power. It needs to be stored and processed locally. Offline analysis pipelines, it sounds like you guys are going to do some of that in the workshop, so that's cool. And I think, very few studies are equitably designed. So I think if you know, I watched the video yesterday, and you guys are all mindful of this, but you have to be careful of people flying in, and you got to follow the money. Where's the money? Is the money in Africa, or is the money overseas? And who's on the high impact papers? Is it you, or is it the people who you're helping, right? So there's a lot of things that need to be considered now that genomics can happen by anyone that can fly in there and do anything they want. So I think that's going to be an important thing. Okay, so COVID happened, as we know, and all of us decided we were not going out to talk to farmers because of spread of COVID and all those sorts of things. And one of the things that our group did was, a year after COVID happened, there were literally no SARS-CoV-2 genomes from East Africa. And so we realized very quickly that this whole idea that agriculture needs to be over here and health needs to be over there isn't really the best way forward, right? Peter, like, we realized we had skills, we had reagents, but yet we weren't even able to engage with people because there's this siloing of basically international institutions that sort of work together, but sort of don't. And then it took months for there to be genomes from East Africa. And then when some of them were put out there, there's fear, right? There's fear of being embargoed or not able to travel. So anyway, I think that we realized we did this really cool stuff with farmers, and yet we were disconnected from the health sector, which is one of the reasons, and even biodiversity. So one of the reasons why, Peter and I really committed to waking up at 5 a.m. was to connect with you all, because we realized that when pandemics happen, we all need to be in there. I don't know, Peter, do you want to say something about this?
No, I just wanted to add quickly that we felt that the technology, molecular biotechnologies, is something that can be shared across different fields. This paper we wrote was to highlight the capacity of using this portable kit. And yet, COVID was limited by the equipment being in institutions that were far away from borders. And so you could see trade, like trucks coming from Kenya had to be stopped, and for several weeks they had to wait for PCR to be done. Yet we were doing this real time just before COVID in the farmers. So I think that we need to transcend this thinking that these are agriculturists, we just needed to be equipped as protected. We needed to have PPEs to be sure we are safe. But that was the article. The article was highlighting the capacity dilemma of big institutions having these tools and not linking with the users. You can continue, Laura.
Okay. So I don't know if I can go back, you guys. I'm not that. Okay, so I just want to say that one of the other things that was happening during COVID was I realized that, I think we all realized, Peter, that we had generated a lot of data, but we really weren't putting it somewhere so it could be reused. And I was new to the cloud and how you do data management sharing. So I joined this consulting company called BioTeam, and we do work in Uganda with the African Centers of Excellence in Bioinformatics, and it's at Makerere University. And one of the big things, and I'm sure later in your workshops you're going to see, you have large amounts of data that require specialized training for analysis. And so over the last sort of 2 to 3 years, we've been engaging with the African Centers of Excellence in Bioinformatics. And our company BioTeam has provided this kit that's on the left, this compute and storage. And so I spend a lot of time now working with students there to help them curate their data and make sure they have put it somewhere so it's fair, as they say, findable, accessible, interoperable, and reusable, because that's really important. The data is the power. So spending a lot of time with people in data. And that is the end. Peter and I have worked with hundreds of people, and we've trained hundreds of students all across the continent. And, the training is one thing, it's access to the equipment and the knowledge that matters, because you can go to a training and if it just dangles there, it means nothing. So I am somebody who really cares about that. So anyway, I will, are we taking questions? I don't know what's happening.
Yes, there will be some questions for you.
Okay.
Maybe
great.
we can take one from here and one from the online audience.
Okay, sure. Sure.
Before we do that, let's give a round of applause for Dr. Laura and Dr. Peter for demonstrating to us that genomics is actually portable, but we need the innovators, the technology companies, to be able to come up with equipment that are portable, that can be used at room temperature. And I've actually seen that farmers are able to wait patiently for their results. I mean, that's really commendable, and we applaud you, and we hope that we will be able to see more of these types of projects from. So there are people who are asking for your contacts, Laura, and
Sure.
I'm sure they will be able to find you on YouTube, but also on LinkedIn, and they will be able to connect.
Sure. Do you want me to stop sharing? Sure.
Yeah, Professor Mugai?
Yes, go ahead.
Yeah, I have some question and also comment. I don't know, you want to start from the floor first?
No, you go first.
Okay. Yeah, thank you, Laura. It's good to see you again. I don't know, many of us may not know, but Laura, I got to know Laura in 2019 when I was writing the concept note for the African BioGenome Project. So I was moving around the world, trying to contact, key people that I think could come on board. So I spoke with Laura first. I think that was in August or September. So I think, Laura, this is the evolution of that concept note. I know I saw your tweet the other day and it resonates. And what we've achieved, perhaps it could be a discussion for another day, but we bring quite a lot of people together, and I'm sure this will provoke action. And a lot of what you've just said resonates, basically, because I know you do quite a lot of work with Nanopore, particularly. And one of the things I have known, or I do know, is that we need to, just as Professor Mugai said, take it closer to the people. And one way in AfricaBP we're doing this now is, through these regional workshops and distributed practical sessions. So, because one of the things I've thought about is we could have an equipment in, say, South Africa or elsewhere, and someone from Cameroon goes there or to UK or wherever, and they return back to Yaoundé and they don't have the, high maintenance equipment in Yaoundé to continue. So we thought that going forward, we are going to focus on what is available in the region. So I don't know, is Professor Leibert here? Leibert, you want to wave? And the Professor Antoine? So one of the things we said is, let's work with people on the ground. And you can hear what Professor Mugai said. If it is pipetting, if it is DNA extraction, only these are good skills. So again, many African scientists, with this workshop, we know many people have the opportunity. When I started my PhD, I got a scholarship to Cambridge. I didn't have the huge opportunity in terms of, with the practicals. So we have to read, study more hard and things like that. So now we're saying, let's go to University of Yaoundé 1, go to University of Kinshasa. People don't have to travel far away. And what that does is that it does one of two things. Laura, it does something closer to what you're saying, whether you use the nanopore in that location or use the equipment. So if someone in base is Kinshasa, they don't need to travel out of Kinshasa or something, because those things are there already, and they don't need to start looking for the trainer. Perhaps the trainer have actually gone back and things like that. So, but I have 4 questions. So that was more of a comment, and also thanks to Peter as well. I saw while you were making the presentation, this actually resonates to me, because when I grew up in Africa, I was one of the beneficiaries of cassava, because cassava was used to pay my school fees from primary school to secondary school, and then when I got to university. And of course, in Africa, women, when it comes to cassava, that is very, very, is actually very important. It serves as a source of revenue for people. for most people in Europe, sometimes I know maybe the school fees is free and the government does this, but in Africa, you have to cater for yourself and do all of those. But I have 4 questions for you, Laura. The first one is, so it'd be nice to kind of, again, if privacy allows for you to get, for us to get the slide, because the presentation connects. I'm sure it connects with so many people on this call today. So if you can get the slide, that's 1. 2, I would like to know, what is the stage of this cassava virus project? So I'm not asking because it resonates with me, partly because I, as a young boy growing up back in northern Nigeria, I actually benefited from cassava and taking it to market and then getting it sold and then using it to pay school fees and then having to read and things like that and get good grades. So, but I'm asking now on a more general, neutral platform, what is the stage of the projects, and can you also share maybe perhaps the pushbacks you got and what were the challenges? And if we also know the stage, how can AfricaBP come in? Because this might be helpful in seeing, okay, if the projects have stopped, how do we come in? How do we support it? And how do we ensure that this continues to make impact? And of course, I'm also, one of the things I've said is we have the reference laboratories. We also have taking all of this to closer to the people through also other means in both ways. So if you can explain those things, that would also help to kind of see how can we make this more sustainable, particularly.
Okay, number 1, you can definitely have the slides. I sent the link to the lovely Josiah who coordinated all of this. So big shout out to him for helping us. I sent the Google slide. The video is kind of bulky, but the slides are there for access. The state of our project, it's interesting. So we were doing this in 2018. We got a lot of pushback. As I said in the presentation, people thought that we were, first of all, you have to know that the politics around cassava are huge in the international development space, because there's millions and millions and millions of dollars being given to researchers to study this. I don't know how much of it is going to the African continent, to be honest with you. I mean, many of the big high-profile projects are based in the UK, which is a problem, in my opinion. I'm going to speak truthfully here. I know this is live on YouTube, but I'm going to just speak the truth about what I think, because I can, and it matters. So you have our group who basically decentralize sequencing, and you all understand that when you partner with people outside of the continent, they're getting millions of dollars to do sequencing. So we now disrupted that common way grants are written and the way people basically benefit off the backs of African scientists and people. So we got a lot of pushback for doing this because we disrupted the thing. And a lot of people doubled down on regional labs for sequencing. And here we were taking it out to the field, which didn't go well for a lot of people. So there's just a lot of politics in cassava research. That's all I'm going to say. Peter, I know Peter is much more like, but me, I'm like, There's just a lot of politics in it. And we got a lot of pushback, and we got told, oh, sequencing the genomes is overkill. Just do take a picture on the phone, or, by the time you're not going to, we want early diagnostics, right? We want when the plant looks healthy, but it's sick. By the time I can take a photo of the leaf on my phone, it's too late, right? So anyway, I think that there's space for all of these things, I think, is the summary in now that we've been doing this a while. So our project is, I think there's space to collaborate on the project and share a lot of the things that we're doing. A lot of us are still waiting for the perfect opportunity to reinvigorate the project, because we want it to be, we will always do what's best for the farmers and the scientists in the region, and not for the people who want to buy us. Does that make any sense? I don't know. There are like 200 people on this call and I'm speaking like this. But anyway, Peter, do you want to say something?
I actually think there are also more on Twitter and YouTube. So,
oh, good Lord. Well, I think it's important. I think it's important to be truthful because you just saw something that literally happened, not from millions of dollars giving us a grant and writing a project. It was literally grassroots. Let's work at the national agricultural centers and let's do what we can to make it happen. Like,
I agree with you because I did say yesterday that AfricaBP is being built from the ground up. Yes. So we started. You probably also know when I did reach out to you, Laura, and I think it didn't start in that September or so. But in March I kind of thought let's do this grassroots because that's one way that I've thought that this could work. And grassroots to me means grassroots. It means someone in Uganda, someone people who would otherwise not have the opportunity, and to actually get some of these things. So maybe Peter, do you want to comment on some of this? It would be nice to also
to quickly comment that the room for collaboration is there, because even though we are not completely now with the national institutions, we are in positions where we're doing the same work. We can still do it. Now, one area that before COVID we were looking at is using this simple technology at the border sites of countries to facilitate trade. And we did write a project which wasn't eventually funded because of COVID with the East African community, for instance, and they're having these challenges of having, problems of, let's say, they have rice, but people are packaging rice that they call a certain variety, which it isn't. And they are struggling because morphological characterization is not possible, and yet we can use genetic tools in a very simple way like this portable sequencer without too much facilities at border points. All we need is build a genetic base of these genetics, or whether it's corn, whatever the crops and the pests in the regions, and be able to revolutionize the way we do science. So that's not just science for the sake of publication, but facilitating trade, better production and all that. And somebody asked whether we could use this in livestock. Laura, you remember you had a student who used this at the University of Sokoine University in Tanzania?
Yes.
Yeah.
Yeah, people are using it in livestock for sure. And I think you're right, Peter. We realized that in order for the project to be sustainable, we had to get government buy-in. And that means showing the utility of this in higher value crops, right? So in Tanzania, we were working with rice, and then we went to cotton. Cotton is something that's really important that has viruses as well. So I think where we were thinking the next sort of steps would be, of course, we've shown it in cassava, but getting some of those higher value crops, showing the utility there. We thought the governments would be more supportive and possibly, help with allocating resources to the scientists who are doing the work on the ground. So that was our trajectory in the evolution of the project. So
thank you, Laura and Peter. So I think what would happen is I would again outside the workshop, carry on the conversation and see how we can explore to try to carry to do maybe another version of that. Hopefully, maybe with the discussion with Anne and Catherine and others, we can see how that will go. Yeah, I do agree that we need to take this closer, to the people, and then having the reference lab and having the portable equipment and things like that.
So, I think the mixed approach is smart. Thank God, it's really smart to have, because you need the hubs to help analysis, and then you push it out and you refine your method in the lab, and having that hybrid sort of approach really helps move the technology closer for sure.
Absolutely, that is definitely we're having the hybrid approach. So this is why we're doing this, because I've seen that. I've seen what it can do. Even 3 years ago when I reached, or 4 years, I think it's 4 years ago, I can't remember, when I reached out to you, essentially. So we'll carry on this conversation. We'll be in touch as AfricaBP, either myself or Anne, and then we could see how can we could potentially have a version 2.0, wherever that is, and how can we, make this more sustainable and also support what you guys have done so far. Over to you, Professor Mugai. Okay.
Thank you very, very much. That was, a very, very interesting conversation that you had there, talking about accessibility, talking about bringing technology closer to the people, looking at the actual needs on the ground and addressing these. And this is really, really important. And definitely, this is a sort of collaboration and partnership we will definitely want to take forward. And let's give a round of applause for Dr. Laura and Dr. Peter for their excellent presentation and for showing us that it's possible to be bold with your idea and taking it to where it matters, and we can actually see the results. And thank you for the courage of going against the grain in protection of the African farmer and the African scientist. We want to switch gears a little bit and begin another aspect that speaks to the heart of the African BioGenome Project, and that is precisely the mentorship that African BioGenome Project is envisaging for early career scientists. Myself, probably, thank God, is going to be around for a much longer than some of us, but I want to think that for Africa, it's not only important to do this capacity building, but it's also very important to ensure that those who we touch, those who we train, those whom we mentor, take over from us and actually run away with their dreams. And this is something that speaks really, really to the heart of my being as a person, as a university professor, lecturing, teaching, doing research with many, many graduate students. And therefore, this particular session is a panel session on what does it take to become an established scientist. And to help me discuss this, I want to invite Mr. Paul Kagame, who is a PhD student from Ghent University. Come forward, please. Let's give him a round of applause. I must admit that Paul is my student currently. We also have Miss Vivian Kathambi, who is a PhD student at Ghent University, an employee of the National Museums of Kenya. And well, I have interacted a lot in the field with her, and I'm very happy to see that she's stuck with the science. And welcome, let's welcome her with a round of applause. I also want to welcome to the floor Dr. Eunice Machuka. Dr. Eunice Machuka is a fresh PhD graduate from Jomo Kenyatta University, another of my students. Okay, I mean, I had to bully them into coming, and that's why we selected them. And as they come up to the floor, I would like to just engage them in a few questions. And this is just sort of like to act like a motivation for all the early career scientists that are there who are progressing from master's to PhD and from PhD to postdoc and on to becoming established scientists. So perhaps we can start with Dr. Eunice Machuka and ask her, when did you graduate? How did you know what was the right project to choose? How did you select an area in which to become an established scientist in? And is it worthwhile? Are you earning more money now that you have a PhD? Is it worthwhile? I mean, I don't know whether it's a Kenyan or African thing. We follow the money. So I want to know, was money your motivation, or on what was it?
Okay. Thank you, Prof, for the opportunity, and thank you all for the attention as well and the questions. So I'll assume the graduation you refer to is the most recent. And so I graduated in June 23rd, 2023 from Jomo Kenyatta University. It's a joint degree with the Pan-African University of the Institute of Basic Science. It's a mouthful. It's PAUSTI in short, for those who want to either find more about this. They run scholarships and they have fellowships for MSc and PhD. So I have been working for a while, actually. I'm one of those people who've been in research for the longest time. I had an opportunity to do my PhD immediately after my master's. That's way back. I'm not citing years so that you do your math backwards, but that's not a trick. But then I got an opportunity to go and work at a medical research institute in the Gambia. It's called the MRC. And I thought that would be a better way to gain experience and probably gain focus as I decide on whether I want to continue with science. So while at the MRC in the Gambia, I found science to be very, very interesting, very exciting. With science, there's the ups and downs, there's the interesting side, the fun bits. But what made me want to do science is really the urge for me to improve society. I have an inclination towards clinical research, that is to improve the livelihoods of humanity, or looking at livestock, or in my field, because I did molecular biology, I can work with either humans or livestock. So my tools of engagement would be applicable across both humans and livestock. So there's that passion of wanting to improve livelihoods, but also you asked about the money. It depends on where you work. So if you are in the private sector, it does pay well. But also the hours are much more. You put in a lot of hours, a lot of family time taken away. So the sacrifices are there, and it needs really the motivation and the keen interest and the drive to want to be away and, put in the hours. Yeah, but no change yet. So we are hoping to transition. As you said, it's a bit fresh. So at ILRI, we are transitioning hopefully in the new year. Thank you.
So thank you very much, Dr. Eunice. And now I turn over to Vivian. Vivian, you have an established career in the National Museums, where you have established yourself as a specialist in taxonomy. And what motivated you to apply for a PhD, look for a PhD and commence? I thought you were very comfortable. And was it because there was more money? I mean, I have to understand whether it's really a money issue or what is it? Just tell us.
Okay, yeah, I have a career at the National Museums of Kenya as a taxonomist and an ethnobotanist, but I realized I am truly passionate about rural communities and their quality of lives, and to influence decision-making maybe in Kenya and even regionally, there is a qualification that's required. But I also knew that this was part of the plan, like from the onset. I knew I would go to the end of the studies. And I think science for me was like, it's inborn. I was caught so many times trapped in streams looking for tadpoles and all those things, and burying chameleons and singing songs, burial songs. I was very attached to nature since birth, but also the quality of life. To be truthful is when you grow yourself, your quality of life also improves, and those that are around you. So it's a personal goal and a career goal to continue with this. Thank you.
And Paul Kagame, let's move on to you, because you finished your undergrad in Jomo Kenyatta, and then you won a scholarship to go do your master's in botany in Wuhan, not knowing that Wuhan would be ground zero when it came to the pandemic outbreak that was experienced. And what was surprising is that you chose to stay in this city where millions were losing their lives in order to earn a degree. I perhaps need a little bit more explanation on that.
Okay, thank you so much, Prof. Yeah, I graduated from JKUAT, that was recently in 2017. And then I worked as a research assistant at COHES, still at JKUAT. And then, yeah, I was lucky enough to get a scholarship and Belt and Road scholarship in China, where I went there and did my master's. And as you said, nobody expected it. Yeah, so when it happened, it was quite hectic for us, for the students who were in Wuhan at that time. So I'm always grateful to God, the point that I survived during that time. And honestly, I wouldn't tell you that, like I wanted to stay there, but what I wanted was just to finish whatever I went to do in China, because I knew out of that, I will get something that I can come back home and share with other students, come back home and share the experience that I had in Wuhan, the experience that I had during the outbreak and the how do they call it? The curfew and everything. So for me, it was a learning step. And I think out of that, it made something in me also like come out, And yeah, I'm grateful for that event, actually. Yeah, I must say that. Yes.
And of course, it's open doors because you were able to finish your master's and now use your master's to register for your PhD. But it wasn't easy.
Yeah,
How many applications did you make? And did you get accepted on your first one?
I did. It was over 100 plus. And every day I used to do them, the time zone in China, it's quite, they're quite ahead. So I used to do them in the evening. And then when I wake up, I just wake up to see regrets, regrets email. But fortunately, I managed to get out of 100 plus, like 3 of them. And I had, I chose the one in Ghent because of the project. I was so convinced that I don't just want to do a PhD. I want to do a project that can have an impact to the society, try to solve societal problems. And when that opportunity came, and it was like an open door to bring different institutions together, that is JQuad, Royal Botanical Gardens, Kew, Ghent University. So I really took it, and I knew out of this PhD program, other people will also benefit, because out of that, we had an MOU also with JQuad and Ghent and Kew Gardens. And from there, I've seen a lot of students trying to get opportunities also in those institutions. So it's not only just doing the PhD and enjoying doing the PhD and the project, but also trying to help the society, try to encourage the young generation of scientists to work towards that path, get the knowledge, come back home, and empower the young generation.
Amazing that over 100 plus applications to get 3 acceptance letters. And the question in mind is how many of us give up after, getting 1 or 2 regrets? And if you have gotten 80 regrets, do you have the energy to go on? And you have seen that you have to go on. But I want to turn to Dr. Eunice and ask her, do you think that it's important to earn a PhD in this discipline? Or and how do you select a supervisor or a project?
Okay, I think it's important. As I mentioned, I had backed down on taking on a PhD earlier, and went on to work. I worked in the Gambia for about 4 years, and I realized you can work at some levels, but there's so much you can do at a certain level without a PhD. For example, you cannot lead a project. You cannot be a PI for some projects. There are some things you cannot do without a PhD. If you're in academia, there's some things, for example, you cannot supervise anyone with a master's or a PhD. So you cannot grow in your career. So I think it's important if you're considering the academia or the research path, or if you want to lead your own research team, that you get your PhD, if possible, much earlier, so that you can have the skills and the experience to be able to really get to that level. In terms of choosing a supervisor, I think, first of all, it depends on the area that you're working on, the topic that you're working on. And if you have the flexibility also, if you already know the person that you'd like to work with. For example, I had the privilege of being at ILRI. I had been at ILRI for about 4 years before I chose to proceed and do my PhD. And so by then I knew the area I wanted to work and the people I wanted to work with. And at the university, I inquired, you may not know this already, she was my professor. I inquired for a list of professors who are experts in the field that I was choosing. And the coordinator of our program gave me a list, and I told them, I'm looking for a lady who's done either genetics or biotechnology. But then I also went online and I read, you may not know this as well, I read your work and I was like, I want this one. So the coordinator was like, how? She never taught you during the coursework. How do you know you'll be compatible? I was like, no. I read her work. I want to work with this person. I think she will be a good addition to the team. So I was lucky because I was allowed to also choose at the university. So some universities may not allow you to choose, but it helps when you can contribute by either reading their works or if they've taught you and you've really had good interactions during the lectures or during any other sessions. It's good. It helps even, for example, during such sessions, if you see someone that you really like the kind of work they do, to say a word or two to them, and you never know, they could be your potential advisors in the future. Thank you.
So I can see Vivian nodding and agreeing with you, Dr. Eunice, and I want to know what she's thinking. Eunice. Sorry.
I'm thinking supervisor or your mentor is a very important person in your academic journey. For my master's, I had a difficult time with my professor because the people skills were and I had no idea what I was doing. I really struggled, to be sincere. I almost quit, but I didn't. I continued, and I thank God I was graded excellent. And that's where we met with Paul in China. I was also doing my master's in China. And now for my PhD, and I knew I could not, I was actually offered an opportunity in China to continue with my PhD, but I declined because I knew I didn't want to study in such an environment. And perhaps even for another thing, if you're frustrated, you cannot do quality work in academia. So I decided to come back. I was hopeful. I trusted God to come back, continue looking for opportunities, and thank God I got one. Yeah, thank you.
Perhaps, Paul, you can contribute to that debate and maybe even tell us about the role of mentors, because you were able to publish right from your undergrad, I think, and on to your master's. What is the role of mentorship, and maybe something about how to choose a project and how to choose a supervisor?
Yeah, so for me, I think from the start go, even when I was doing my undergrad at JQuat, there was always this feeling of trying to solve societal problems. And once you already know what you want to do, what you want your research to align with, I think it's now easier for you when it comes to looking for a supervisor, because now you're looking for someone who at least will mentor you towards that particular direction. But I must say, during my master's, it was also quite hectic, because I was getting in the field of bioinformatics and I was so fresh. I didn't know anything. And the supervisor that I had, there they have a different culture. Maybe they're expecting you already to know all these things, and then you go there with a fresh mind. So you really need someone who is patient enough. And sometimes as a student, you feel like you're being pushed so hard. And at that time, you feel like you want to give up. But as time goes by, you start being appreciative of that particular era of your life. because right now, I might say like I'm having a quite, I can say, easier time compared to how I was during my master's. But what I'm feeling right now is as a result of what I went through during my master's. So I think when it comes to looking for a mentor and looking for a supervisor, it's critical, and you just need someone who will understand you, someone who will be patient with you, and someone who is willing to see you grow. You know, sometimes it reaches a point your supervisor he wants you or she wants you to stay there, but at the same time, she's pushing you, like talk up her, just go. I want you to go and develop and network, and then you'll come back here. I think the same thing what also you, Prof, you did with me. I was in JQuat. You told me to go and try new things in China. When I finished in China, you said, no, you cannot stay there. You have to go and try other places. I moved to the UK. You told me, no, you don't have to stay there all your life. Go somewhere else. So also, if you have that supervisor or that mentor who wants you to grow, I think it's very critical in someone's career. Yes.
May I just add something? Paul, you've said something that has resonated with me. At one point when I was starting out, I thought a supervisor was supposed to, work with me, hold my hand, do ABCD with me, like we, on a nature walk. But I learned that actually your supervisor is meant to just, point you to the right direction and lead you to your independence. So at the end of it, it's almost like you occasionally just check in on them and you come out of this really very independent. And as a mentee, this is a way to gauge whether you've really grown, as Paul puts it. And as a supervisor, this is an achievement. If your student is able to come out of this independent, able to really make these decisions on their own and, grow. The aim is just to be independent and, like letting go of a chick that can fly out there on their own.
Yeah, about that. It's unfortunate that you can be a doctor who is not confident because you are not allowed to get out of that cocoon, because that's what you find, and that frustrates you, especially if you've studied abroad and come back to Kenya. Especially coming back, you are so not ready for what's happening. Like, maybe you didn't grow writing grants, proposals. That's what the institution will need you to give them. So if you didn't have that exposure during your studies, it becomes so hard for you to fit in. And it's also a place of readjusting. So it's so important to stay with someone who pushes you, yet guiding you, because you don't know anything. You don't know so much, you know something, but you also, I remember my PhD supervisor said, Vivian, I want you to do that PhD that you'll be an authority in and that you want to do. And now I'm actually eager to do the best and get there. So yeah, it's good for also coming back and giving back, because at times you don't understand what your employers need when you come back with that degree. You need to know how to be here and work and produce results. Yeah.
Yeah, I think what I can say is that in life in general, sometimes you have to know what you're serving. And I always tell my students or colleagues or friends that it's either you're serving your purpose or your need in this life, and the only difference between these two is that when you serve your needs, it's only you and the immediate family members who will benefit from you. But when you're serving your purpose, not only you, your family members, but even the people you don't know, even people you've never met will have an impact, will feel an impact from what you're doing. The only problem with serving your purpose, sometimes it takes longer for you to see the fruits. But when you're serving your needs, you're getting the instant gratification there. You're working, you're getting paid, you help yourself with the money that you're getting, you're helping your family with the money that you're getting. But when it comes to serving your purpose, it always takes time, and sometimes that patience is what we usually don't have. So just know what you're doing. What's your motivation for doing that master's, for doing that PhD, for doing that postdoc? Are you doing it because you want to help people? Are you doing it because you want to solve societal problems? Or are you just doing it for you to be called a doctor or a professor, or you just want a pay rise, or you just want to feel good? So that will be my last words.
Let's give them a resounding applause as you take your seat. I think this is a conversation we can have for very, very, very long. Is it worthwhile to go back to school to do a master's? Is it worthwhile to go back to school to do a PhD? And now that I have a PhD, do I have to go back and do 1 postdoc, 2 postdoc? And where is that going to lead me? We just want to inspire you as African young scientists that there is a whole future and there is a career in science. Most of us think that science doesn't pay, but remember, not all pay is monetary. You get to see that farmer getting her results on the farm immediately. You see her children going to school, you see her building a house, and those are some of the paybacks that you get. But science also pays. And remember, as a scientist, you get to travel. You get to travel this entire world. I mean, you go for workshops, you go to the field. It's only through science that I've been to northern Kenya. I mean, it's not a place you would select as a family destination for a holiday, but research has taken me there. So if you want to see the world, stick with science. If you want to actualize your dreams, stick with science. If you want to make a difference to the people who are around you, people you don't know, people who will write to you and say, thank you for diagnosing my cassava tree because now I'm able to build a house. That is the impact of science. And I think it's more rewarding than you driving a nice big car or living in a big house. It's more rewarding when that neighbor's child who would never have gone to school is able to go to school because of something that you've done. So stick with science, do science, write grants, inspire the next generation, be a good mentor, use this facility and any opportunity you have to open doors. And that's exactly what we want to do in African BioGenome. We are working with scientists, but we are also very, very keen on mentoring early career scientists and pushing them up to the best that they can be, because they are the next leaders of the institutions that will be doing this genomics and bioinformatics. So we move on to the last presentation of the day, which will be by Dr. Thangod and Professor Julian of Usuji of the African BioGenome Project. And they will be presenting a little bit more about the African BP Open Institute workshops, which most of you have been asking me about. The fellowships, the Institute fellowships that are upcoming. And I turn over to you, Thangod. You can share your screen and tell us more about this. Thank you.
Okay. So can people see my screen, please?
Share it again, please. It's not come.
It's not come. Okay. So I need to. For some reason, it's telling me that it's just showing like plain black. So if I pass this on to, so if I pass this on to,
pass it on to
Edwin.
the IT.
Yeah,
Yes.
I think that's what I'm gonna do just now. So let me grab it. So Edwin, I think I'm just gonna send this to you, please, and then I'm gonna find Edwin on the call, and then I'm gonna send it to Edwin. Okay, Edwin, I just sent you the slide, please. On the chat is a link, a Google link. Have you seen it? Please confirm and help share. So is Edwin on the call
Have you
yet?
sent it on the email?
Yeah, I've sent it on the email. I've sent it on the chat box. So I sent it to Edwin. Is Edwin on the call? It's on the chat box.
Is it the Zoom chat box?
Yeah. So I'm not sure. I think my screen is still showing.
Okay, so he's just typed out his email address on your chat box. Just check and send.
Did you type it here? I can't find it. I can't find it.
Thank God. Do you need me to try to help share?
Yeah, I think that's yeah, because for some reason it doesn't let me share on my
Okay,
machine,
if you send me the message in the chat box, I'll
yeah.
try to log in.
So do we have Osuji on the call, please? Julian?
Yes, I am here.
Okay, Julian. Okay. So Julian, if you perhaps Julian might, because he will be doing the presentation first. So Julian, do you wanna try to share from your end?
Yeah, I think, can you see my screen?
Yes. Okay, go ahead. I think that works fine. So so I just sent you the yeah, I think that works fine.
All right. Thank you. Greetings, everyone. You're welcome to this presentation on AfricaBP Open Institute Fellowship, building the next crop of African biodiversity genomics scientists. We all know that Africa is rich in biodiversity, a genetic bedrock for building sustainable and climate resilient ecosystems. This ecosystem provides food, promotes health, offers alternate and clean source of bioenergy, buffers the impact of climate change, and supports livelihood. Unfortunately, the loss of this biodiversity has far-reaching damaging effects on all life forms, our environment, and livelihood. Africa BioGenome Project, to attend to this situation, was established in 2021 with the goal of sequencing 100,000 genomes in 10 years, thereby generating 203,000 gigabases of DNA sequences. AfricaBP considers that the best approach is to support sequencing and analysis efforts on the continent and sees the need to develop the manpower of African scientists to do this. And for this reason, we have the quest of getting this done through the grand challenges. And one of the grand challenges, one of the 5 grand challenges is technology development, knowledge exchange, and the industrial grand challenge. This grand challenge established the Open Institute for Genomics and Bioinformatics, which is meant for capacity building. And this Open Institute, to do this, has established a graduate fellowship program, which is an international exchange program, which is supposed to run on partnerships. The partnerships is tripartite. You have the AfricaBP, you have African institutions and partners, which include the institutions and the corporate bodies. And to justify this, we have a program that will generate sequence data and contribute to knowledge and technology transfer, provide support for bioinformatics and genomics capabilities, and leverage on already established partnerships. This has some objectives to produce at least 20 beneficiaries. This number can change. It's not hard now because we are still working towards it. But then the objective is that these people who are going to be beneficiaries of the fellowship will be in biodiversity genomics, agricultural medicinal industrial raw material genomics, ethics, policy, and social issues, and sampling and biobanking. Basically, these are going to be their areas of expertise from which they will get launched into the fellowship. And then the role of responsibilities of the partners are here. For instance, we all know Africa BioGenome Project role as a coordinated Pan-African effort to build capacity, generate, analyze, and deploy genomic data for improvement of sustainable use of biodiversity and agriculture across Africa. And then the partners will have their own rules. Roles of the partners include to promote homegrown and regional research networks for human capacity development in Africa. They will also contribute funding for African graduate students and researchers to study in tertiary institutions and recognize the capacity of Africa BioGenome Project for assessing regional students in biodiversity genomics. They are supposed to commit to joint calls for fellowship. You may know that we have already had a call that was sent out, I think, last month. And this call, which is a joint venture between Africa Genome Center in Rabat, Morocco, and Africa BioGenome Project, is a fellowship in biodiversity genomics and bioinformatics. And interestingly, about 1,292 at the close of last week applied, and out of this number, 20 were in the process of applying, and 238 of them have already submitted the application. And that also gives, more credit and value to the present fellowship we are working on. We may recall that at the end of the workshop in University of Port Harcourt last year, Africa Center of Excellence, field research had promised to get in, some students of Africa BioGenome Project to come in their programs and then have scholarship. That actually gave us the beginning. That was what started this fellowship in practice. And now we have restructured that call, that interest, to involve all of the African AfricaBP institutions that have actually hosted this 2023 series of fellowship, workshops, plus 20 other institutions that have joined in this call, in this workshop as partners, hosted workshop, workshop practical and other in-person activities. So basically, this new fellowship we are designing now, working on, will include about 20 African institutions, not just University of Port Harcourt. This fellowship has, some eligibility criteria. For instance, those who are interested in applying as fellows should have should be African and then have affiliation to any African organization or institution. They should have relevant experience in fields like molecular biology, genomics, genome sequencing, assembly, annotation, bioinformatics, sample collection, and ethics. They should be at any time, they should be at any stage in their career. For instance, they could be students from BSc to PhD, they could be lecturers, up to full professors. They could be industrial professionals, NGO, and governmental professionals. The fellowship has responsibilities for different levels of participation. For instance, we have responsibilities for home institutions of the prospective fellows. We have responsibility for prospective fellows, those who have been selected, and then AfricaBP. For instance, the home institution should have organized any of the 2023 AfricaBP Open Institute regional workshops, hosted any of the practical sessions, or whose poster adverts, will be there for the 2024 workshop series call. The institution should nominate a candidate for the fellowship, should be also ready or capable of sponsoring the candidate. I'll be happy also to develop curriculum and research programs in biodiversity, genomics, and bioinformatics at the end of the fellowship and cover possibly visa processing fees for the fellows. The prospective fellows also have some responsibilities. First, they should be able to apply for the fellowship on the AfricaBP website. To be able to arrange their home institution for nomination and provision of a letter of support. They should be able to identify an African organization for the residency or internship. They should arrange with the home institution to develop proposals for research programs and biodiversity genomics and bioinformatics. And then should be able to design and deploy curriculum and research programs in their home institutions at the end of the fellowship. The selected fellows should be able to apply for a visa, for the international phase of the fellowship. They should be able to arrange with any of the African Genome Center to observe up to 1 month residency or internship before starting the international phase. They should commit to implementing the development of curriculum and research programs in biodiversity, genomics, and bioinformatics in the home institution upon completion of the international phase of the fellowship. The selected fellow also has the freedom to seek other sources of funding and could self-fund. Such person can willingly fund himself or herself. AfricaBP and ourselves will have responsibilities like provision of scientific directions for the fellowship, call and for applications and advertisements for the fellowship program, shortlisting of the selected fellows, and AfricaBP also should help to seek additional funds for the fellowship. The funding structure is that the home institution should be able to, like I had mentioned earlier, take care of some of the costs, especially within the Africa Genomics Center. And then the partners who are in this whole tripartite system should also bear the cost of the fellows in the countries where they are, where the research is holding. They should sponsor the research, take care of accommodation and living expenses for the period of stay away from home institutions. Well, at the end of the fellowship, we have some outcomes which are expected. First, the fellow at the end of the fellowship should be capable of developing curriculum for genomics and bioinformatics programs in their various institutions across Africa. The fellows also should be able to develop research programs in genomics and bioinformatics in their various institutions, and should develop leadership skills in genomics and bioinformatics needed to set up or drive the proposed AfricaBP Open Institute Centers of Excellence in biodiversity, genomics, and bioinformatics. We also have some outcomes, expected outcomes for the AfricaBP and the partnership as a whole. We should have at least 10 beneficiaries, could be up to 20 for 2024, and at least 1 reputable journal publication to candidates at the end of the fellowship. And the entire program should support capacity building and academic mentorship, and should also affect more biodiversity data, generate more biodiversity data for agricultural conservation, and biodiversity genomics. Thank you for listening.
Yeah, thank you, Julian. So I wanted to add some comment, but I wanted Professor Julian to finish up. One of the main things in AfricaBP is on, how do we equip and, give more, opportunities to African scientists. And there are 5 grand challenges. So if we recall, rather, in the presentation by Professor Anne Mugai yesterday, we talked about the 5 grand challenges, which is bioinformatics and genomics technologies for the agri-environment, conservation of endemic species, and we also have, policy. We also have the knowledge exchange and industry, and of course, livestock improvement and health. The meeting we're having today is focused on the knowledge exchange and industry grand challenge, commercialization, knowledge exchange and industry. And within that, we call it the Open Institute. Mainly it's going to be open. It's also, the idea is to lower the barriers that oftentimes prevent the advancement of genomics and bioinformatics knowledge across the continent. We published the progress we've made in the Open Institute in September in the journal Nature Biotechnology, where we highlighted, what we've done and where we are going to. And in doing that, in it, we have more like a knowledge exchange component as one of the priorities. And in that priority, one of the ways we want to start to deliver it is on providing fellowship opportunities to African scientists. These opportunities will be in 2 folds. The first fold would be more of like project-based fellowship opportunities. So, and that's actually during the workshop in Morocco, we had the opportunity to begin that. And I think that is also one of the benefits of this workshop, because it provokes, action and progress. So I'm also hoping that at the end of the East and Central Africa workshop, there will also be provocation of such progress. In the West African workshop, we also had a similar opportunity, and that has now metamorphosed into the African BioGenome Project, the African Biodiversity Fellowship for Emerging Genomics Leaders. So I'm going to talk about the first one, which is more of project-based. The idea is that we will partner with African genome centers or African institutions with high genomic capabilities. There are on the continent, and we'll work with them alongside other partners to provide funding, and we would recruit, African scientists to get trained end-to-end on genomics sequencing. And when I mean end-to-end, I mean end-to-end all the way to publication. So we started the first one. We launched that in partnership with the African Genome Center in Bangor in Morocco. And the idea is that the application is currently on. I also just shared it in the chat box. The idea is that we would train, we would have 3 cohorts in a year. And hopefully we can take around 10 scientists per cohort. So we're looking at 30 scientists per whole cohort. But if that number drops, so we might look at maybe 5 scientists per cohort. So it's going to be very simple. We're going to have like library preparation, for both Illumina and PacBio sequencing. And then we're going to go all the way to the actual sequencing, the analysis, the assembly, and ultimately getting the data as a report. So that report would be either a genome note. So in a genome note, whereby we describe, oh, this is the statistics, this is the N50, this is the number of contigs, just as typical statistics, and just a few things about the organism and things like that. Or depending on the results, we're going to go all the way to full publication. So the idea is that we'll recruit around 10 or 8 or 5, depending on the numbers. We're going to finalize the numbers after the call. Then we are going to, those people, let's assume at the moment 10 people, they would spend time. So the fellowship is the sequencing, the lab space, the computing infrastructure, and everything, the catering and meals, again, is covered, basically. But either the home institutions or the participant will be able to cover that. So in the future, we'll be able to we're still in discussion to see how we can get more funding to cover 100% of the thing. But the main thing is that the laboratory space, the reagents, the consumables, every other thing, and the time they spend there is actually covered for 1 month. So they're going to spend 1 month there. So we're also in discussion with others to see how we can have those pockets of fellowship, because it's very clear that we need more people to get involved in this, to actually do end-to-end and just spend 1 month, do sequencing end-to-end, get it all the way to publication. So that is the first one, and hopefully we'll be able to do that 3 times in a year, and that would potentially give us around 30 scientists in Africa who could go from end to end for both PacBio and Illumina. And the idea is that we're going to merge this data, the PacBio and OmniSea on Illumina NESIC. The idea is that we're going to merge this data and then hopefully create a chromosome size assembly. So it's going to be an intensive 1 month for the selected candidates, and it's more on the advanced side. So that is one fellowship. And so we're also in the lookout for other opportunities. So if there are people or anyone who wants to get involved in that institution that can actually support such project-based, then that would be great, especially in your countries, then that would be great. Then the second one is that we're in discussion with other partners to have the Emerging Leaders Fellowship. So it's going to be very simple. One of the biggest challenges in Africa has been curriculum development, research, development of research programs and things. And we also highlighted it in our paper in Nature Biotech quite recently. And the idea is through these international fellowships, is still under discussion, and hopefully we'll be able to finalize this either next week or in the new year. We'll recruit. So it's going to be tied to these workshops we're doing now, the regional workshops. So it's going to be open only to institutions who are participating in the AfricaBP regional workshops, either mainly by organizing the practical sessions or by hosting the regional workshops. So the idea is that this will allow us to ensure continuity and to ensure mentorship along the line. So when the opportunity will be open to those staff in those institutions, or members of staff rather, and then they will now be able to apply for it. So we're looking at having, for a start, around 5 scientists. It's going to be simple. We shortlist the scientists, and then they will be able to have like a rotation in one of the big African institutions or centers that does high grade or high level genome sequencing with very good facilities and most of the genome technology facilities and infrastructure. Perhaps they'll spend around 1 month, and then they will now be able to spend maybe at least 3 months outside Africa, working with some of the best and also top genome centers as well. Together with other people. So the idea is that by the time they return back to their institution, they could potentially be able to commit to curriculum development or research programs. And one of the criteria is that they'll be able to get commitment, hopefully from their head of the institution, either through support or through commitment that they're going to provide them the support to be able to deliver on the curriculum. So this is going to be a bit of a comprehensive program, and it will evolve gradually, and it's going to be very, very dynamic. But it's also going to be restricted to only institutions participating in the practical session. And to participate in a practical session or host a practical workshop, just as we said, and Professor Ann mentioned, even pipetting and DNA extraction, these are valuable skills. You don't have to have a genome sequencing machine like thousands of dollars. No. And once you are able to participate in these regional workshops, you will now be, your students or the students in your institutions will now be eligible to take part. So at the moment we're going to start with just 5 opportunities, 5 number of applicants, 5 number of fellows. But I see that number increasing over the next few months, because we're also in discussion with several other partners, to be able to have more numbers added to it. So I think that is just more of a summary let me see if I'm missing out on anything. I don't think so. So again, that is just supporting what Professor Julian has said. And I'll be happy to answer any questions. And yeah, thank you for listening.
So thank you very much. Thank God. Thank you very much, Professor Julian. I think it's important to also mention that you can follow this information on our website. We are on LinkedIn, we are on X, we are on Instagram, and by everything that we're doing, we're trying to make sure the dissemination is through this fora. And therefore, if you follow us, you'll be able to get this information. But for you as a young early career scientist, the easiest way is to be part of us. So apply through the website, go to our website, click on the contact me and ask to be joined, and then you'll be immediately automatically be put on the mailing list, and therefore none of these opportunities will miss. And we also disseminate information from our partners and our sponsors who are offering different opportunities, people who have conferences and they are partner with us. We are able to share that information. So you're not only getting information about AfricaBP, but you're also being kept abreast of some of the trends that are happening worldwide. I don't know whether there's any elaborate burning question. You remember that for any fellowship, it's very difficult for a fellowship to cover you 100%. So you might be asked to pay for your medical or look for an air ticket here and there, but the bulk of the training itself will be fully covered by others. So we also ask you and beg you to, dip your hand in your institution's pocket, ask your institution to pay your air ticket, to continue putting them on the medical cover, so that at least that way we can be able to take you. But if you cannot take yourself to this particular lab, then it will be very difficult for us to train you. So you look for some of those monies from even other agencies around and other fellowships that are around to support small things like medical, like travel, like, visa fee application, those things. I think if you are able to find someone who can do that, we can be able to do the rest.
Yeah, so just to add, Professor Mugai, so while we would be happy to, provide 100%, sometimes you need to have these discussions and it takes a long time, and, they are just, and so it's just, it's not Anne. I'm sure if Anne is the richest person in Africa, she will probably dip her hand and pay 100%. So but we'll have to approach partners, and then we have to speak, and then we'll have to come to a consensus. And for me, I think this is actually a good will I say deal, or a good opportunity. We know we would have, be able to do 100%, but this is gradual. We're just going to evolve over time, and hopefully we'll get to the 100%. But we'll look at what is covered. You spend 1 month, and all the reagent, consumables, compute resource, everything is covered. But then the travels, just as I mentioned, and a few other things are not covered. But I'm hoping, because most institutions in Africa have little funding to cover some of those things. For instance, I've seen for this practical workshop, I've seen quite a number of people in their institutions who want to attend in another location, and they needed our advert to apply to the national grant agencies just for the travel grants and things like that as part of it. So we'll just continue evolving, and by the time we get we'll come up with a discussion or finalize the discussions for 100%, we'll also advertise that. So we'll just advertise as, these things evolve. But I think for us, more importantly in the project, is to get one genome sequenced through this workshop, through this fellowship rather. So each time we have the fellowship, we get 10 African scientists, all trained end to end. We want to publish one genome. And you can't tell what that does to the fellow. You also can't tell what that does to their institution, especially if the institution were able to cover the travel cost. So it will also be helpful to see that the institutions can cover little things here and there. I know it's very difficult, challenging in Africa, but as we evolve, we also need African institutions to support in that direction, and push some of their staff so that they can get this training, return back, and then be able to apply for large grants, to cover these things. So it's a bit of a challenge rather, I must say. Thank you, Professor Mugai.
Thank you very, very, very much. And I would like now to invite Dr. Catherine Ziomo to give us a vote of thanks. We have come to the end of this first phase of the African BioGenome Project Open Institute for Genomics and Bioinformatics workshop. The next 3 days, of course, will be hands-on training. And I want to thank you all, and to do a better job, let Dr. Catherine come forward.
Thank you, Prof. Ann, and thank you to all of you for being here. I would really like, before I say the vote of thanks, I just want to talk about the workshops tomorrow. So we have 3 workshops that will be happening here, both in the labs and also in rooms. So the one being led by JB ONT will be in room 720. That's the room that you see when you get to the reception. And the Illumina one will be in Borana room, with the room by the cafeteria. And the one for Inkaba will be in Dama Lounge, which is next to Borana. And for the labs, I mean, the sponsors will work out the logistics on what time to start, but those are the rooms. To those who have been selected for the workshops, for the practical sessions, congratulations. And I hope that that can also lead to the fellowships that that Thank God and Prof. Julian were talking about. So now that we have that out of the way, I would like to, first of all, thank our Director General, Prof. Polone, for giving us this opportunity to host the AfricaBP. A month ago, we did not think it was possible to have it here because of the logistical nightmares, but because of his support, because of his commitment to seeing this happen, to seeing the AfricaBP become a reality, he made this possible. So if we can just have the round of applause for him in absentia. And also for being available yesterday. He has traveled to Dubai for COP28, but he made sure that he was physically here yesterday for the workshop. This just shows how committed, or how he wants this to succeed. It's an ambitious idea, but when you have the support of your leaders, of your institution, it makes the job very, very possible. And with that, I would also like to thank him for allowing staff, ILRI staff and my colleagues to be able to be a part of the AfricaBP community. It takes hours, it takes time to have something like this, not only this workshop, but also all the planning, all the thinking, all the ideas that are coming together for the AfricaBP community. And so it takes leadership that is really seeing the vision, that is really seeing ahead of time, to have your staff be a part of this. So I'd like to also thank him for that. And we have all been fed for the past 2 days. I'd like to thank our ILRI for preparing the meals and making sure that everything to do with the meals and the logistics happened on time, and also it was possible. And most importantly, I'd like to thank the ICT for staying with us even up to this time. The workshops on the schedules were ending at 5 p.m., but it's now after 6 p.m. and they're still here with us. So thank you very much for making this possible. The streaming and everything that has happened, we did not have any technical glitches. And I think the people online can also attest to that. And to all the presenters who also sacrificed their time, there were time differences. You know, you have said I will have this talk at 2 p.m., but because of the changes in the schedules, they ended up having their presentations 30 minutes into the time. But still, we didn't miss any presentation. And I'd like to just thank them for that flexibility and also adjusting to make this possible. So if we can just thank our presenters. Now, having something like this takes funding, takes resources. And I would really like to deeply thank our sponsors for contributing to this workshop, and also to the AfricaBP. As it is, we're still fundraising or putting together, mobilizing resources that are required to have the fellowships to also have our work going on. But our sponsors have really devoted some resources. So I will name those that have exhibited, that have also contributed financially to the workshop. I'll start with Inkaba, Illumina, Science for Africa, Change Biotech, MGI, FNS Scientific, Bio and Imaging Technology Institute, the University of Addis Ababa, the University of Yaoundé, and the National Animal Genetic Resources Center in Uganda, the University of Kinshasa, and also Thermo Fisher. Thank you very much. If you can just thank our sponsors for the resources that we have had. And I especially want to thank all of you, those who are online, those who are physically here, who have made it to the workshop. You know, you can plan a wedding and no guests show up and it becomes a flop, but you have really made it successful. We had more than 400 people online yesterday, and those who consistently stayed throughout the 2 days, and you who are here, we have all been together. I think this was really a successful workshop, and I'd like to thank you all for being here even up to this point. Thank you very much, and a round of applause to you. And I hope that through this workshop, it has given you insights into how the AfricaBP operates and how we can form this network, strengthen it in terms of building infrastructure on the continent and the capacity in human form, the people, the students, those who are aspiring to also advance their careers in bioinformatics and genomics. This has been a platform for you, I think, to also learn from industry, from technology providers, learn from professors, and learn from other people, even ILRI staff who have been here. I hope this has been useful for you, and I hope we can continue to build this critical mass of scientists who have the knowledge, who have the understanding of manipulating genomes and protecting biodiversity. Finally, I would like to thank the team that has been behind the scenes. It really takes a lot to have this, and I'd like to first of all start by thanking Dr. Thank God, who is the engine that has brought us all together. If we can just thank him for his drive. I don't know how Thank God does it, but he does spend a lot of time on this. And I'd like to thank him so much for this. And if we say Dr. Thank God is the engine, obviously there is need for the fuel for it to move, and Prof. Anne for really making sure that this happened. Would like to really thank you and your team for all of this, for the time that you have put. I would also like to thank Dr. Josiah Kuja, who couldn't be here, but has put in a lot of times for the planning, a lot of hours, and a lot of time into making this happen. We also have Mr. Paul Kagame, who also contributed immensely as we were planning for this. And my colleague, JB, who was also part of this planning committee. So it's always the team behind the scenes that puts in the most, that puts in the hours that are required for this. And with that, thank you so much. I think this has been a successful workshop. We will continue communicating online on our social media platforms, keep following us, and also contributing to this network. Thank you.
So Dr. Catherine has forgotten to thank herself. But she was, in the height of all the stress and pressure, she was the calmest. She'll be like, what needs to be done? Don't worry, it will be done. And I want to thank her so much for, being the link to the DG, who's been extremely busy and extremely supportive, but she got a lot of the doors opening for us here at ILRI, and I'd like us to give a round of applause. So thank you. And I think with that, we close the 2-day workshop, and we look forward to seeing those of us who will be here tomorrow in the morning. Thank you all.